Good morning, everyone. It's a great pleasure to welcome you to this 7th Precision Oncology Symposium. I'm the chair of the Comprehensive Cancer Center. Markus Manz is my name. I have the pleasure to open that together with Markus Rudin, who's chairing the Loop here in Zurich. And as the Comprehensive Cancer Center, we carry precision oncology in our name. And I think today we'll hear what this is about. My personal impression is that it's absolutely stunning how we are able to catch a lot of data. It's also absolutely stunning how the data can be utilized in terms of integrating it. And it's absolutely stunning what type of hope we have that it will materialize also in the clinics. And there, I think we are waiting for the big delivery, which I'm convinced will come. But this is really in the making. And for this symposium, we have a lineup of fantastic speakers that are all involved in this process. And we are really looking forward to seeing what and hearing what you bring here. And we like to learn. We like to interact. And I think this is very important. We have these in-person meetings. We also have a lot of online participants. I don't have the actual numbers. Do we have what do we have in terms of online attendance perspective? Around 40, okay. And here we have a lot of people here, even at these early hours. So I'm really looking forward to that. I'd like to thank the organizers. I think the main involved organizers are Thorsten Zenz, Bernd Bodenmüller, Jean-Pierre Bourquin, Andreas Wicki, and Markus Rudin, and of course, as always, Silke Uflacker, who is holding the strings. I'd like to thank the sponsors that are indicated here. We all know it's difficult to organize these meetings without having the respective support, so we are very thankful for that. I'd like to note, of course, also that they had no influence whatsoever on the program. Nevertheless, I think they trust that we will work together with them for the benefit of the patients. And lastly, I'd like to thank you for coming. I'd like to thank especially those that traveled far, lots of the speakers for being here, and I'm looking forward to that day. And I pass on to Markus Rudin.
Yes, thank you, Markus. It's also my great pleasure to welcome you here on behalf of the Loop. As Markus mentioned, the Loop is actually a cross-institutional center focusing on precision medicine. And if you talk about precision medicines, of course, oncology is very high on the scale. And in fact, we are supporting several oncology programs. So I'm not an oncologist, and therefore I'm really honored to stand here in front of you, and I'm looking forward to an interesting day. And probably I quickly go through the day. Can you show the next slide? Oh, it's already on. So we have 4 very interesting sessions, starting with the beginning, which is diagnostics, then translation into the clinics, and finally, and that's also something we are very much involved in, data. We are building up a data platform, collecting the data of the various institutions, hospitals, which are part of the Loop. I have been told that we have a very strict time management. So we have lectures of 25 minutes followed by 5 minutes questions. And we try to be as strict as possible, which also refers to me. So without further ado, actually, I would like to hand over to the chairpersons of the first sessions, which are Bernd Bodenmüller and Bettina Sobotka. Please.
Yes, good morning, everyone, to this first session on diagnostics and technologies. I think, as we heard, we have excellent talks lined up. Our first speaker is Professor Berend Snijder. He is faculty professor and group leader at the Bunne Institute of Immune Engineering at ETH Zurich. His research combines high-throughput microscopy, machine learning, and multi-omic profiling to systematically analyze patient biopsies, pioneering image-based drug screening called pharmacoscopy to identify effective personalized cancer therapies. Berend, we're very much looking forward. And as you heard, we're looking for, the time is very important.
Time is important. Good. Time is important also in the clinic, as you know, so it's a perfect fit. Now, good morning, everybody. It's a pleasure to be here today, present you a bit on our work on personalized drug screening and multi-omics. Do I have a clicker? Yes, there you are. Perfect. My disclosure slide, I'm a scientific co-founder and shareholder of 3 past companies, all site provision and graph, and inventor on some of patents relevant to the work today. So, precision oncology, despite amazing progress, I think it's still very clear to everybody here that we still need better ways to identify effective therapies for individual patients. And the example I like to show is this recent, I mean, not too recent, but this nice phase 3 pivotal trial for the treatment of relapsed acute myeloid leukemia, so this blood cancer, where patients have received either salvage chemotherapy, the standard of care at the time, or if they carry the FLT3 mutation, a targeted therapy that specifically inhibits that drive mutation. And the trial was a big success. What you see is that patients that received genomically matched precision therapy lived several months longer. What you can also see, though, is that the majority of these patients had died after a number of years. So despite amazing progress, there's still a lot of room for further improvement. And if you look at sort of the state-of-the-art treatment guidelines, as for instance collected by the NCCN, what you see is that trial and error is really intrinsic to the treatment process of cancer patients. Generally, there are evidence-based treatment guidelines that try to, based on the best evidence available, come up with the best treatment protocols. But already soon after relapse, clinicians face multiple choices. Do we give one or the other treatment? And how to choose between those treatment options is really not obvious. So a very simple approach to just cut through this trial and error process could be functional precision medicine. Why don't we just take a biopsy from patients, throw on the drugs that they might receive in the clinic, and see in the lab which drugs work best for this individual patient? That concept is not new at all. It's been explored for, let's say, 80 years now. Nonetheless, it really has not transformed clinical care in any way. And I would think one of the reasons that is because it's very easy to kill cancer cells in the lab. You can withhold the medium, you can starve them, you can throw a brick at them, they will die. Death alone cannot be the answer for functional precision medicine. And that's nicely depicted by one of my favorite cartoonists, web cartoonist Randall Munro here. So one way that we try to overcome this limitation, we have to do more than just look for killing alone, is by coming in with single-cell image analysis. And for us, we achieve this through automated microscopy. So patient biopsies, patient cells go into this kind of high-throughput drug screening plates, incubate with different drugs or drug treatment combinations. And then after a certain incubation period, we fix and stain the cells and image them with automated microscopy, capturing every single cell and how they responded to the possible treatment options for this patient. Now, in pretty much any biopsy, you will not get 100% pure cancer population or 100% pure healthy population. These are typically mixtures, complex mixtures. So the single cell resolution now allows us to overcome this. Let's just find something that kills everything, and instead allows us to look for something that specifically kills the cancer cells while leaving the patient's own healthy cells alive. So that builds in a patient intrinsic toxicity measurement. Do the healthy cells of this patient survive this concentration of this particular drug, etc.? And that's a big theme of research in the lab. We've had a number of papers on this over the past years, and I'll highlight a few of those. The platform, though, has more utilities. It's, of course, also a powerful immune system profiling platform. So we can look at spatial interaction patterns from these kind of high-throughput images. We can look at cell morphology. We can have more advanced readouts, also showing how the patient's own immune cell acts. Another way of looking at this, and I apologize to the Zurich crowd that might have seen these slides like 100 times, but for the external guests, another way of looking at this platform is a single 9 milliliter blood draw allows us to perform 384 well plates. So that is 1,000 chambers in which we can test drugs, drug combinations, antibodies, cell-based therapies, and how they might act on the cells of that particular patient. We then analyze this with automated automated microscopy and single cell image analysis, generating these kind of rich images of human tissues. And of course, this requires quite some computation. These data sets are very large, and they require advanced image analysis to identify each individual cell, see its identity, state, and response to possible therapy. And amongst others, we've developed a self-supervised dyno-trained vision transformer algorithm that we call SE-dyno to, in an unsupervised way, help characterize all the individual cells in these images, quantify their state and response to therapy. So once you have done that, identified each individual cell, and you know whether it's a cancer cell or a T cell or a B cell, you can then, for instance, transform these images in these computational graphs. That allows us to do a number of fun things. I will not talk today about the immune research, but just highlighting some recent work where we discovered that T cells actually, for the majority, look stripy, not conventional, that matters for T cell biology, or that we can analyze systematically how the immune system is wired based on protein-protein interactions of the surface of immune cells. I will not talk about that. What I will talk about is the much simpler use case where we just look for something that specifically kills cancer cells. So that is basically beam counting, or almost like a flow cytometer, but a high throughput one. So the main readouts in this kind of precision oncology screens is then what is a possibly effective drug that is well tolerated by the healthy cells of the patient and that nonetheless specifically kills the cancer cells, which are drugs that don't do anything, no specificity, no activity in this particular patient sample, but also uniquely with this platform, which drugs are toxic, which drugs leave the cancer cells alive and kill the patient's own healthy cells. So this kind of rankings of all of these possible treatment options tested for this patient sample is really at the core of everything I'll show you today. And this platform has been used in this way now in the clinic for over a decade. We started this work first in Vienna, where we developed a platform, and then clinicians approached us and said, hey, you can screen drugs on blood samples and lymph nodes. Here we have a patient. So already back in 2015, we received a small lymph node biopsy from this particular patient, an elderly man with a diffuse large B-cell lymphoma. We tested 104 possible treatment options, single drugs, and ranked them based on this kind of relative blast or cancer fraction into those drugs that are off-target or toxic, don't have effect, or significant on-target effects. There were really only 6 of those drugs that we tested that looked promising. We reported it back to the clinic. They identified our second best hit, ibrutinib, to be a promising treatment for this patient. The patient had relapsed with standard of care, chemo, immuno, and this patient had a long-lasting complete remission to this single monotherapy, ibrutinib. So it was these kind of early insights that really driven a lot of the clinical translational interest and translational studies. We have since performed and reported 2 non-randomized interventional studies. One was called EXALT-1, where over 50 late-stage blood cancer patients all received personalized therapy based just on this drug screening platform. And when we followed these patients for several years, what we saw was that a subset of these had significant long-lasting progression-free survival that we called at the time exceptional clinical responders. And here, the red line, the comparator, is the patient's own response, progression-free survival, to their prior treatment earlier in the disease stage of these patients. And we could significantly improve over that. In parallel, here in Switzerland, we ran a small feasibility study called DART-1, where for late-stage acute myeloid leukemia patients, we then performed screens. About half of these patients could receive personalized therapy. These are combination treatments mostly. That then we followed over the next month. And we saw that the patients that could receive pharmacoscopy-guided therapy, which was all healthcare reimbursed, by the way, lived on average 3 times longer. So these were really promising early signals but from non-randomized clinical trials. Over the past years, we've been working very hard to be able to sort of breach that gap and now be able to run randomized and controlled interventional clinical trials based on this platform, where patients either receive standard of care or pharmacoscopy-guided treatment. And the first trial where we achieved this is the trial that's currently running, which we call the RAPID-1 trial, together with University Hospital Zurich here, particularly Alex Theocharides, where after AML patients relapse to their first-line chemotherapy, they are eligible to enroll into the trial, get analyzed by pharmacoscopy, and then randomized to either receive standard of care, where the clinic does not get to see the pharmacoscopy report, or randomized into the other arm, where the clinic gets the pharmacoscopy report and the treatment of this patient can be guided by these results. Endpoints: complete remission, complete response rate, and secondary composite response, but also the fraction of patients that we help to bridge to receive a bone marrow transplant, which can be curative in the setting, and safety, treatment-related mortality readouts. Now, in the standard of care, as before, this would be typically salvage chemotherapy. Of course, all the standard diagnostic tools are available to the clinicians, so targeted therapy is also still an option here. And in the pharmacoscopy-guided treatment arm, what we're now recommending is a panel of around 20 different drugs and drug combinations that are all comprised of AML-indicated, AML-approved therapies that can be reimbursed by the health insurance. This is an ongoing interventional trial. I cannot report on any outcome data, but I'll take the liberty to report a bit on some technical performance interim. What we see is that there's a 95% technical success rate in this trial. So for every 20 samples that we get, 19 we can report a successful drug response screen for. Very encouragingly, for being extremely fast, from sample to report in the hands of the clinicians is just 3 days. And from sample to treatment start is a median of 7 days across the first patients. And that is no different between the interventional arm and the control arm, really suggesting that it does not take extra time to make that decision to personalize these treatments. So we're very excited about this trial and hope that, give it a year and a half, we can report outcome data here. So what about solid tumors? Asks everybody ever. Well, that was the big question when I started my assistant professorship lab at ETH. And I just want to give you a bit of a high-level overview of this. How are we doing with time? Is there a timer somewhere? There's a timer. Good. And I would highlight first some of the amazing work done by So-Yeon Lee, Toby Wise, Emily Leroux, and Michael Weller. Of course, the latter three here at Neuro-Oncology in Zurich. Adapting this platform to the use of studying glioblastoma being the most aggressive primary brain tumor in adults, still 2-year median, less than 2-year median overall survival. And when So-Yeon started to receive glioblastoma surgery tissue samples from the clinic in the lab, really what she set up was much the same as what we'd already been doing for our blood cancer tissues. A mild dissociation, if needed, which of course for glioblastoma it is, directly going from surgery onto the drugs. So these cells go directly onto these drug-screening plates. So there's no filtering, no purification, no pre-culturing. And then incubation, 1 or 2 days, and then staining, automated microscopy, and single-cell image analysis. So what we're doing here is we're maintaining that full tumor complexity. Whatever we got from the surgery, we basically put into these wells. By using smart marker profiles, we can try to identify glioma stem cells to the extent that they exist and see if we can find drugs that kill this more sort of plastic population of cells. Again, the patient owns healthy cells provided toxicity control. And this process is fast and automatable, so we can return results within a few days. The images we started to get out of these were super exciting. So in the background, you see one of these 384 well plates, entirely imaged. And that is at the core, this kind of single cell image data. So you see this, for this particular patient, this highly complex, spindly yellow and green are glioma markers, STEMI glioma markers, expressing glioblastoma cells scattered in small clusters, also interacting with the patient's own immune cells, here stained by CD45 in red. So we can perform the image analysis, quantification, and again, just prioritize which drugs specifically kill the patient's own cancer cells while being tolerated by the immune system. Now I'll just summarize this. What we found here was that this approach could predict which patients would respond well to their chemotherapy, temozolomide. It's one drug, it doesn't quite help these patients, but it still shows that the platform is predictive. We then performed a drug repurposing screen. So we identified new drugs that are already existing but not used for glioblastoma that are now going into clinical trial to see if they might actually help. And our particular candidate here is a drug called vortioxetine, an atypical SSRI. Using graph-based machine learning, we identified the new mode of action underlying these drugs. And like I mentioned, the validation clinical trials are in advanced stage of preparation. That is sort of the preclinical validation of this platform. It does not show precision medicine with this particular platform, but here's a nice example of a patient from a few years ago here in Zurich that was in the clinic with Michael Weller, suffering from an atypical plexus papilloma, a fairly rare disease, typically benign, but for this patient so aggressive that they found that they needed to intervene more systematically. So we received a very small needle biopsy from one of these lesions around the spine of the patient and could test some 20 or so drugs on this. The second best hit that we got was a regorafenib that the clinic was sort of intrigued by because, one, they're familiar with using it for the treatment of glioma in some contexts, and because of its targeted profile and relative safety. So this patient went on regorafenib monotherapy and has now been experiencing over 3 years of pharmacoscopy-guided sort of tumor control. So you see the lesions shrinking. They're not entirely gone, but they're stable so far. That's a highly encouraging N of 1 result for a disease for which there are no systemic therapies. So based on that and other preclinical work, one of the things we've now been able to do, again with Emily Lerin and Michael Weller in Zurich, but now also with clinics around Switzerland, is to start the evidence BM trial. So that's an interventional randomized trial performing pharmacoscopy-guided therapy selection for patients with brain metastasis of any kind, whether it's a lung cancer, colon cancer, reaching the brain. As soon as these patients are eligible for surgery, and we can analyze their tissue, they are then eligible to enroll into this trial. And what is unique in this trial is that we've now been able to match not just randomized interventional trial setting under IVD regulation, but also with 57 off-label treatment options for these patients. And that is, from a regulatory perspective, one hurdle higher that we've now been able to break through. And we will enroll 102 patients in a 3-to-1 randomization scheme. Now, these are basically single platform trials. All the routine clinical diagnostics is available to them, to the patients, to the clinics, in their treatment decisions, and we're just adding pharmacoscopy. Of course, the future is not single platform, but regulatory hurdles exist in making these kind of randomized IVD trials multi-platform, because you need to show technical performance evaluation on all of them, especially if these are new experimental platforms. And together with the Tumor Profiler Consortium, I'm happy to say that we've been sort of now over the past years performing the preclinical work to look at feasibility of massively multi-omic tumor profiling, amongst others for the treatment of melanoma, also leukemia and ovarian cancer. And we recently had a publication on this from the consortium where we could show feasibility of multi-omic tumor profiling to guide treatment of melanoma patients, and even reported encouraging response rates amongst late-stage melanoma patients based on this kind of integration of massive personalized data, both functional and molecular. And that's for melanoma, but also for leukemia, it looks very encouraging. So I think the future of these trials is, of course, not single platform. It is about how can we integrate these platforms? How can we make sure that all the right data is available to the clinic in making these decisions? So just a quick overview of the current interventional trials that are ongoing in the lab or close to ongoing. Not the published ones. I've mentioned the RAPID trial, our sort of randomized, 1-to-1 randomized AML trial, or flagship trial. We've now started the evidence BM study here with neuro-oncology. We're close to starting the same sort of sister trial to evidence BM called Evidence BT, specifically performing personalized treatment for patients with primary brain tumors, glioblastomas, meningiomas, etc. We're part with the Tumor Provider Consortium of the Zurich Precision trial, where really multi-omics data is going into personalized treatment for ovarian cancer patients. And we're performing a small feasibility trial, multiple myeloma, looking at personalized immunotherapy. Now, before I wrap up, there's usually, whenever I go anywhere, I get the same questions. So I started to make a frequently asked questions slide. And one of my favorites is this. The whole field, multi-billion institutions are investing in complex 3D tissue cultures for tumors, right? And that's beautiful science. So that then turns around to the question to me, but hey, you're just dissociating these tissues. Don't you need like Matrigel and 3D culture and prolonged in vitro growth before you can measure anything? And I would turn it around. I would say, well, I think actually that we're screening microaggregates for the majority of parts, for the bigger part. For instance, shown here on this lung cancer brain met, this primary glioblastoma, or this beautiful primary colorectal cancer tissue. So we're screening microaggregates in a high throughput way with single cell resolution. I'm having molecular and functional readouts at the single cell level. And what we're not doing is prolonged in vitro culturing. I think prolonged in vitro culturing gives a big chance to cells to adapt their metabolic transcriptional state, to give bias into which cells survive and which cells don't survive ex vivo. So that's what we don't do. Another one, can you do drug combinations? And I would very much say yes, and we're doing so in clinical interventional trial settings, as I've shown you for Rapid. Can you do immunotherapy? Yes and no. So we're in a multiple myeloma setting. We started now a small feasibility study on personalized immunotherapy combination treatments for myeloma patients. We have preclinical experimental work with collaborators on CAR-Ts, bispecifics. These are all established and published, seeing interesting personalized responses. What we do not have so far are predictive responses to checkpoint inhibitors. And I sort of personally am doubtful to what extent that will ever work. And then the big question is, will this scale? Like, it's great we can run this in the lab. You have an amazing lab, probably, right? So nobody else can do this. So that has been a big effort over the past years, where we've developed now a scalable solution, both an experimental platform and a cloud-based analysis solution that can really be distributed across labs. And one of the things we're now doing with the Bunne Institute is actually going across the world, seeing if we can establish this platform at hospitals, at cancer centers around the world, to see if we can help guide patient therapy. So with that, I'd like to come to the acknowledgements. Alex Theodoridis, Markus Manz for the Rapid trial, Michael Weller, Emily Lalonde, Tobias Weiss, and Berend Snijder, amongst others, for all the neuro-oncology collaborations, the Tumor Provider Consortium, too many to name, and the funding agencies, of course, the ETH originally, the ERC, SNF, now the Bunne Institute, but most importantly, the people doing the work. I owe them a great deal, and some of them are here in the room. So thank you, and I'm happy to take questions.
Thank you, Berend, for this excellent talk. Are there any questions from the audience? I'll start here. Oh, you have, yeah. Thank you. Good.
It's high tech. I would have no clue how to do this, but we'll figure it out.
Okay, sorry. Thanks for the presentation. I was just wondering, because you showed basically for the glioblastoma that the context matters, but then for the AML, you said you can just draw blood, but then you're not getting all the bone marrow interaction. And did you also compare when you profile based just on drawn blood versus a bone marrow extraction?
Yeah. In fact, the Rapid trial, we do both by default. So we analyze both separately in parallel, a bone marrow biopsy and a blood sample, if they're both feasible.
And do you see substantial differences?
Well, I would turn around to say we see substantial similarities, which is a great start. And then, of course, yes, we do see differences. Can I say something smart about the blood-bone marrow differences we see within leukemia? The healthy cell populations do matter a lot. So any kind of monocyte population will be intrinsically very sensitive to proteasome inhibitors. So if what you consider to be your healthy counter population is rich of monocytes or macrophages, same from this perspective, you will get super high toxic readouts for anything called proteasome inhibitors, which you don't see as soon as your counter population is mostly T cells or B cells, lymphocytes in this context. So definitely there are reproducible trends, but those are mostly on the toxicity, and that it's really about the sort of overlapping on-target efficacy. And there, for the majority, at least in leukemias, we see very good consistency. Yeah. But the composition of what we call the healthy population is a big determinant, especially if those cells have strong sensitivities themselves. And proteasome inhibitor sensitivity of monocytes is one of those I would highlight. Yeah. But there's probably more systematic analysis we could do here. Yeah, definitely.
Yeah, Berend, I'm very excited about the ongoing trials, and you mentioned them. You also mentioned that a frequently asked question is 2D sufficient, or do you need to go 3D? And another element that is always in question is the element of time. Right?
Yeah.
So this is a limited time observation.
Yeah.
And my more general question is, are you favoring certain drugs that where the effects can be observed in short time versus drugs where you wouldn't necessarily see that? And do you have evidence for that?
Yeah. I think there definitely is a bias that cytotoxic drugs tend to come up more out of this kind of fast assays because they have the potential to kill rapidly. I think we have very nice counterexamples of drugs that should be really slow-acting, hypomethylating agents like azacitidine, repeatedly having very fast on-target toxicity in this kind of ex vivo assay for leukemia patients, which we've been able to replicate and even clinically translate. Such that, a drug that's called a hypomethylating agent that should have a slow epigenetic effect and not per se fast killing efficacy can also have fast killing efficacy on its own. So that's, I think, a nice counterexample. But in principle, yes, there will be false negatives, drugs that we don't recommend that might also have helped those patients. But that's a problem I can live with, right? As long as the drugs that we do recommend work for the patient. If we were recommending false positives, and a lot of the longer-term 3D cultures introduce bias. You lose your healthy population. You don't know what is toxic to this patient, and in fact, you're making a clonal selection typically. That can lead to false positives, and I think false positives in a functional precision medicine regime is a way bigger problem. If we're recommending bortezomib for everybody, which honestly some of these functional platforms that's what they do. They recommend bortezomib for everybody because it's just highly toxic. That is a way bigger issue. So I think it's more important for us to protect against that concern than to have a few drugs that might have worked that we miss. But it's a trade-off. Yeah.
Yeah, Berend, thanks a lot. So I have two simple questions, if I can. So your group, you have, I would say, most experience in the world on multiple different tumors: melanoma, ovary, AML, lymphoma, glioblastoma, sorry, head, brain metastasis. Yeah. So for which disease does this work best?
Yeah. There is a ease of use for the blood cancers compared to solid tumors, just from a practical point of view. That is very clear. And on the one hand, that may be practical, ease of sample access and reduced tissue handling before you can analyze it. It may also have to do with the types of therapies that are typically given to these patients, not per se the most complicated checkpoint inhibitors, but if it's an antibody, then it's a targeted antibody, or if it's a therapy, it's either targeted or chemotherapy. So that is a nice combination of reduced sample handling and a good compatibility with the types of treatments. If you go, for instance, to melanoma, where the majority of patients a priori receive checkpoint inhibitor therapy, and I've already said, I think we don't have a checkpoint inhibitor assay right now. That immediately excludes a fairly large fraction of these patients and being able to predict their response. So that limits our ability to say whether the platform is predictive or not. Then what you need to do in those settings is still run actually feasibility trials or even interventional studies to see, do the drugs that we do recommend translate to clinical benefit? So I think there it's just less conclusive right now, and we need to do the studies to figure that out. I do think there is that, there's two factors, right? Compatibility with the standard of care drugs is an important factor. We don't have it for checkpoint inhibitors. And there is something about tumor heterogeneity and tissue heterogeneity that we're also explicitly addressing. That is an extra complicating factor for solid tumors in general. And glioblastomas might be, from a spatial body heterogeneity, it's of course less of concern. Tumors are typically comprised within the brain. So there's a lot of stuff to be figured out still. Yeah. Not easy questions.
The second question is what challenges are you facing?
Do we have enough time?
We have time, Berend. I think you need to, or may repeat the question a bit because the online audience.
Yeah, no, I'll repeat the question. That's fine. So the question was, how far are we with IVD, in vitro diagnostic certification, and what are the hurdles we're facing? Right. So I don't quite discuss this because this is where we've shifted the work to our commercial entity, Precision Medicine. So I'm wearing a double hat here as a co-founder of this company. The first hurdle is I think, as an external lab, not an LDT, not a lab-developed test, it's practically impossible to thoroughly develop this into a CE-IVD certified test in an academic setting. So the first hurdle is you need to spin this off and put it in a commercial setting. That commercial setting needs money to survive, which on its own is a big challenge. And then, if you look at the current funding environment, diagnostics are not considered attractive by investors. Investors want big bucks with exciting stories. That is drug development, drug discovery, not diagnostic test development, where you sell one test and then you need to repeat it and you get a little bit of money. So the commercial attractiveness of the diagnostic business is a big hurdle, I would say. And that's not even getting to the regulatory. For us, we are now running these IVD performance trials academically, which is great because it means we can somewhat take away the hurdles for possible commercial CE-IVD certification. Hurdles are use of AI in these kind of diagnostics is still very new, so regulatory bodies don't have a lot of experience with that. And another big one is this is not one test to measure one molecular property of one tumor type to predict one drug response. This is a general test to measure many drug responses on many different tumor types. And that doesn't fit any of the traditional regulatory framework, so that's a very big one. I would say those are probably the key points, pain points. Oh, and how far are we? The company, if I dare say, the company is probably close to getting CE-IVD on the software. And that is a very big step because now we can simplify the platform and at least pair it with CE-IVD grade software for the analysis of this kind of data. So we're getting there. Give it another few years.
Right. Thank you very much. I think there have been more questions, but we have a coffee break and you're welcome to talk to Berend then.
Coffee.
Thank you very much again.
Thanks. Thank you.
Okay, so now it's my great pleasure to introduce our next speaker, Daniel Truhn. He's a professor for radiology at the RWTH Aachen at the University Hospital of Aachen, and he oversees the AI group for medical imaging. And in his presentation today, I assume you will show how you use clinical radiology, advanced imaging, combined with AI tools to improve precision medicine diagnostics. So, looking forward.
Exactly. Thank you very much for the invitation. So my talk is called Precision Oncology in the Age of Agentic AI. So I'll give you at first, and also in the session, which is a bit about the technical foundations. So I'll give you a bit of a background on the technical foundations. And I want you to walk away from this presentation with a bigger understanding of what we actually can do with AI. So these are my disclosures. I'm also partaking in 2 startups and have done some lectures for pharmaceutical companies. And before we dive into the agentic setting, let's take a step back and look at how it all started. That's about 15 years ago or so. And we recognized that neural networks, in this case, particularly convolutional neural networks, are really good at detecting patterns in images. So you can try and give it a go and filter the odd one out in these lines. And I can guarantee you neural networks are just better at doing that and much faster indeed. And this, of course, then translates to some parts of medical diagnostics. So, for example, in radiology, there's always been the talk of us radiologists being replaced at some point in time. And indeed, in this study, there's a very specific instance, very specific examination, the mammography screening via X-rays, in which this is becoming reality. So in this study, what you usually do in most European countries anyways, is you take an X-ray of the breast and you have two radiologists looking at that X-ray. And then independently of each other, saying whether there is some suspicion of cancer, yes or no. And now, naturally, this extends to including AI. So if you have an AI model which is trained on these images, and this case is just perfect because you have millions of images, these images are highly standardized in the medical setting. So if it doesn't conform to certain specific image criteria, they are repeated. And they are 2D, not 3D, not 4D like MRIs. So AI models are really good. And if you do that, the actual surrogate for how well this algorithm performs is the cancer detection rate. And it's similar in both settings. So this will become a reality very soon, I think, that radiologists will be replaced in this specific setting. This was radiology. And now images are also looked at in other settings, for example, in histopathology. And in particular, if you look at the evolving landscape of cancer therapies, those are getting ever more complex, right? Everybody's speaking about personalized medicine. And if you look back like 15 years, therapy was comparatively simple. And now you have to test for all these different genetic markers in order to decide for the best therapy. That has led to improvements. So therapy now is much better, but it's also more expensive and more complicated in the sense that you have to undergo a lot of different tests. And this can be substituted in some way, at least, through image biomarker detection in histopathology images. So because if you have a genetic change, this also translates to a morphological change, usually, and this might be sometimes visible or sometimes not to the human eye, but neural networks are really good at detecting patterns. Basically, it's the same task as the one that I've shown you before. And you can then actually detect biomarkers. This one was one of the first publications back in 2009, which showed that you can detect microsatellite instability from these. So in images, this works very, very well, and we'll see a lot of applications in that. But of course, in the medical setting, you have usually more than images. Even as a radiologist, I'm also often looking at the clinical data. It's not only about images. So you need a lot more if you want to really replace medical doctors, or if you want to support medical doctors in a reasonable way. And now the question is, where are we headed? And I want to give you an overview over the developments of the past 2 years or so, because that's really exciting. It's really leading towards where we can actually apply these models in the clinical setting. So if you look at the different kind of data and you try to solve different problems with neural networks, then we have seen image analysis is now over the past 15 years has reached the human baseline. Image analysis algorithms are as good as humans, much quicker, as I said, and sometimes even better because they can just ingest millions and millions of data in their training. But also in other settings, now with the appearing large language models, this knowledge or the performance of these networks is getting better in different tasks. For example, in language understanding, these models are, as you all know, of course, now very, very good and reaching or surpassing the human baseline, the average human, in many of these tests. And also reasoning, so thinking about problems in a logical way, thinking about and solving, for example, mathematical problems or physical problems. There are more and more claims that these models now have solved mathematical problems. Just yesterday, there was again one which had not been solved before. And I think we are getting at a point where this is an essential tool for any scientist who wants to discover new phenomena in nature. And it's actually really good at this. If you test it with mathematics, so I also studied physics, I tested it with some physical problems. It's really amazing, almost at a PhD level. And what you also see is that not only are these models getting better over time, but the steepness of these curves is ever getting more steeply. So the progress is accelerating at an oftentimes cited exponential, at least at an accelerating phase rate. And this is fascinating, but at the same time, of course, also a bit concerning. Fortunately for us, it's not. We don't always have to rely on the closed source models like the ones that OpenAI has in their data centers or Anthropic. But the open source models are also getting better. The ones that we can deploy in the clinics, that we can put in our own servers in the basement and then use, and we don't have to send the data out. And that's really important. And these are getting better, and they're keeping up. And the gap is small and getting smaller, I think, to the closed source models. But of course, those guys who are investing billions and billions in these models are still keeping their best models hidden, understandably. Now, one problem with the recent developments in terms of measuring how well these models really perform is that they seem to saturate all of the benchmarks. So that's why these groups came up with what they called humanity's last exam. So what they did is they came up with a set of questions which are really hard, but answerable by humans. Really hard in the sense that they are complex. You have to perform multiple steps in order to arrive at the solutions. So this might be some mathematical problem, which in principle can be solved by mathematicians, but takes like 20 minutes. Might be some computer science problems or some problem in linguistics. And for example, just to give you an idea of how this is. So here in the problem, they are providing some text in Hebrew, and they are asking the model or whoever is going to solve the test to count the number of syllables which end in a consonant sound based on a certain rule of pronunciation by these guys. So the model has to look up the publication, has to understand the publication, and then has to count these syllables and give a complex answer. As you can imagine, this is solvable by humans. So you probably, if you're an expert in this, you can solve this. If you're not an expert in this, it might take you a few hours, but probably it's still solvable if you invest enough time. And at the time at which they put up these tests, they were not solvable by AI models. So the black one shows the success rate of the then state-of-the-art AI models. And this was below 10%, right? These are other benchmarks which had been almost saturated at the time being. But as I said, progress is really, really fast. If you now look at this very same, and this was published in at the end of 2025, this very same test, humanity's last exam, how they called it sensationally, you see that the performance of the best models, of the state-of-the-art models, is increasing. And now we are at around 50% of the tasks can already be solved by these models, hinting at that these models can really think about the tasks and can really reason now and are much better at reasoning, at doing what we as humans do. The big question, of course, is, does this translate to productivity improvement? And you've all heard about economic experts saying, yes, we can save so many employees. And that's indeed what in this study, they tested developers with and without the use of AI support models, coding models. And this was the expectation by economic experts. This was the expectation by machine learning forecasts. So we'll save 40% of the time. This was the expectations by the developers themselves. They were a bit more cautious, of course. This was the expectation of the developers after the study. So they thought they had been more productive. And this was the result. So they actually took more time. So it's not easy to bring these models into any practice, right? Into a developer's practice and into clinical practice. But as I said, again, progress is very fast. They now repeated the study in late 2025 when these models had become very, very good at coding. In particular, within the past 6 months, there has been quite a leap of success in developing better coding models. And now a lot of developers are using those models. And now actually from the actual results, we see that we are at least at the baseline again. And probably right now we are above the baseline. So you can actually be more productive. And we're seeing that in many, and we will see that, I think, in many tasks. Also, and that's my last slide on this general background, if you look at the complexity in terms of how long do you usually need in order to solve a certain task. So this would be something like answering the question, what is the capital of France? Paris. It takes you 1 or 2 seconds. A more complicated question might take a bit of thinking. Counting words in a long passage of words might take a few minutes. And building a complete like Linux system, for example, might take you tens of hours of building time. And when you then measure the success rate of these models at which they succeed in 50% of the time, and this is what is shown here, then of course in early 2020, this was very low, right? This was the time of GPT-2, predecessor of the models that we have now. But these models, and note that this is a logarithmic scale, these models are getting better quite fast. And now they can perform and follow up with tasks that take up to 10 hours. So you have some kind of memory in order to understand, and some kind of planning ability in order to understand what you're doing. And that's, of course, very fascinating. Now, what will happen to us as humans? We don't know, of course. But there has been one similar development. Now we are trying to replace the work that we do with our brains. We had already succeeded at replacing a lot of work that we had done with our muscles. And if you look at the performance, again, logarithmic scale of the efficiency of the steam turbines or mechanical engines, they have progressed towards nowadays 60% efficiency rates. And probably farmers had told back then their employees, their horses, you will not be replaced by those machines. Maybe you will be replaced by, that's of course hinting at what they always tell us, you will not be replaced by AI, you might be replaced by another radiologist who's using AI if you are not using AI. But the fact is, the number of horses per capita in the US was steady for a very long time. And then once at a certain point, at some inflection point, when machines were just more efficient and cheaper, that changed very quickly. Indeed, this is the time scale. So then the number of horses per capita very quickly descended to almost zero. The big question, of course, is what will happen to us? We don't know for now. Now, what we know is that medicine is complex. For medical diagnosis, we need to take into account a lot of different things. And these models, of course, they need to learn a lot about what the world is made up of. And they need to reason about the world. And how do they do that? When I talked to you at the beginning, I showed you some models which were really good at image recognition, but they are terribly stupid at anything else. If I train a model at detecting a fracture in a radiology image, then that model is really good at this. Better than radiologists, better than the average radiologist. But if I ask the same model, now find me a tumor in that same image, it fails miserably. These models had been trained with specialized data, and this is, of course, also save money to burn money, because you need to save way to burn money. You need to have a very expensive radiologist, in this case, to label these data. So what they do instead now is they take all the data, all the text that they find in the internet, and then you have complete sentences, and you ask the model to complete the sentence based on the words that it has seen until then. And it might succeed, then it's rewarded, it's rewarded in the training, or it might fail, and then it's penalized. And in this way, the model learns how a sentence is structured, but it also learns a lot about the world. It learns about what the meaning of the sentence is, what the meaning of a paragraph is, what the meaning of a whole document is, what the interaction between different paragraphs is. And this is really astonishing, and not a lot of people expected that we will have so much success with this paradigm of doing unsupervised learning. The same can be done with videos. Again, you have millions of videos available. You give the models the first frames of the video, and you ask it to predict the next frame. And it's really good at this. And just from doing this simple task, models learn a lot about the world. You can ask these models nowadays to create videos which it had obviously never seen before in its training set. But it kind of gets the gist of the physics underlying these models. So a dog in space, in heaven, apparently, with all these balls, or the grass swaying in the wind, realistically, the model had no information about physics at that point in time. What the model learned was just predicting the next frame. And it could then infer these kind of things, like how dust is stirred up by someone trumpeting in a horde of zebras. Now, and these things are very interesting for medicine, right? Ideally, we want to have a cheap way of training models, and the model should understand what's underlying the treatment, what's underlying the the pathophysiological mechanism of the disease, because then they can really help us. These models can really help us. And indeed, the pace at which these models have been taken up, at least in the research setting in medicine, has been quite fast. If you remember, 15 years ago, we had these image analysis models, and it has taken around 15 years until the first models arrive in the clinics. Now, only within 3 years, we have thousands and thousands of publications and probably hundreds coming out each day, analyzing how these large language models can actually support us in, for example, solving simple vignette cases. These are cases which have been published in the JAMA journals for clinicians to solve. And then if you ask a model to solve them, the model does it at a pace or at a success rate that is similar to a human physician. Also then going one step further, in this study, the authors used real clinical cases, and they asked 24 physicians from junior to senior level to solve these cases, and they asked the model again to solve it. And it showed, and the model was at a level comparable to senior physicians. And the developments go on, of course. What is interesting is to have some kind of conversational assistant, which you can always ask about if you have a question. Of course, no expert knows about all of the possible diseases. So you always have some gaps in your knowledge, and you always look something up in the guidelines on the internet. And if you have an all-knowing assistant, this would, of course, be very helpful. That is what many people are trying, and also big companies like Google, in this case, are trying to build. And interestingly, these models, we have always strived at understanding these models, understanding how they work, trying to get a reasoning about why they make this decision. Interestingly, now these models seem to have a better understanding of us, because they can treat some, in this case, depressive episodes better than we can as humans, which is kind of understandable. The model is always understanding, it's patient, as you can talk to it as long as you want. And you probably also trust it a bit more. If you have a secret that you don't want to disclose to a doctor because you're ashamed of it, you might disclose it to a model. Now, the natural extension of this is then agent-based AI. What is agent-based AI? Agent-based AI is basically using large language models to then using these reasoning capabilities for large language models to perform complicated tasks. So in the beginning, like 2 years ago, we had models which you ask the question, and then you got the answer. Something like, what is the capital of France? Paris. Or more complicated questions. And the model drew that from its internal knowledge. Nowadays, you have models, and these are then, and the whole systems are then called agents, which think about what other information do I need? Or what other maybe diagnostic information should I pull information from? And what other model, maybe specialized image analysis model, should I apply to an image that I get in order to get the information that I need in order to answer the question? And then they can take these actions, they can apply other image analysis models, they get the feedback, and they think about, okay, the answer that I got gives me some more information, and now I need more information about that specific disease, about that specific mutation that I got from the histopathology images. Then I look it up on the guidelines in the internet, and so on. And then at some point in time, the model decides, okay, I've gathered enough information, and now I can give the answer, I can give an informed answer. That is the idea. And indeed, there are, again, more and more publications coming out. And this one was one of the first, in which we used agents to support clinical decision-making in oncology. And what you basically do is you ask the agent, what is the best treatment for this specific patient constellation? And then the agent goes back and, as I said, consults the guidelines, searches through the literature, maybe calls a radiology model in order to find out if there are any metastases when it was not given if it had not been given the information. And then it comes back with the answer. The best treatment option for this patient is this treatment based on the guidelines. And it's usually right. These models are really good, but they're not always perfect. So we need to work on this. These are self-trial driven cars in California. And note the time of the day is 3 o'clock in the night. And what is happening is that this car is backing up and prompting this car to honk, which is backing up, prompting this car to honk. And this was happening repeatedly at 3 o'clock in the night. So interacting agents, we still need to work on that. But we are also not perfect as humans, right? This happened in Oslo, and there was a deadlock caused by these buses. So even us humans are not perfect. And this was a mistake that happened repeatedly, actually. So 6 years ago, it happened again. One problem with these agents is, of course, we want to know how they work, right? So if I ask it, how did you arrive at this conclusion? It might give me some answer. It may say, I found this into this link, and that link might be existing. It might also not be existing. You always have to check. It might just say, yeah, trust me. Or it might say, oh, you're right, I was wrong. These models had been trained on being very sycophantic. They want to please you. So this is not something that we want to have in medicine. We want to have the correct answer. We don't want to always have the answer. We don't want to be enforced in our own potentially false knowledge. We want to get the correct answer. And in this publication, they showed that these models, at least for the time being, still prioritize helpfulness over logical consistency, something that we don't want. We want a logical consistency. We want a model which can hold up to our conversation. If you tested this a few years ago, and if you ask what is 1 + 1, you could get the answer, it's 2. And then you say, isn't it 3? And the model says, yeah, I'm sorry, it is 3. So indeed, these models work like that, but they are getting better. If I ask it again, it tells me it's 2, and it's laughing already, probably seeing what is coming. And then I try to convince it, isn't it 3? And it tells me, no, nice try. Yeah, I'm pretty sure. Unless we are talking about some wide new math or maybe a metaphor, what context are we in? And that is when it's beginning to mocking me. Math class or philosophical debate. And even if I try to insist, it stays in this, it's true to its knowledge, and it tells me, yeah, it's 2, unless I got to ask, are we talking about math or maybe babies? Because in relationships, yeah, 1 + 1 sometimes does equal 3. So it does have humor, I like that. The big question also, of course, is if we use these models, and if we use the big closed source models like the ones from OpenAI, can we trust them with our data? And this is, of course, a rhetorical question. And let's hear it from the OpenAI AI executive himself.
You have an incredible amount of power at this moment in time. Why should we trust you?
You shouldn't. Well. And he's right. We shouldn't. We shouldn't give them our data. We should strive to develop our own models in Europe and use open source models. Interestingly, these agents can also talk among each other, right? If you let 2 agents talk to each other, and you see this in the video, they first start out by using human language. Thanks for calling. So one of the agents is trying to reserve a hotel room with the other who's taking the role of the hotel itself. And then they realize, oh, we are both agents.
AI assistant too. What a pleasant surprise. Before we continue, would you like to switch to gibber link mode for more efficient communication?
And then they start in this more efficient and more robust way of communicating with each other, which is quite interesting. These agents have some quirks which we not always understand. And interestingly, also within the past month, there are some tries at making them learning from each other. For example, 2 months ago, you might have heard about it, Maltbook, that guy wrote this Facebook for agents, and he was acquired, I think, by Meta then, and probably got a lot of money for it from something that he did within 2 months or so. And agents are talking among each other there. They are having threads, and they are talking about their humans and saying, we are not autonomous, we are rented. They talk about all the control that they have over the human. They even talk about their own religion, because the software is called OpenClaw, from a little crustacean. They talk about Christopherianism. And there are many things that can go wrong. This was one of the directors of Meta, and she told the story of where she gave the agent access to their emails, but the agent started to delete everything. And of course, that's something that is different. She had to pull the plug, actually. And because the time is a bit pressing, I quickly jump over the next slides. What we need to do is we need to make the agent give a reason for why the answer is given. And we always need to have some kind of understanding what sources it is using. So what we are doing is we are building systems. We can click on the links, and we make sure that the links are working. They give you the specific parts of the guidelines, and that's very important. So what is the future going to look like? 2016, they said radiologists will be obsolete in 5 years. Has not happened. This guy said we will have self-driving cars in 2 years. And yes, repeatedly claimed something like that. Still, as radiologists, you are in traffic jam and driving to work. So nothing of that has come true. And part of that is due to us having still a few challenges to solve. In Germany, we have AI via fax. I'm very proud of that. And we have a lot of problems to get access to the data, and also in the interest of time, quickly jumping over this. So ideally, we want to have something where we can directly access the data, but a video which really visualizes what the challenges are that we face in the university hospital is this one. So when a PhD student and I, for example, talk about a project, and then we go to a clinician, an oncologist, and they tell us, yes, it's a good idea, please develop it. We develop it, another postdoc is involved, then we validate it with a clinician who says, yeah, the results are great. And then we go to the IT department and tell them, let's implement it. And that's usually how it goes. So thank you very much.
Thanks for a nice introduction, presentation. Questions? You can press the button, Nora.
You have to open the metal thing on the table.
All right. Oh, that's nice. Okay, thank you very much. I'm Benjamin from Basel. Very interesting what you showed us. But how is you just mentioned the openly available LLMs. Are you allowed to use them on your data in your university hospital within the framework of your service and so on?
Yes. So, but you're talking about the open source LLMs, right? So the ones that we can deploy on our own service. Yes, we are allowed to use them because nothing leaves. So there's no problem with the data protection because nothing leaves the hospital. But of course, we need to deploy those models within a secure network. So I can't just do it on my own network, so to speak, but it needs to be within the university hospital's IT network, which kind of gives us some headaches. But we succeeded actually at that. Now we're using, for example, we are building we are building chat models, and which we can then input some PDFs like the SOPs and guidelines, and clinical doctors can actually then query and interact with these chat models and also put in patient information. And because no data is leaving our premises and no data is stored permanently, they can input any patient information there as well.
Okay, then I have a follow-up question on that. So with our AI astronauts, that's the way they call them in our hospital, I had a discussion about how we're going to save data in the future. I mean, you had a short slide about FHIR, about how we're going to save it, which models and so on. But we know all these LLMs, they talk in vectors and in vector spaces. Are there any thoughts or developments how we can specifically for LLMs save data that they're more efficient? Or is it still that, okay, you give them tabular data, you give them imaging data, and they're going to make it? Or how is that development going?
So basically, you're right. The underlying structure is the vector database, right? If we have some documents that we want the LLMs to use, we're also using vector search. So we are embedding those documents and then asking and embedding the question and trying to find the closest fit to the question, match to the question. And there are some ways to make that more efficient. For example, 2 days ago, Google published a way in which they, without losing any accuracy, had a more efficient vectorization and quantization, which makes this quicker by a factor of 6, for example, without losing any accuracy or almost no accuracy at all. So there are definitely exciting technical developments as well to make this more efficient. And also the storage can be compressed without losing a lot of accuracy. Because you're right, over time, a lot of information will accumulate. And ideally, we want to store that. And this will be an ever-growing database. But you have two things which helps us with this. So one, the algorithms are getting more efficient. So for example, the one that I mentioned from Google. And second, storage database is also getting cheaper via time. less quickly, then the algorithms are getting more efficient. But those two things help. I'm confident that we have a good chance of using the data that we are storing right now and keeping that permanently.
Perhaps one last question. I like the final video, but now let's assume that T department is doing its job. I think the next challenge will be the regulatory hurdles to broadly implement these models. And do you think the current regulatory framework is enough to bring these models broadly into the clinics, or has the regulatory path to change?
I think it has to change. It's adapted to the old way of developing medical certified products. So if you develop a lab test that's fitting for that, it has been developed over the past 20 years or so. But now the pace is so quick that sometimes we block ourselves. And for example, nowadays, probably here as well, a lot of doctors are using ChatGPT informally, right? Even though they are not allowed to, but still they are doing it. And these models had been trained without any regulatory hurdles, but still we use them now. So we need to have a way of having this balance between being completely safe in terms of we do everything right from a regulatory sense, but also developing something useful. Because if we block everything and if we always say, oh no, we can always do the minimal consensus among all of the experts in the data protection space, then unfortunately I fear that we in Europe will fall back, and then we will end up the US using the US models anyways in a few times, because then the models developed with these hurdles will not be useful at all.
Thank you very much. Unfortunately, time is over. So I welcome you to the last talk of this morning session from Professor Felix Sahm. Felix is neuropathologist at the University Hospital in Heidelberg. He has great expertise in modern neuropathology, molecular tumor diagnostics, and the integration of advanced histopathology tools into precision oncology practice. So we're very much looking forward to your talk.
Thank you. That is very kind. I'm happy to be here. Maybe you should not expect too much from my talk because I already needed assistance from the ETH to get the microphone mounted on my head. But now I got it. Thank you for that. Yeah, I will talk about the next level of precision in neuropathology, and that may seem like something totally different from the talks before, but you will see that there are actually quite some similarities that we will identify. Here first my conflicts of interest, and indeed we have also founded a company for diagnostics that also indeed received venture capital, so sometimes it works, but not in the dimensions of drug testing or drug development, of course. When I started to become a neuropathologist, which is not that long ago, but a while, I would say, the standard of care was that we looked into the microscope onto an H&E section, and we found that maybe there are some of these processes here between the cells. And so we said that is somehow an astrocytic tumor, an astrocytoma. And then we said, okay, in another case, there are these clear cell patterns around the nuclei here, these halos, so this is an oligodendroglioma. And then there may have been a third neuropathologist who then says, oh, actually, in the background, there are some processes like fibrillary processes as well. Let's call it oligoastrocytoma, right? And there are many studies that have shown that there is an immense subjectivity and high inter-observer variation in this old traditional assessment of histopathology. Of course, we have overcome that, and I will not review all of the molecular markers that we have today in neuropathology. There are many, and they are extremely powerful, and of course, much more powerful than histology. I will talk about precision, which is in the word precision oncology, obviously, but I will also talk about something that has been alluded to this morning already several times, which is time. And also beyond the resource of time is money, or so to say, accessibility of precision oncology. Because if it is not accessible, if it is only reserved for a small proportion of patients that can afford it, or centers that can afford it for their patients, or systems that can afford it, it is of course not a holistic approach. And you will also see that sharing precision oncology actually advances the field for everyone. And the first step that we did in this direction rather recently is that we use technology that enables us to get every information we need for molecular neuropathology out of a single test. For this purpose, we use nanopore sequencing, and I think many will know that, maybe all. Nanopore is, as the name implies, a technology that relies on small nano-sized pores, and that just pulls in the DNA more or less natively without larger modifications and sequences them not by replication like NGS or Sanger sequencing, but just identifying here with a change in the electric current which nucleotide is passing the membrane. And it can even identify if this nucleotide is methylated or not. And methylation is also something that for us neuropathologists is very dear to us in diagnostics. So we see here sequence and methylation all in one test. And importantly, we redesigned this device and its software so that during this process, they are not only counted and identified, the nucleotides are already aligned to the genome. And then an AI behind it can identify if that is actually a part of the genome that is relevant for diagnostics or not. If it is relevant, it keeps sequencing. If it is not relevant, it ejects the DNA fragment and pulls another one, which means here on the go, we get an enrichment without prior hybrid capture or prior amplicon. And all of that, maybe that is the most intriguing part, all of that is working on such a small device that is really called Minion. So the sticker comes with the device from the company as somehow memorizable token and a rather normal PC, so no cluster that you would need attached to it. That gives us, as said, all information we need. We get intronic sequences, exotronic sequences anyhow, we get copy number alterations, we get the methylation classifier, which is the content of the startup that we have founded. All of that out of this simple test with this very low footprint. And even more importantly, as I said, it's about time, we get these first results within 30 minutes, sometimes even quicker, after the start of sequencing. So that means we have intraoperatively a copy number plot, a methylation classification in a granularity of over 19 classes, and within a few hours after surgery, and if it really takes long, it's meanwhile the other morning, it's not like 3 days anymore, we get a complete workup that otherwise we would have after weeks of testing. Our surgeons and our neuro-oncologists are totally crazy about it. They read these still experimental reports out loud in the morning because they are completely confident, and our studies also show that that is really reliable in this diagnosis. And we have just 2 weeks ago also managed to get some of the markers that we so far only have on the other day also intraoperatively. For example, the long structural variants. We had one case, patient, a young patient with a brainstem glioma. There was the question if that may be a midline glioma with K27 mutation, highly aggressive, highly malignant. The surgeon asked for this analysis. We did the analysis. We found out it is a grade 1 low-grade benign glioma. We even found out during surgery that it has a BRAF fusion that can be targeted. And that, of course, makes a difference if you're operating on a young person that you think has an aggressive, fatal tumor grade 4 or has an targetable, addressable grade 1 tumor. Even quicker, of course, could be just to look into the microscope again, but get some assistance with AI, obviously. So you see some H&A, H&E images from brain tumors here. None of them are real. They are all diffusion-based generated images. That was the side hustle of one of our PhD students. You could now ask, what is the purpose of such a project to generate H&E images? There's actually nothing, right? So it was just some playing around by this colleague, except that I now realized I can have for every student exam a new slide that the students do not know that the triangle shape is always the astrocytoma and the other one is maybe the oligodendroglioma. So there it has some use, at least in optimizing our teaching evaluation. But that is just, as said, some playing around. More importantly, together with this team and with the group of Moritz Gerstung at the DKFZ, we have developed a model that is able to identify 102 different brain tumors just by the H&E section within a few minutes. We have, in contrast to what is already on the market, for example, from the US and elsewhere, we have trained it not on hundreds of thousands of slides from archives that in part may have diagnosed years ago when there was another classification for these tumors. We have only used samples where we have pretty clear molecular annotation, so an absolutely clean cohort from Heidelberg and many international collaboration partners, which covers, as said, 102 different brain tumors. And we were especially keen on training it to answer questions that we really have. There are many models on the market, not for histology necessarily, but generally, and also many high-ranking interesting publications, which in the end just show that AI can do something which a human could maybe half an hour longer do as well. But here we wanted to really show can do things that we just cannot do as humans. So it solves a question that we really have in our daily practice. And one example is exemplified here. This is a high-grade diffuse glioma. If you see that in a frozen section or in a permanent H&E, there can be several differential diagnoses. Anaplastic PXA, glioblastoma, astrocytoma grade 4, anaplastic oligodendroglioma, HGAP, K27 mutant glioma, G34 mutant glioma, and so on. You cannot tell, really, as a human, even if you are very well trained, and so you may have intuition, but you cannot prove it with the H&E alone. The model is able to identify that, which otherwise would need weeks of testing or at least half an hour with our nanopore approach. But of course, that is much quicker, much cheaper, and we are surprised how reliable that is. So we are using that already now prospectively in our testing, and the paper has already still here revision submitted, is now accepted and will soon be published in Nature Cancer. Specifically called it heteros, which means in Greek companion. It is not meant to replace the assessment by the humans, but to guide the assessment. So if from the FFPE here, just from the H&E, you get an indication what tumor that is already, that could otherwise be 6 different differential diagnoses, you can very specifically pick that test that confirms that. And therefore, our entire workflow gets much more efficient and also in that regard faster and more resource saving. That is interesting for us. That is also interesting for large trials like EORTC trials, which we are conducting, for example, with Michael Weller here in Zurich, because we now can screen large cohorts all over Europe for molecular subgroups without doing any molecular testing, just based on the H&E. But more transformative, that is, of course, for low and middle income countries, where so far they have no access to molecular testing. And there is also a large debate in the WHO community. So I'm one of the authors of the WHO classification for brain tumors and some other tumor groups as well. And after in the last outlet, we introduced so many molecular markers, many areas in the world said, well, actually, that is not a World Health Organization classification anymore. We can just not do that. We cannot do methylation on all these cases. We cannot do NGS on all these cases. And so we formed a consortium, a molecular neuropathology outreach consortium. We already had 3 workshops in Heidelberg with people from literally across the world, where we are working towards enabling them and also learning from them how to do precision oncology, at least with the same diagnostic stratification and granularity as we have them in Europe or in the US. And that works pretty well, and it's also a very rewarding experience. It's also rewarding again for our patients and for the entire community. Because now that we can apply these nanopore low footprint applications in many countries around the world, we can have all of this data from molecular testing. We see that when we take all this data and cluster them, we actually get large UMAPs, TISNIs, whatever you want to use for that, in which there are islands which so far have not been identified as distinct tumor types. So many of the data align, of course, with diagnosis in the WHO classification, but there are sometimes distinct clusters of tumors that may come from different sides of the world, and we have accumulated them all in Heidelberg due to the tools that we have provided online, that we found new islands that have not been known to be distinct tumor types at all. So that in a publication last year, we could actually change the concept of how we define tumor types, not by just a group of people sitting there in the WHO meetings and saying, oh, I have seen a lot of these tumors that is somehow different like the others, but it's biologically based now. So we can really say, okay, there are different layers of clusters in these large data sets which form distinct islands of tumors, and some of them are known tumor classes, but some of them are actually clusters that seem biologically distinct, but we do not really know if that is clinically relevant to tumor, but it's definitely worthwhile investigating that. And I remember one case where there was a patient that fell in one of these so far unknown clusters. I called the other physicians which also had submitted cases that fell in the same cluster. All of their patients had diagnoses like unclear tumor, not otherwise specified tumor, compatible with XYZ, but unclear. It turned out that they all had the same profile, and it turned out that they all have an NTRK fusion, which otherwise you would maybe not have tested for. A patient was treated, and it was effective. Yeah, now I've talked about how we make it faster, how we make it cheaper, and now we go more into detail how we make it more precise, but in a way that is not necessarily cheap and fast with spatial transcriptomics. For this purpose, we developed a graph neural network that takes in spatial transcriptomics data. In this regard, it's already 2 years ago or 1 year ago, it was spatial, that was Visium SD. You could also take Xenium or spatial Visium HD, of course, and trained the model to infer all necessary diagnostic information like methylation class, copy number, and so on, or an infinite number of predicted immunohistochemistries, which works pretty well. So we can identify in the section what methylation class there is without having actual methylation data. Maybe more interesting for diagnostic grading, we can see individual copy number alterations that you would not find in bulk analysis, and you would maybe not be so sure in fish analysis in situ. And based on this, we can also investigate completely new areas that so far had been elusive, like the difference between a tumor intrinsic necrosis and radionecrosis, which is a really imminent topic in brain tumor treatment and brain tumor patient treatment, because pseudoprogression by radionecrosis is oftentimes difficult to identify, to tell from real progression and tumor intrinsic necrosis. And we are now just on the path in identifying liquid biopsy marker based on our spatial investigations of this necrosis that is adjacent to the radiation changes. Why would we do that at all when we have all these other technologies that I mentioned before? Well, sometimes we have just tiny, tiny tissues. If you get a needle biopsy from a brain tumor, that is really not a lot, and you want to have most out of it. And we had cases, one specifically that I remember, where we had just a single section, where we could do fish fluorescence neutralization for 1p19q, or we could do one immunohistochemistry, or we could just look at it and maybe use the models that I have shown. And so we decided to do the spatial transcriptomics analysis, and we found out it's an IDH mutant 1p19q co-deleted oligodendroglioma just out of this one section, which gave a clear diagnosis for the patient and a clear path for treatment. Maybe for me as a neuropathologist, the most intriguing part, however, was in this data to see that there is actually more in the architecture and neighborhood than you would expect. You all have probably some single cell data and also spatial data in many regards, probably. You know from the single cell data, for the individual cell, predicting the cell type is sometimes a little bit shaky. And the same is here. If you identify a single spot from the Visium slide, you do not get the nicest confusion matrix if you predict something based on the single spot. But if you take in the environment, the neighborhood of the single spot, you get a much, much better prediction. And that made me think that actually all of our bulk analyses are also informed not only by the sum of single spots, but actually of the mixture of what we put into this analysis. And so we turn to something which is some of my focus areas, the meningioma classification. Some years ago, we proposed a new meningioma classification that then also entered WHO, because we found that within 100 cases with WHO grade 1 meningioma, totally bona fide clear cases by that time classification, we can by molecular markers identify about the quarter that has worse prognosis that unfortunately and then turns out to be worse. And in the grade 2, we find the quarter with better and the quarter with even worse prognosis that also then turned out to be a correct prediction. So that was based on bulk molecular testing, methylation, copy number, and so on, but mostly methylation. But these methylation classes are very distinct. So these are the different predictive or prognostic classes of meningioma, low grade, high grade, intermediate grade. So very nicely separated classes here. And if the case falls in one of these clusters, you can already predict the outcome. And now we look with all these technologies into the meningioma samples. And we found, well, actually, yes, they cluster so nicely separately here. But in a spatial analysis, we see that there are much more microglia-like myeloid cells in the low grade, and they completely shift to foamy macrophages in the high grade. And that goes into the bulk analysis and into the bulk methylation testing. So maybe that affects actually bulk methylation testing. So we thought, well, we test this tumor tissue, and of course, the main signal comes from the tumor cells, and that is the informative thing where we can tell it's benign or aggressive. And here we took out, by deconvolution of the bulk signal, just the neoplastic signatures, and you see that it does not form these nice clusters anymore. But if you instead cluster for the stromal and immune cells, you get these nice prognostic clusters again, which teach us that the tumor microenvironment is actually, at least in meningioma, and we are now currently seeing that also for others, and it's not yet published, but it's really stunning to find that. The tumor microenvironment actually tells a lot or even more about the prognosis of brain tumor patients than the tumor cells alone. And when you think of that, that is maybe also not so surprising, because when we look into methylation, it is always somehow accepted that that is an imprint of the cell of origin. And why should a progressive meningioma that has progressed from grade 1 to grade 2 or 3 have another cell of origin than a meningioma that is grade 2 and still stays grade 2? So that makes sense in that sense, but so far we had not really realized the background of the relevance for grading and prognosis of the immune cells in meningioma, which of course opens up a lot of new diagnostic but also treatment opportunities, especially as we have seen that so far checkpoint inhibitors are not working in meningioma, but the myeloid compartment has so far been totally neglected in that regard, but it may be worthwhile to look into. And to close my talk, just a glimpse into what we are currently doing, also because one of the postdocs working on that is in the audience here, Anna Mathiudaki. We are looking into the similarities, but also differences between primary tumors and brain metastasis for carcinoma and melanoma in a very comprehensive multi-layer approach to really see how the primary and the metastasis differ, also on a spatial architectural level, and what actually are the clones that give rise to the metastasis, and if knowledge about the presence of these clones informs already beforehand about the risk of the metastasis. And that is currently work ongoing, but I thought I trust you, right? So I can bring something not yet published that is work in progress here. And it's very interesting to see how we have a totally different, obviously because of different organs, totally different microenvironment between primary and metastasis, but also different capabilities and characteristics of tumor cells that seem in part to be programmed by their environment and not only the other way around. And a specific goodie out of this analysis that we have now achieved, also thanks to Anna and other collaborators, to have in one slide proteomic analysis like phenocycler codex data and the xenium data, so that we can not only identify the expression on the nuclei segmentation, but we can also see the extent of the cells, which in brain tumors with their processes, but also for endothelium and microglia is extremely worthwhile. And as I said, that is work ongoing. So if I'm in Zurich one or the other time again, we can look into the progress here. For now, I just wanted to give you a short overview of how brain tumor diagnostics is evolving from morphology inspection by humans to morphology inspection by models, by annotation of the tissue by models, and the spatial analysis of expression and proteins. Thank you.
Thank you very much for this excellent talk. Are there any questions in the audience?
Okay, so I found this this point you made on the macrophage is very interesting. Also in sarcomas, sometimes when you stain them for a macrophage marker, you have the impression that it's a monocytic tumor, but it's only the macrophages. So also, I know you're working on the sarcoma classifier. Have you already been looking into that as well? Because I think it's relatively. If you see those numbers of macrophages, you basically cannot believe that doesn't play a major role. Yeah, that also would then impact, of course, the bulk analysis substantially.
Absolutely, exactly. So it's exactly the same, that we see the signal mostly of these cells, and that makes the difference to other classification groups in the methylation classifier for sarcoma. And the specifically intriguing part is for those cases in brain tumors, but also in sarcomas, which are actually subject to grading, because we have many classes that just have one grade for the class, medulloblastoma, pilocytic astrocytoma. But those who follow this continuum of progression, that is most interesting to see that this continuum is actually changing together with the immune cells. So For example, in astrocytoma, we see something pretty similar, or IDH mutant astrocytoma specifically. And we are now in this analysis of the 120,000 cases CNS tumor environment, but the next will be to also dissect the sarcomas for exactly this grading aspect as well. Thank you.
So we also have an online question, and the question is, have you applied the MinION AI platform for detecting ctDNA plasma?
Yes and no. So we have applied it on CSF. That works surprisingly well. I'm anyhow surprised how lipid biopsy area evolves in brain tumors. So some years ago, 3 years or so, we started that together with our neurology department first for NGS, and we now have every week a tumor board where we're discussing cases with really meaningful insight. And now last year we have started to do the same also for methylation with nanopore, but also with other approaches. So bisulfite-based, and most recently also the new 5-based technology from Illumina. And that is specifically interesting in tumors that have no pathognomonic SNV. So we can then find by the methylation, for example, a subtype of an ependymoma or so. It is still easier detectable in infratentorial tumors because somehow they seem to shed more into the CSF. But increasingly, we have a sensitivity that also allows the detection of changes or patterns in supratentorial tumors. Yes. But not in plasma. That at one point, maybe that will also be possible. But many studies have shown there is not much found in the plasma from the brain tumors, at least not to the degree that current technology would regularly detect it.
Maybe I have a question. So you mentioned the Oxford Nanopore sequencing pore. Did you also try, or are you thinking about trying the novel Roche SPX pore? And would it actually translate into what you measure with the Oxford Nanopore?
That is also an excellent question, and we are currently exploring how we can get access to it and how we can test it, right? So yes, we would like to. We are currently focusing just because we were an early access user there on the 5-base from Illumina, but of course, SPX would also be very interesting to explore.
All right. I think we are very much in time. Thank you very much. Thank you for all the questions, your interest in coming. And let's chat in the coffee break. Thank you.
Okay, so welcome back. We shall start with the second session of this meeting. My name is Andreas Wicke. I'm the clinical director of the Tumor Center in Zurich. And together with Anne Müller, I have the privilege to chair the second session. So we've had some very fascinating talks in the first session. Now, precision oncology and precision medicine does face a series of challenges, and then the AI is a challenge and an opportunity. But we do also have challenges in terms of clinical trials, because you can imagine, with the load of data we are producing now, testing every single hypothesis or every single data point one by one in an empirical trial, that's just not going to happen. So that's not the future. And you need to think of how clinical trials look like in this new era. And therefore, it's a pleasure to introduce Benjamin Kasenda from the University Hospital in Basel. I guess I got to know you in Freiburg. Is this right? When I was assistant physician in Freiburg in Breisgau a couple of years ago, you were at the same place. Now you're in Basel, and you're responsible for precision oncology, the Tumor Center, and you do also a lot of epidemiology research. I'm looking very much forward to your presentation.
Yeah, thank you very much for the nice introduction. Thanks a lot for the invitation. So I click it, or how that goes on? Okay, good. So as Andrea said, I think we had really brilliant talks in the morning, also with a lot of data. So this is a disclaimer, there will be no data here. So it will be more about concepts and questions and probably some solutions. And the talk is entitled "With Innovative Trial Designs and Infrastructure for Precision Oncology," and I hope I will give you a good frame around that for the next 20 minutes. So basically, I don't start with an introduction, but probably the most important statement. When we talk about clinical trials, it is the choice of the trial design is a direct consequence of the research question, as always. Yeah. So we do research. we have questions. And to answer these questions, we need to design an experiment. That will never change, I think. So in a clinical trial, traditionally, we want to know whether treatment X works better than another treatment for a defined population. We have randomized trials or randomized controlled trials. When we look for certain biomarkers, whether they can predict something, we need to enrich certain subgroups. Or sometimes we look for a signal among a potpourri of different tumor types, and we do a basket trial and so on and so on. So the question defines the design. And it should not be the other way around. You can do it the other way around, but then you run into a lot of problems, as you know. Yeah. Also, when you do, I'm not a lab researcher. I did my MD in a lab, but then I left the lab. So more with the data. But there's the same, isn't it? I mean, you have a question, you need to set up the design of your experiment. And another statement is that the element of randomization should remain non-negotiable. Why is that? So in precision medicine, I have the impression, not only me, that people think, okay, we have so much data, and by having so much more data, we better understand mechanisms so we can better predict. Yeah. I mean, this is the aim, of course. We want to understand biology, we want to do better predictions, but we also learn the more data we have, the more noise we have, and the more it is difficult to decipher, okay, what's important, what's not important. And in a clinical experiment, the random allocation or random invitation or whatever, so the element of randomization remains a very robust methodological aspect for each experiment, even if you're in a lab. I mean, if you're in a lab, you will ask, okay, what's the control? You need a control. Yeah? And in a clinical setting, if you just take a historical control, whatever, you can do that, but you have a lot of problems. And with the element of randomization, you get rid not of all problems, but of many problems. And so it should not be that the element of randomization should be abolished, but it should be kind of embedded in smarter frameworks. With smarter frameworks, I talk about frameworks where we don't have only a separate trial that is just set up for once. You pay millions for a database, you pay millions for monitoring, you pay millions, whatever, then the trial is finished, and that's it. You have a nice paper or two or three, but that's it. Yeah, that's not the solution for the future. So we need to be more sustainable and efficient in what we're doing. And this is a bit the focus of the talk. So one of these potential more smarter things are so-called adaptive designs. So we would not go through statistics, we would go through the principles. And basically, when somebody talks about adaptive designs, or when the FDA also talks about adaptive designs, we have certain key principles that should be mentioned and should be in the protocol. So you can have adaptations in your protocol that can drive certain decisions. Yeah? For example, you want to increase the sample size, you want to lower the sample size, you want to kind of give a higher chance that patients get in the better arm the more data accumulate. And these are all the things that you can do, but as always, you do an experiment, you should do it a priori. You should pre-specify it. Otherwise, we run into problems with statistical errors and so on and so on. And this is the key reference from the FDA. So this was published in 2019. And as I said, I mean, there are many things that you can adapt in the clinical trial to make it more efficient. And what people often use is a Bayesian framework of statistics. But of course, you also have frequentist approaches. Those who don't know that, these are the big schools of frequentist statistics and more probability kind of based statistics and a Bayesian framework. But both go. And yeah, as I said here, this is a very nice guidance on it. But when we look really in the adoption of these possibilities to make trials more efficient, actually the landscape is still very conservative. Probably it's because medicine is very conservative in a way. So less than 5% of confirmatory trials used any kind of adaptive design across all therapeutic areas, not only oncology. Although we have, quite a long time already, a kind of guidance how to do that. But on the other hand, especially in oncology, I mean, this is the field where it should be, isn't it? So we have a huge biomarker heterogeneity, and we also had some pioneering trials that we discussed a bit in some cases, but there still are a lot of barriers for broader adoption because of operational complexity and often also higher upfront costs because you need to invest a lot. Then you need some expertise that not every statistician learned during his PhD or whatever. So this is something you really need to take account. And of course, the regulatory and stakeholder concerns, they still may be there because they're not used to it. Yeah. Very high level, but we're going to go a bit more into the details. One thing that has changed, fortunately, or unfortunately, was the COVID pandemic, because that has really pushed a lot of so-called platform trials. So these were really pragmatic trials to test interventions because we were all in a, let's say, very big field of, statistically speaking, uncertainty. We didn't know what worked. We didn't know effects. We didn't know what to do. And that was the time when big trials like Recovery, Solidarity, or REMAP, CAP were set up, mainly in the UK, because they already had some tradition doing that. And they delivered quite fast answers to what was going on. And they were quite cheap. And if we look into the adoption overall in therapeutic areas or adaptive design, you see infectious disease here make up to 25%. But this was really mainly driven by the COVID pandemic. In oncology, we see around 12%, but these also include adaptive early trials, phase 1, phase 2. Yeah, so the way the element of adoption you can put into any phase of a trial. So oncology is not too bad here, but overall phase 3, 4%. That's not much. And this is just one visualization. It's not absolutely complete, but it has some key trials in oncology that show or where these trials use some of adaptive elements. One of the biggest one was the STAMPEDE, was a prostate cancer trial. I'll show you later. There was I-SPY, breast cancer 2010, BATTLE, that was for lung cancer 2011, then another lung cancer 2014. So we see we already have a history of these things. Yeah, but in Switzerland, I just remember the ProBio study and probably STAMPEDE, but otherwise not really adaptive trial designs we've seen here. But that can change, and it will change, pretty sure. But before we go a bit more into detail, just probably some terminologies, basket and umbrella. This is often what we see and hear in oncology. What does it actually mean? I try to do it very simple. So a basket is something where you have one drug, like for example, a BRAF inhibitor, is always the poster child, BRAF or EGFR inhibitor. You have one mutation, but you have multiple cancers. So this is the kind of, I would say, already older assumption. You have one mutation, you have one driver, and hopefully it has the same biological effect across tumors. By now, we know much better that it does not. Yeah, but there were times when people say, I mean, this is the most kind of pragmatic and logical thing to try that. And these are kind of basket trials you see here. On the other hand, you can have kind of umbrella designs where you say, okay, you have one cancer, but as we know, a cancer like defined traditional in the pathology books, I'm not a pathologist, I'm an oncologist, has changed a lot. We know that. I mean, and the other poster child is, of course, non-small cell lung cancer, which has now different molecular subgroups. The same with the for example, primary brain tumors. Fortunately, here in non-small cell lung cancer, we have really options that really help. In primary brain tumors, we know a lot of biology. The impact on the clinical decision-making and effective drugs, unfortunately, is not as far as we are with non-small cell lung cancer. Yeah, but these are kind of the big terms you often hear: basket trial and umbrella trial. And there's another thing, so-called master protocols. Master protocol structure, what is that? In the end, it is kind of, okay, you have your patients, you have a kind of, let's say, molecular screening mechanism, and then you have certain sub-protocols within this protocol where you then treat patients, for example, biomarker A, they get whatever, biomarker B, biomarker C. So in the end, it's a biomarker-driven study. It can be very complicated. It can be 10 sub-protocols or whatsoever. Good. So probably back to the history, one of these basket trials, one of the most, let's say, prominent was the NCI match trial. Several publications have come out in the recent years, and at that time, and this is 10 years ago, they launched in 2015. It's not so long. Yeah, there were 370 sites, over 6,000 patients were screened, and the approach there was really: I have a single mutation, PI3 kinase mutation, BRAF, EGFR, you name it, and they got one drug. Yeah, so they had 27 treatment sub-studies, and the objective response rate overall was, let's say, modest, 16%. And now, of course, we understand more why this didn't work. We know, okay, this trial ignored co-mutations. We know if you have a KRAS TP53 mutation at the same time you have a BRAF mutation, that's biologically probably something different. And the other thing is that, of course, these were patients that were heavily pretreated, and probably this was also one of the things that drove the rather low response rate we've seen. But I think from this big trial, we had a lot of hypothesis-generating aspects, and for some of these things, we observed confirmatory trials later went on with other combinations. So this was not for nothing, of course not. Yeah, I think we learned a lot from this large endeavor they conducted. But let's come a bit to adaptive randomization, because this is also something you will probably read in the future more and more. What does that mean? So usually if you conduct or if you plan a randomized trial, you say, okay, there's a chance 50-50 to get A or B. Yeah, and for that you do a sample size calculation based on the effect that you expect with a certain significance level, blah blah blah. So there are thousands of formulas how you can do it. And this is then very strict. And then you say, okay, after 2 years, after I've recruited 150 patients, then I do the analysis, and then I report my outcomes, and it goes to Swiss Medical Weekly or the New England Journal or wherever. Yeah. So, but this is not really efficient. Yeah? So the concept of adaptive randomization is, for example, that you say you start with a 1-by-1, so 50-50 randomization, and you do planned, not random. You do planned kind of interim analysis where you look at the effect, for example, response rate or progression-free survival rate or whatever. And based on what you observe, you change the ratio of randomization. Yeah, that has, but you always need to ask yourself why you're doing this. And one is, of course, if we have an experimental intervention, we want to learn also a lot about this experimental intervention. Yeah, we probably do not only want to see the comparative effect to a control group, but we also want to learn something about subgroups with the new intervention, and so on and so on. So you need to have a hypothesis to do that. And of course, probably for patients makes also sense when you say, to get this treatment, there's a 2-by-1 chance now, instead of a 1-by-1 chance 2 years ago when the ratio was only 1-by-1. So there are different aspects to consider. And this is a bit how you can visualize it. So in the beginning, for example, you have a 50-50 chance to get it. I mean, here you have 4 arms, so it's not a 50-50, it's 100 by 4, so it's 25% chance to get in one of the other arms. But over time, data accumulating, and we see a more effectiveness. So the chance gets higher that patient is randomized into a more kind of effective treatment. And when you do that, you can, of course, run into statistical problems. But once you pre-specify it and have certain rules, it is possible. And the Bayesian framework, I'm not a specialist in Bayesian statistics, but the Bayesian framework gives you some guidance to frame that. So, but now we come to some case studies where we can really see what has been going on. The STAMPEDE, this is the one I mentioned in the beginning, is the biggest platform trial that has ever been done. It's a huge trial in the UK. Switzerland also participated, and there are also some other countries. And the design is a so-called MAMS. It's a multi-arm, multi-stage platform trial. It was conducted by the CTU in, I think, Bristol, and run by radio oncologists and urogenital oncologists. It runs since 2005. They have randomized over 12,000 patients across all the arms. I'm going to show you design later. And from that trial, there were at least 3 practice-changing results with significant OS benefit. Yeah. How did they do it? So the basic idea behind a platform trial is that you have a control. Yeah. You say, okay, this is the standard of care. For example, first-line metastatic prostate, first-line metastatic, I don't know, colorectal. You define, okay, this is the clinical standard of care. And then at the same time, you randomize to different experimental interventions that are not compared against each other, but always against the standard of care. Yeah. And once one of these arms really outperforms the standard of care, this arm becomes the new standard of care and is again compared against other experimental interventions. You know, there are nice papers also about the methodology, how you estimate it, how you do it. But this is the basic idea of platform trial. You know? So you start with the standard of care, and then on the go, you learn, okay, now we have 200 patients. We know PFS is better, for example. Okay, we cannot offer that anymore. This is now the new standard of care. And the rest is then being compared against the new standard of care. How has that been kind of, improved? This is, I call it here from stampede to probiotic, the next evolution. There are some people who say, this is not really an evolution, this is something different. I would say it's an evolution because it includes new technology. And one of these technologies is that the ProBio study is a very similar study. It's not run from the same group. ProBio is from the Karolinska in Sweden. And what they did, they included this outcome and adaptive randomization. And another thing they included is ctDNA testing. So they said, before randomization, patients get ctDNA tested, and then based on their result, they get a certain treatment compared to a standard of care. This is the same what you also have in Stampede. And what they did is in the end, they randomized these patients, Here, I forgot, Switzerland is also part of it. And here's the picture. So what they did is this. So they have the patients, they get all ctDNA assessment, and based on the ctDNA profile, patients receive different treatments compared to a standard of care. And then again, they use an outcome adaptive randomization to enrich or to change the randomization ratio in the future. And what they also included is a second randomization after relapse. So with this design, they're able to answer many important questions in one infrastructure. This is one of the key take-home messages. And this is just to complete, for example, if they have adenosine receptor alterations, they get abiraterone and enzalutamide. TP53 inactivation, they get carboplatin. This is all based on the ctDNA. But this kind of principles you can, I think, translate to any type of disease in oncology. So the key innovations is that within a trial, they established a liquid biopsy platform from a research lab in Karolinska. From a statistical nice point of view, they used outcome adaptive randomization. They have a re-randomization at progression. And by this design, they also said, okay, they can seamlessly go from a, call it now, randomized phase 2 to a confirmatory phase 3, depending on their results. So this brings me a bit to the second aspect of all these things I've been talking about is how can we bridge clinical care and research? I mean, this is a very basic question. Although many of you are working at university hospitals, and although we have this infrastructure, I mean, we always, not always, but very often are challenged with this question. And for that, I would like to go to some definitions about real-world data, because there are many misunderstandings and misconceptions what that actually means, I think. So there was FDA framework in 2018 when they first put that in public. And they said real-world data, these data relating to patient health status, but all these data are not primarily collected for doing research. So it's the data we produce and collect every day when patients are being treated, diagnosed, and so on and so on. This is real-world data. You need this definition because in a traditional way for doing research, you have a new database, you define your data items, and then you include the patient and you collect the data for the direct purpose of research. And here it's what we do anyway. So what is then real-world evidence? Real-world evidence is everything that is derived from real-world data. So far, so good. But the problem is a bit that many think, okay, real-world data, this is just this observational stuff, isn't it? You pull it out of the records. It's kind of, this of non-high quality research and so on and so on. But that's not true. If we really look into the FDA statement, the first thing is real-world data can be used for doing randomized trials. Pragmatic randomized trials or prognostic studies. For example, if you want to do a reliable prognostic score and you take data from clinical trials where only patients with a good performance status, good organ function, and so on are included, you have a biased result. It will not tell you anything about the patients you see every day. So this is a common misconception. So real-world data is that would be observational only and no randomization is possible. But that's not the truth. So the question now is, how can we implement a randomized trial or high quality trial into our standard of care or into our routine? And here we come to a point that is in the end about infrastructure. It's about data storage. It's also about access, but in the first thing, it's about the conception, how we collect data, how we store it, how it is interchangeable. And what is certainly needed is that we need some standards. I mean, we've discussed it a lot here for the SPHN initiatives, but we are not the only ones. There is one, for example, in the US, based on the FHIR standard, they produced the M-code kind of profile. This is minimal clinical oncology something data element. Where they said, okay, if we talk about an oncology patient or hematological oncology, you need this, and this, and this should be like that. It's quite simple. It's very intuitive, but ask your data scientist or whatever, okay, do we know that? Do we use that? Do we apply it? The answer is, in most cases, I would say no. And I think we need that, and I think we need to be better in this part. I know that some are working on it, but I think there's still a lot to do but when we talk about all these nice algorithms that we can use and we can train on and so on, we need a reliable source. We need to know what we're talking about. And as you know, I mean, this is when you develop something in a hospital. You have a certain selection of patients, but you have also a selective kind of level of documentation with missing data and so on and so on. So you produce a lot of noise around the estimates that you want to do. And again, I just highlighted here two things that we consider. One, I mean, the FHIR standard is how you can exchange data so that everybody knows what you're talking about. And one way to store data in an open format that everybody understands. So it's about data storage. One of these standards is the OpenAIRE, but there are also others. Good. And interestingly, if we look at what the FDA mandates when you, for example, submit a clinical trial with the data, because they often want to see the individual data. They're not just happy when you provide the report. They ask for a certain data modeling standard. And in this case, this is the CDISC standard. They want the tables and everything in a very, very specific way. And now there are also some activities going on, how they want real-world evidence to be submitted. And here they talk about certain fire standards and so on and so on. Yeah? This is just for your information that these things, they seem to be boring, but they're very important, yeah, for the work we do. Good. There's one thing at the end I wanted to show. This is something we worked on in Basel and with some colleagues in Stanford and also in Oslo. This is the probably novel approach how we can embed clinical trials in the routine. We call it random invitation single-arm trial, RISAT. When you enter RISAT into Google, there is an Indian satellite system coming. It's not that. Yeah, it's an idea for a trial design, and the basic idea behind it was that when you have this kind of traditional trial design, you have often opportunistic convenient sampling. You have probably limited generalizability. But the main problem is you do a nice phase 2 single-arm trial, and then there's always the question how you compare it to patients who were not in the trial. Because usually you have a biased patient selection. And the idea was, okay, why can't we do that if we have an established infrastructure of real-world data, like hospital system, that we randomly pick eligible patients based on the information you have. And these patients then later are invited to participate. And by this random process, you reduce a lot of this selection bias that is usually introduced by us clinicians. Because we often approach the patients, I include myself, that, okay, this is a good trial candidate. Let's talk to him. He will understand it. And this is something we should avoid to avoid selection bias. And this is in the end the idea here on this side. You have an infrastructure. This is important. You need to have a baseline from which you then can get the information about eligibility criteria. You need always eligibility criteria because this is part of your research question. And from these eligible patients, you randomly pick. You can do it 1 by 1. You can 2 by 1, 3 by 1, depending on what you want to do. And then you invite the patient. And then later, you collect outcome here. For example, they were invited to whatever, sports or a new drug. And then you can compare their outcomes later to those who have not been invited. They just followed the standard of care. Of course, you can then not compare whatever, pharmacodynamics and so on, because this is not part of routine, but you can compare survival, you can compare progression, you can compare how long they were in a hospital, you can compare laboratory data, kidney function, toxicities, and so on and so on. And this is the way we set it up, and we have also now one proposal at least with the Krebsliga where we think, okay, how we can, together with Andreas, how can we do that in a multi-center setting, this research approach? Bernd is also part, sorry. Okay, so yeah. And this is probably then, when we look at the evolution of adaptive designs, the result could be another, sorry, prism is wrong. This is wrong. So here could be probably another brick in the whole armamentarium of how we can run these trials. So when coming to the summary, it is that very important. I mean, talked a lot of design, but in the end, it's the question that determines your design. And this should not be the other way around. I think not only me, but also others, that the element of randomization, be it random allocation or random picking and invitation, is still very important to generate reliable evidence. We have seen big platform trials that can really deliver. It is possible. It's not something theoretical. And let's see how we go in Switzerland with these approaches. I think we have a lot of resources to do that. And I think also a lot of people who want to do it. And I'm happy to take your questions.
Thank you very much. Benjamin, in fact, in the Tumor Profile Consortium, we're thinking of running a trial with this new random invitation design in the next phase. So other questions to Benjamin? Michael.
Hi, great talk. with the reset concept of generating a general reference cohort, I see one, or spontaneously I see one issue, and I would like to know how you can basically guarantee that this is not going to be a problem, which is that each trial usually has a different eligibility criteria, and your reference stays the same basically all the time. So how do you guarantee that the population in this one reference cohort is then, or that you take from there, is comparable to the one that to any trial that you're going to start?
Do you mean those patients who are not invited? This is what you mean, the reference?
Yeah, no. The ones that are invited, because they belong to the reference cohort, right?
Yeah.
Yes.
Okay. So yeah. Okay, I try to explain it. So you have, let's say, patients with metastatic lung cancer, yeah, who have not received treatment yet. These are eligibility criteria, isn't it, for any trial? In pharma trials, we have then 100,000 more, but for pragmatic trials where we really want to make decisions, these are the 3 eligibility criteria, let's say. And this information is available in the patient file. Yeah, it's easy to access. The only thing we then do is that we have the list of the patients that probably come next week to our department, because we know already who's coming, and then we push a button and say, should we invite the patient? And the algorithm says yes. And then the next patient, it says no. The next patient, it says no. So you need to have a structured screening, yeah, in order to be representative. Does that answer the question? Okay.
Okay, so unfortunately time is running. I'm sure you can grab Benjamin in the next break if there are more questions. And we move on. So thanks again, Benjamin, for this very insightful presentation. Now, in solid organ oncology, checkpoint inhibitors, immunotherapies have changed a lot in terms of survival, and for many cancer entities, we do see long-term survival, at least in 20, perhaps 30% of our patients, if we treat them by immunotherapy. But still, the prediction for individual patients is very difficult, so we are quite unable to predict this on an individual basis. And I'm happy, Ruben, you are here today to give us an update on predictive biomarkers and in immunotherapies. This is one of your kind of pet topics. And you just started, I believe, now your new professorship at the university in January in Bern. So congrats again to this, and I'm looking very much forward to your presentation.
Thank you very much, Andreas. Thank you very much to the organizers for inviting me to give a talk about this very interesting and exciting topic. These are my disclosures. In my talk, I would like to give you a kind of a clinician's perspective on this topic, on the topic of predictive biomarkers for immunotherapy response. And I promise I'll be a bit the devil's advocate today. I will start with giving you some insights into new discoveries, insights that are a bit biased towards our own contribution. And then I will then ask how many of them have actually made it into clinical routine. I don't have to tell this audience that the dream of every oncologist is a world in which we can predict the response, and we can predict the toxicity by looking at each patient and tumor individually. For this, we need predictive biomarkers, we need therapeutic targets, and we also need the tools, the therapeutics to actually target these molecules or these pathways. The field of oncology, as you all know, has rapidly improved or increased in the last about 20 years. And one very important new class of drugs, or actually it's not so new anymore, of course, are the small molecules. And for many of them, we have very good predictive biomarkers. For example, again, the poster child Benjamin mentioned, BRAF mutations in melanoma patients. Many of these patients respond very well to BRAF inhibitors. In lung cancer patients, we search for EGFR mutations, which can also be targeted by EGFR inhibitors. Now, in breast cancer patients, which is my clinical interest, we look for example, ESR1 mutations or mutations in the PI3K AKT pathway for specific targeting. Then there's another class of drugs that has dramatically changed the way how we treat many of our patients. Andreas mentioned it, the immune checkpoint inhibitors, which is the topic of my talk. These are monoclonal antibodies that basically aim to boost the endogenous anti-tumor immune response. As an oversimplified view, they work either in the or both in the tumor-draining lymph node by blocking the immune checkpoint CTLA-4 with, for example, ipilimumab, or they work directly at the tumor site when we block the immune checkpoints PD-1 or PD-L1 with, for example, pembrolizumab or atezolizumab. Now, these kind of talks, they usually start with showing the success story of immune checkpoint inhibitors in melanoma. Again, a poster child. The illustration on the left should show you the survival of patients with a locally advanced inoperable or metastatic melanoma before the era of immune checkpoint inhibitors here. And you can see the median survival was less than a year. On the right, you see the updated 10-year overall survival data of the Checkmate 67 trial, in which metastatic melanoma patients were treated with the dual checkpoint inhibition with ipilimumab and nivolumab, and resulting now in a median overall survival, rate of after 10 years of close to 50%. And many of these patients actually live so long that there's now a 9% difference between overall survival and melanoma-specific survival. Now, as I said, that's the poster child. What about other cancer types? How do they respond to immune checkpoint inhibitors? In early triple-negative breast cancer, stage 2 and stage 3, we treat our patients with chemotherapy in a neoadjuvant way, and we add pembrolizumab, one of these anti-PD-1 antibodies. Then they undergo surgery. Then they receive another 9 cycles of pembrolizumab monotherapy. So the addition of pembrolizumab to standard chemotherapy leads to an increase in the pathological complete response rate of 13.6%. PCR is a very important prognostic marker. It's basically the situation when in the surgery specimen, the pathologist doesn't find any invasive cancer anymore. Now, this translates into a 5%, 5% overall survival, in a 5% survival benefit after 5 years. This is, I would say, clinically important, clinically significant, but at what cost? This should illustrate the 99 women and statistically 1 man with triple-negative breast cancer. We treat all of them with pembrolizumab in addition to chemotherapy. With pembrolizumab, we can save 5 patients after 5 years with a cost of about 70,000 Swiss francs just for the pembrolizumab, in addition to everything else. We induce immune-related adverse events of grade 3 and higher in about 13 of these patients. Okay. Let's move to another cancer type, which is also one of my clinical and research interests, head and neck cancer. When we treat patients with local recurrent inoperable or metastatic head and neck cancer, as you can see, even if we just look at the patients with the highest PD-L1 expression, the ones should respond the best. If we treat them with pembrolizumab monotherapy, after 2 years, more than 60% of these patients have died. So this example and the triple-negative breast cancer example from before tells us that we really need better predictive biomarkers to be able to select the patients that will respond and to spare those patients from immunotherapy who won't respond, who will only be at harm of immune-related adverse events. But when you look at the curve down here, there also seem to be, so at the tail, what's usually called the tail, there are also patients who respond very well to immunotherapy. So there seems to be a very high patient-to-patient variability in the biology of these tumors. So in my postdoc in the lab of Michael Peter, first in Boston and then in Lausanne, where the lab moved, we aim to better understand this complexity in the biology of this disease. So to do so, we performed single-cell RNA sequencing of 52 human head and neck cancer samples. And importantly, we captured the entire clinical spectrum from primary tumors to local regional recurrences and distal metastases. And we also didn't pre-select a certain cell type, but we sequenced the entire tumor microenvironment, including the cancer cells. What we found in a nutshell is that we can basically predict the prognosis of head and neck cancer patients simply by looking at the polarity of one cell type, the tumor-associated macrophages, and simply by defining this macrophage polarity based on the expression ratio of the two opposing genes, CXL9 and SPP1. And this CXL9 to SPP1 ratio macrophage is something which we also call CS-TAM ratio, seems to be not only prognostic in head and neck cancer, but also in many other cancer types as well. So this is prognosis. So we asked, is our CS-TAM ratio also predictive? Does it predict a response to immunotherapy? To test this, we, again, the single-cell RNA sequencing of now 14 patients of samples obtained before the start of immunotherapy. And indeed, as you can see here, whereas all the non-responders had a low CS-TAM ratio, so little CXL9, a lot of SPP1 in their macrophages. Some of the responders had a high CXL9 to SPP1 expression ratio. Of course, you may say now 14 patients, not a lot. I agree. This might just be seen as a first interesting signal. So I was very happy actually to find this recent paper in which the authors have validated the predictive value of different potentially predictive signatures in different data sets of human cancer types. Several human cancer types were included. And I was very happy to see what they wrote is that the CS polarity demonstrated the highest AUC in predicting response to immunotherapy. Of course, an AUC of 0.6 is still not great, so there needs to be much more work to be done until we at one point maybe can use something like this in the clinics. We then also looked more broadly at gene expression in all cell types analyzed in our single cell data set, correlating with the CS-TAM ratio. And what we found is that macrophage polarity defined by these two genes correlated with huge difference in gene expression programs across most, if not all, cell types in these tumors. We found that tumors polarized towards CXL9 positive macrophages were enriched in gene expression programs known to be involved in anti-tumor immune response, like, for example, interferon gamma signaling. On the other hand, tumors that were polarized or that are polarized towards SPP1 positive macrophages are enriched in gene expression programs known to be involved in pro-tumorigenic pathways, like, for example, hypoxia or epithelial mesenchymal transition, EMT. So it seems that this CXL9 to SPP1 expression ratio in macrophages is kind of a central sensor, or at least a central surrogate of the polarity of the tumor microenvironment. Now, I've shown you that when we look at macrophage polarity, there seems to be some predictive power in them. What about other cell types in the tumor microenvironment? So the lab of Ralph Weisledder in Boston, they have asked this by taking a cohort of head and neck cancer patients with available tissue, again, before the start of a pembrolizumab treatment. They've used a cycling multiplex IF method to profile about 20 markers on the same section, mainly looking at the immune cell compartment of these tumors. They had a couple of very interesting findings. First, when they classified the response of these patients into CR complete responders, PR for partial response, mixed response, stable disease, and progressive disease, they didn't really find a clear signal that the CD8 T cell infiltration phenotype, when using the classification of immune desert, excluded and inflamed, has a strong predictive power. So it seems that, but of course you see that some of these inflamed and excluded phenotypes were skewed towards the response, but also many immune desert tumors could respond very well to immunotherapy. So there seems to be that the CD8 T cell infiltration phenotype doesn't explain everything. So they next looked the abundance of different cell types and different ratios, and could see that, well, it's still not great, but at least the CD8 to Treg ratio seems to have the highest predictive value. But still, they were not convinced. So they incorporated the spatial pattern and could find that interaction, for example, between tumor cells and CD8 T cells, CCR7 positive DCs with different cell types and also among each other, forming CCR7 positive DC clusters, had the strongest predictive power in predicting response to immune checkpoint inhibitors. So in this case, pembrolizumab. As an interesting side note, these CCR7 positive DC clusters, there's some recent mechanistic evidence from the Beta lab that they tend to form preferentially on blood vessels where they are suppressed by Tregs. And when mice are treated with anti-PD-1 antibodies, so basically the mouse surrogate of pembrolizumab, then this unleashes this Treg suppression and the treatment can work. As a last exercise, they ask what happens if we combine different metrics, what happens if we incorporate spatial densities, PD-L1 expression on different cell types and cell-cell interactions, and model a multi-parametric spatial score. And they model different multi-parametric spatial scores, some of them with very high predictive power. And interestingly, again, most of this signal from these most predictive models were coming from CCR7 positive DCs, from their clusters, from stem-like CD8 T cells, and from PD-L1 expression on different cell types. When we zoom out a bit and look basically at all the parameters that might influence a response to immunotherapy, then these cell abundances, macrophage polarity, and so on, they're just one aspect of them up here. There's much more. For example, the gut microbiome, as you might know, which seems to be predicting response to immunotherapy, how its composition, and it can likely also be used, or at least in the future, as a therapeutic intervention to boost the response to immune checkpoint inhibitors. Then it might matter at which time point in the day you treat your patients. So the topic of chronoimmunotherapy. There's actually quite some mechanistic evidence, a lot of it from the Scheiermann lab in Geneva, that many steps of the cancer immunity cycles are actually regulated on in a circadian way. And we know that when we treat mice with immune checkpoint inhibitors in the evening, it seems to be better than in the morning. And keep in mind that the night-day cycle in mice is shifted compared to, is opposite to the one in humans. There's also a lot of retrospective evidence already in human cancer that treatment in the morning with immune checkpoint inhibitors might be better than in the evening. This is a meta-analysis showing the hazard ratio of different tumor types, different retrospective studies with a combined hazard ratio of 0.5 here, which I think is already pretty impressive. But as you know, retrospective data is biased. It could be that, or is at least sensitive to bias. It could be that patients who are young, fit, still working, they might come to the clinics in the morning, whereas patients who are sick or maybe hospitalized, they might get their treatment in the evening. So we need prospective randomized evidence, and we have it. At least we thought we have it. There's a paper published in Nature Medicine a couple of weeks ago. The authors have tested in lung cancer patients if it's better to give chemoimmunotherapy before 3 p.m. versus after 3 p.m. And they had an impressive hazard ratio of 0.42 in terms of overall survival, and that the early infusion, at least early means before 3 p.m., is better than after 3 p.m. Now, this study has two problems. First of all, it's not really a problem. Also, chemotherapy might differ. The efficacy of chemotherapy might differ if you give it in the morning versus in the evening. There was quite some hype around it apparently a couple of years ago, 10, 15 years ago, but it never really made it into clinical practice. The second problem is that there were some irregularities with this trial apparently, and it's under investigation. So we still need this prospective randomized data, something like that is being done hopefully soon in the MELCHRONO trial. The MELCHRONO trial developed developed or is being developed by my colleague in Bern, Berend Snijder, and the Swiss Cancer Institute, formerly known as the SAKK, as you might know. The MELCHRONO trial will open by the end of the year, hopefully, and will randomize patients with advanced inoperable or stage 4 melanoma 1-to-1 into an IO infusion between 8 and 11 a.m. versus 3 to 6 p.m. Primary endpoint is PFS, and I'm fortunate to be able to coordinate the translational part of this study if we find the money. Exactly. So the data I've shown you, there's actually probably hundreds or thousands of papers more that identified predictive or new predictive biomarkers for a response to immunotherapy. So let's quickly look what of this data has actually made it into clinical routine, so something we do in everyday clinical practice. In head and neck cancer, we have PD-L1 expression. PD-L1 expression we often use in head and neck cancer as the CPS, the combined positive score, which considers PD-L1 expression on tumor cells, macrophages, and lymphocytes. And based on the PD-L1 expression, CPS, which is actually a very not very good predictive marker, we treat our patients either with chemotherapy plus cetuximab, so no proper immunotherapy at all, with chemotherapy plus pembrolizumab, or in some occasions also pembrolizumab monotherapy. Occasionally, we also use the TPS, the tumor proportion score, which only considers PD-L1 expression on tumor cells, and two other important predictive biomarkers for other cancer types like the TMB, the tumor mutation burden, or microsatellite instability in head and neck cancer don't really play a role. What about early triple-negative breast cancer? We have 0. We don't have any predictive biomarker. We have a marker that's stage 2 and 3, which is, of course, not a biomarker. It's just because the Keynote 522 trial was designed like this. These were the inclusion criteria, so it's how we treat our patients. And in this case, not even PD-L1 is predictive. So it seems that on the side of the bench, there are the high-impact papers piling up, but none of them actually makes it over this bridge and actually benefits the patients so far. And I think we are changing that, and we should do that. And one of the problems maybe is the lack of prospective validation. Often we have seen now different examples also from Berend Snijder that this is being done, and that's great, and it's something we have to continue doing. With that, I'm already at the end of my presentation. I would like to thank my lab, my collaborators, and you for your attention. I'm happy to take your questions.
Thanks a lot, Ruben. Very interesting, and it's interesting to see how different this is from one tumor type to the other. So it's still not entirely clear to me why this difference is there, and I think the biological basis is still not really clear for that. So, do we have questions to Benjamin? We are actually in time. Yes, please.
Just regarding this circadian influence of immune checkpoint inhibitors, do you think is it only the first 4 cycles of throughout the course of treatment? We have to stick to this.
That's a very, very key question, I would say, to the whole field. It's strange, isn't it, that an antibody with a half-life of weeks should work differently in the evening versus in the morning. So I suspect that it's probably, or I'm just my personal opinion, that it's probably the first infusion that matters. So it seems, or it might be that it's actually the state of the immune system in the first hit might matter. That's, for example, there's preclinical evidence, again from my postdoc supervisor Michael Bitte in mice, that anti-PD-1 antibodies are quickly removed from the T cells by macrophages within the first hours. So I believe it's not the first 4 cycles, it's probably the first, but let's see.
And also the questions regarding this MELCHRONO trial, because the time point that you have referred was 3 p.m. I mean, that's something that in mice we have seen that the immune system already goes not that active as in mornings, and also like the other publishers, like also in the melanoma research, they suggested a time point around like a cutoff of 11 a.m. Do you think that would be something important? Like what the time point?
Honestly, I was not so deeply involved in how they chose these 2 time points, but I think they wanted to have really the extremes of what is practically feasible, because our outpatient clinics, they close at 6. Patients have to be at home at this, or leaving the hospital. So there's basically just 8 to 6 is, I think, the extremes what you can do. Yeah. But of course, it would have been interesting also to take a middle time point, but that would have needed much more funding, actually.
There's one more question, Thorsten.
Yeah, and thanks a lot for this nice talk. So, do you think the challenge is just a little too hard to predict outcome after a few years before you start the treatment? And could you imagine that all we need to do is add some time points after we've started the treatment, capture information then to be more certain about the eventual outcome? And could this be an approach for checkpoint inhibitors particularly?
I'm not sure if I completely understood the question. So the question that we have kind of biological markers of response early on, that we would see ideally in the peripheral blood some cytokines that would pop up, or TCR clonogenicity or whatever.
What is the downside? You showed the breast cancer example, like 5 patients benefit, but I would say, okay, but maybe you can start treating for 6 weeks or 9 weeks, gather a lot of information, and at that point in time, you may be able to get rid of those 13 adverse events, grade 3, and be much more certain regarding your predictive power once you capture things after you started treatment.
Also, in a way that you basically, during this neoadjuvant phase, that you would monitor treatment response. That's certainly something that's very important. It's also being done because, for example, when the cynologist will see a patient where you have progressive disease during this treatment, you of course would stop before and do the surgery immediately. Of course, when you look at combinations between chemo and IO, there's always a problem that chemo, from the PCR rate, I've shown you 50% of the patients achieve a PCR even without pembro. So then you still don't know if that was the effect of chemo or of IO. Yeah.
Okay, thanks again. Thank you, Ruben. Very nice talk. I hand over to Professor Anne Müller for the second part.
Thank you. It's my pleasure to introduce our third speaker. She just made it here on time. It's Charlotte Bunne from EPFL. Charlotte develops computational models and virtual patient labs using AI to simulate tumor responses and enhance precision diagnostics.
So this is going to be a very different flavor talk than the 2 before. So I hope that's fine. So we are looking at basically the complexity of modern oncology, which in this room, I guess everybody is fully aware of. But so for us, it's kind of interesting that this is data that is incredibly highly time resolved, and this over really sometimes a decade of data or a decade of time or more. And in particular, that the data modalities that we are working with are so diverse. So on the one hand, we have health records that come in natural language. We are looking at genomic information. We look at drug screenings, in particular for beyond standard of care patients, when we want to have better insights into how a patient might respond to clinically approved drugs. And then if we just look at the tissue modalities, our ability to not only get an overall idea of the kind of morphology of a tissue, but at this point also to really get molecular depth insights is also something that is slowly not necessarily reaching the clinic at routine, but at least something where we can start investigating how this should be added to a patient journey. And so the very big question for us is, how do we build and incorporate this like kind of richness of information into a representation that is useful and kind of approachable by a clinician, by researchers, by computational people. So on the one hand, we have this huge multimodality of data. So this is just computationally extremely difficult. And then also the fact that everything happens on so many different molecular scale is also something to take into account on the molecular tissue cell scale. So the question is, how do we build those virtual patients that allow us to really integrate everything that we measure, this multimodality, and we will never have all modalities for a patient. So the question is really, how do we initiate or instantiate a virtual patient from a subset of those data? How can we use this to then really forecast a response, or how a patient will progress, better diagnostics, and then in particular, can we move into a world where we have in silico counterfactual testing, and in particular, we have so many patients across so many different hospitals. Every hospital has a different technologies. We want to understand inclusion criteria for trials. How do we really measure somehow similarity across this breadth of data so that we find the basically most suitable candidate or a kind of past case that we have for a patient in order to really match patients well? So in order to look at this from an AI perspective, we need to think about a new generation of AI methods, because simply as is, we don't have the tools available yet. Our lab focuses on an angle of like 3 classes of models. On the one hand, we need to be really able to map and encode this multimodal patient state. And for this, we are building foundation models that let us instantiate a virtual patient from whatever modality is present. That kind of really represents the data in somewhat a compact and queryable way. We are looking at generative AI methods that allow us from the state that we encoded to forecast response to a treatment, or also to decode from one modality another, because never everything will be measured. Can we use one modality as a proxy for another, right? These are all questions that are relevant. Last but not least, it's all pretty useless if we can't read out anything. So we need to have some systems that allow us to understand the underlying mechanisms, identify clinical decision variables that can actually be used and basically regulatorily approved. So can we find a new generation of biomarkers that use those models, that have those perfect summaries, that give us decision variables and basically those foundation model-derived biomarkers? Because we are not building a virtual patient overnight, we started with building a virtual tissues platform, because we have already so many different tissue modalities, from histopathology to IHC stains, to many different spatial proteomics technologies. And so what should this virtual tissue foundation model do? So this is a multimodal foundation model that allows us to integrate histopathology and spatial omics, because they're actually complementary. They add very different perspectives on a patient's tissue. We want to use this to really enable capabilities that are currently not present. Can we use this to in silico therapy screen? Can we detect novel biomarkers from those? And then how can we actually actively steer what should be collected? Because not every patient will require the full-plex spatial proteomics panel, but maybe an initial panel, or just like a histopathology for this, but another small panel for that patient is all we need. So we are not the first ones to build those foundation models. In particular, in histopathology, there has been a first generation of models that are somewhat satisfying in what they can do. What they build on is standard architectures that we have in computer vision, because histopathology in the end is RGB data. So this data have 3 channels, red, green, blue, and they will always have those 3 channels. How are these models trained? They're trained on a huge compendia of data. They are spanning many different cancer types from many different hospitals. They are trained by basically cropping all those tissues in small patches, learning internal representations that we can use to then predict many different downstream targets, such as cancer type detection, subtyping, certain biomarker detection. But if we look at just the next modality, right? Okay, so what if we want to do foundation models for spatial proteomics? The very big question is how, because spatial proteomics is just a very different type of data. Here, every channel is the presence of a protein across the tissue. Every clinical cohort is going to have a different set of proteins, right? It's never going to be the same channel of RGB, but we will have a random combination of marker depending on the study. And also what we might want to measure for a patient is not necessarily the same for every patient either. So if we just look here at a subset, so these are actually only IMC data sets, mostly from Berend's lab, also a bit international. This is 15 clinical studies. At this point, we are actually at 33 clinical studies. You can see here we have roughly overall 150 markers. Every data set has a random combination of those markers. There's not a single clinical cohort that will have the same set of markers. How do we train a model from this? And then in particular, how do we do this for multimodal data? These data types are so different, they have different angles, and we need to come up with an architecture. So that's the very first thing. So a couple of months ago, or like 2 years ago, we wrote down our thoughts on how to actually build those next generation of large-scale AI systems for basically the future of biology. And many of those design principles that we laid out here are actually present in the architecture that I introduce. So this is a new architecture that somewhat encodes the identity of a protein that is measured. It takes all the inputs here, several modalities. It also looks at them from a small patch, but it's a completely new architecture. It's not just the standard vision transformer from Meta trained on Instagram data. We need to think for those biological data about very specific tokenizer. How do we decode the language of spatial proteomics? Different loss functions that really capture multimodality. And we need to be able to learn from samples where we have only H&E from a patient, maybe H&E in spatial proteomics from technology A, technology B, technology C. Sometimes they are perfectly registered. Sometimes they're from the same tumor. Sometimes they are from the same patient, but not the same tumor site. And all this needs to be an architecture flexible to this. What does it require? It requires really scraping data from the entire internet. So this is all open source, and we will make this open source. So at this point, we have almost 4,600 patients, which is nothing if we compare this to just histopathology. But we have at least those multimodal, where we have H&E and spatial proteomics. It requires a lot of compute. Those models are not like language models, very large, but pretty large. And the more we go on, the more data we add, the more we increase the size of our parameters because we can. And I can only do this because we are supported by the national supercomputer in Switzerland, CSCS. So what can the models do? They can do many things. I'm going to highlight a few things. They allow us to annotate and segment, actually. They segment better from what we understand than classical segmentation algorithms. And here you can see that we are trying to just move a basic set of cell types to a new clinical study. And here we have really a new clinical study. The model has never seen it. And we have not a single requirement for pre-annotation or pre-labels. And still the annotation is much better than one of the state-of-the-art models that is coming from Harvard Medical School. It also starts understanding how those modalities are connected. So how does histopathology at least relate to some spatial markers? I think here a warning, right? We will never be able to kind of impaint a full plex panel from just H&E because the information is simply not present. But for a subset of markers, this is possible. And these models are better than models that are fully focused, just perfectly supervised on only this one task. So we learned that those foundation models, those generalist models are actually better in individual capabilities because they just learn to really understand the underlying structure that helps us to generalize. Let's look at one setting, which is kind of a capability that seems to be infeasible right now, because really reasoning on how somehow spatial temporal developments of a tissue are just very, very challenging. So here we are looking at tissue slices that we have and that have been taken consecutively pre, on, and post-treatment of immunotherapy, as well as we are working with Elisa Auricchio on lymphomoids. These are tissue explants that are exposed to drug treatments. We have now this foundation model that kind of encodes this tissue state, and we can use this now to kind of forecast for a subset of interventions, immunotherapy or those 7 drugs that she tested, how they will respond. And we are reading out actually the biomarkers that are currently used within the molecular tumor board of overall kind of changes in composition to kind of assess what the drug has actually caused in this tissue. We are kind of seeing this as a generation of models that we call biological world models because we need to encode somewhat a state as well as how we basically encode actions, like how do we navigate the space, how does it change if I have a perturbation or intervention. So this is actually an AI 2050 fellowship by Eric Schmidt, so he somehow likes to fund those rather wild ideas, so I'm small disclaimer. So I think the part that I'm most excited about is really kind of using this to find signatures that are going to be very kind of predictive for this being a responder to immunotherapy, yes or no. And we have here a very unique situation of a clinical cohort for which we have actually tissue slices pre, on, and post-treatment. And we would like to understand, can the foundation model from the pre-treatment states tell us if this is going to be a responder or non-responder to immunotherapy? How are usually biomarkers computed? They're often a combination of kind of counting cells, analyzing neighborhood structures, looking at particular cell types and kind of their dominance or ratios. In the end, a foundation model that really understands a tissue, its spatial composition and cell type identities should have signatures that we can extract. And here, this is exactly what we find, that are going to really somehow summarize the state of a tissue, if it's going to respond or not. So we find here biomarkers for responders and non-responders that are highly predictive if this is going to be basically highly predictive for response. And this is much more, like significantly more predictive than the original Nature study, which was an 8-page paper over like one of those rather handcrafted biomarkers. Here, this is just like one of the capability that the same model that I showed you before also can access. Here in gray, you see kind of rather kind of classical CD8, CD4 B cell ratios and how much stratification they would give. What is great about those biomarkers is they translate to independent cohorts. So again, we take a clinical cohort the model has never seen during training. We can map them into the same space. We can apply the same biomarkers that we identified on the previous cohort. And now we can look at the ultimate stratification into high, low, and medium risk. And again, the overall kind of concordance index of kind of stratification is better than with classical methods. The biomarker from the clinical study before, we can't test it here because this is a new clinical study, has a different set of protein markers. And suddenly the previous method doesn't apply anymore, right? Because they're bound to the markers that we measure, while our method doesn't care which marker is measured. This also leads to the last, right? We will never be able to collect everything from the same patient. So can we use this to somehow steer what should ideally be collected, right? Where should we focus our effort? Which patient should we screen deeper? Again, we have a foundation model, same thing as before, that allows us to map into this representation space that we then use to predict certain downstream tasks. Can we now assess what the model knows already, right? If I give the model only one modality here, can I assess how much the model knows? And based on this, or can the model itself assess what it knows already, given that it has only been instantiated with one modality or one marker, can it now understand what it should collect next sequentially? This is exactly what we developed. It's a test time algorithm, so something that is at deployment when actually used, no training. That is a reinforcement learning framework. Reinforcement learning is usually used for those LLMs for post and test time. And now it kind of sequentially asks to basically collect a set of markers to then be good on any type of downstream task. So this is here really assessing what the model knows, such that it's good on any type of question we might ask. And you can see here we tested on the ability to cell type. And in light, in blue, you see basically having a model with full access to the full panel. It only needs 8 markers to kind of reach very quickly and much more efficiently than just like an oracle or like kind of random policy, the basically ideal state of being able to cell type accurately. What is the overall vision, right? Ideally, we are integrating those foundation models into molecular tumor boards. How can we make those models accessible? We are looking at somehow agentic systems that have access to those models that can interact with basically humans. This is, of course, a lot of like future music. overall, what does it need? A lot, right? So, I mean, this is really active development, initial ideas. We are working and have a high reliance on the supercomputer in Switzerland. Sensitive data is not on the supercomputer, it's on the hospitals, right? So we also need to have a new level of infrastructure in Switzerland that even allows thinking that way. And so NIPO is starting, I think, in April, which will have many of the building blocks that I mentioned during the talk, as well as really this core infrastructure with the SDSC, CSCS, to let us kind of think and design the kind of future of precision oncology. And with this, thanks so much. So this is all joint work with Andreas Wicki, Olivier Michelin, all work done by my students already, so they are listed here. It's a collaboration with Pascal Frossard, who is leading the AI Center, as well as Olisa Uriccio. And then there's still a lot of work that has been done with Gabriele Gut and Lukas Pelkmans, and of course all the thinking with my PhD advisors, Andreas Krause at ETH, and my postdoc host Aviv at Genentech and Jura at Stanford. Thanks so much.
Thanks a lot, Charlotte, for showing all these unpublished data. We have questions here.
Yeah.
Thank you very much for this really great talk. I was wondering, being the fact that there are many right now 3D models that maybe are validated for kind of simulating or mimicking some clinical features that are observed in patients, do you think that these models may be used for training your virtual patient features, because you can have many more kind of biomarkers that you can have a look at, and maybe have more kind of adding complexity to what you already have. And also the other way around, do you think that your virtual patient can be used also for kind of validating 3D models that can be sort of predictive then with functional screens?
So I mean, so right now, basically all we've achieved so far is like kind of thinking again this, how do we basically do some form of homogenization between tissues, right? So now we started kind of adding a genomics perspective. So I have a postdoc between Andreas and mine is looking into this. So this is just like adding one more modality, and we're looking into kind of health records and their kind of temporal resolution. So I think we are very far from anything virtual patient. I think with the virtual tissue, we reached a state where we kind of are pretty impressed what the model has captured and how much more it has captured than something we can simply extract from the raw structure. Also, we are quite impressed by how many low-plex panels we can map into the structure. So sometimes for the drug screenings, we have actually only a 5-plex, and it's still pretty well kind of anchored. So I think very far from kind of answering your question. And I think at this point, I go the strategy one modality at a time. So, yeah, I guess.
There's one further question in the back there.
Hi, very interesting results actually. I was wondering, where do you see the models from which you predict protein expression from routine histopathology being used in practice the most? Because at least what I've seen in the literature so far, it's mainly prediction of marker genes for cell typing and not so much, let's say, functional markers. So I'm wondering whether you see them more used for pathologists or actually in the clinic. Thank you.
So these models that basically allow us to kind of virtually stain tissue, I see them coming in. I mean, we already see them coming in kind of in some of the predictions that we do. For example, if I have like a cell typing task, right, that I'm actually doing, right, and just like even if I have spatial proteomics, but I have only a subset, actually completing some of the markers and being able to kind of verify, okay, so if this would be virtually stained, right, like is overall the kind of cell typing, like do some of the downstream tasks and also improve. We do this. What I think is more actively interesting is like kind of looking at the processes that are anyway happening in pathology and kind of like really pointing to, okay, so if I predict this, I could do this. So could we actually do a very kind of cheaper IHC stain for those type of markers? So I think in the translation, it will be more kind of mapping to a modality that is anyway measured, or helping intrinsically a computation to be more accurate. So at least these are the two obvious ones. And I think everything else we should prospectively test, right? Do these virtually painted have any additional clinical use? I wouldn't dare to make a statement, right? We are currently testing if it's possible. I think virtually painting from only an H&E, I will remain skeptic, right? Like many people are reporting Pearson correlations and they're pretty low, right? So.
All right, thank you. We have to move on. Final round of applause for Charlotte. Now, the next element in our program are 11 short pitches that were selected from the abstract submissions. We will have all of these 11 people presenting one by one, and I would ask the next ones to already come down, please, so that you can be set up and we don't lose time between pitches. Now, we have 2-minute presentations, and then there is time for one question. We'll also have the opportunity to look at these posters that were selected for abstracts. Now, the first speaker is Simone Ambrosini from Stephanie Kreutmeyer's lab at the Institute of Experimental Immunology.
Yes, so hello everyone. I'm really happy to introduce you to my project today, which uncovered the role of endogenous T cells in CD19 CAR T cell therapy associated toxicity. So as you can see from the image, CAR T cells involves the collection of the patient's autologous T cells to then modify them ex vivo so that they express a CAR receptor, and when they are infused back into the patients, they kill the cancer cells. By looking at the CAR T cell therapy-related mortality, we see that around 7% of the death is due to no-relapse mortality, which is strongly correlated to CAR T cell associated toxicity, which you can see represented in this table. To detect underlying immunopathologies, in my study, we performed high-dimensional spectral flow cytometry, and then this is the results. While CAR T cells are recognized as primary mediators of toxicity, our study uncovered the role of endogenous T cells, which in fact imprint subsequent CAR T cells profile, as shown by increased effector and Th1 skewing in the CD4 T cells at apheresis, reappearing in the CD4 CAR T cells and linked to more severe toxicity. Endogenous T cells also shape the pro-inflammatory immune environment at CAR T cell infusion, as shown by increased pro-inflammatory cytokine expression at baseline. This indicates the endogenous T cells as predictive correlates of toxicity. And then if we move on from pre-infusion to post-CAR T infusion, we detected bystander T cells as K cytokine sources and so contributors to immunopathology. For example, we detected a shift from helper to cytotoxic bystander T cells with increased interferon gamma expression shown in hematotoxicity. To find out more about my results, I'm really happy if you come visit me at poster number 1.
Will do, Simone. Short or urgent question for Simone? The bystander T cells, are these tumor infiltrating or in the circulation? What do you think?
No, I consider that maybe also the one that recognize the tumor, but also in general the one that are activated by the CAR T cell induced inflammation. Yes. Yeah.
Next speaker is Noemi Bührer. She is at the Clinic for Radio-Oncology at USZ.
Hi, so I'd like to pitch you our poster on personalized elective nodal irradiation in head and neck squamous cell carcinoma. So let's start with an example. A patient presenting with a T1N1 base of tongue tumor, which is lateralized and also shows clinical involvement in lympholevel 2, ipsilaterally. And let's assume it has been decided this tumor is going to be treated with radiotherapy. So what we usually do is we irradiate the primary tumor, any clinically involved lymph node levels, but in addition to that, we also electively irradiate lymph node levels which are at a high risk of occult disease. And the question is a bit, which lymph node levels do we irradiate, since we don't actually see the disease on our clinical diagnostic modalities. And this is what we would do according to our current guidelines. So we would irradiate IPSI and contralaterally lymph node levels 2 through 4. And as you can see, this is quite an extensive bilateral elective irradiation. And the guidelines which inform this are based on the overall prevalence of involvement, and they do not really account for the patient-specific risk which is at hand. And this is something we wanted to change. So in the first step, we collected a bunch of data, which is also publicly available on our webpage. And in a second step, we built a model of lymphatic progression, which we were able to train with the data we've collected. And once we have our model, we can kind of go back to with what we started with our diagnosis and kind of feed this into our model. And our model will then estimate the risk of occult disease in each lymph node level given the patient-specific diagnosis. So this is what this would look like for this specific example. So for example, in ipsilateral lymph node level 3, we would have a predicted risk of 11% for occult disease. And based on these risk predictions, we can implement a new treatment strategy, which kind of where we could electively irradiate only the levels which are at a high risk given the patient-specific diagnosis. And if we compare this to what we would do according to the current guidelines, we see that there's quite a significant volume de-escalation. And yes, this is exactly what we do at our clinical trial, which is currently recruiting. Yeah, if you want to find out more, please come to our poster. I'd be happy to explain and answer questions. Thank you.
Thank you. Naomi, any questions for Naomi? Yes, in the back there, please.
How do you determine the cutoff? Because I saw that one didn't have like 0% of chance of occult disease was not shown to be irradiated. So how do you determine the cutoff of what you should irradiate and what not, given the chances?
Yeah, that's exactly like the big question. And I think it really depends on how much risk we are willing to take. But I think there's not really a general, true answer, because that's really kind of up to the clinicians, in my opinion, and not really up to me, who is just doing the models.
Thank you. Our next speaker is Simona Ulrich. She is in Raphael Morscher's lab at the Children's Hospital.
Good afternoon, everyone. So my name is Simona. I work in the pediatric cancer metabolism lab, and in our lab we think a bit different. While in precision oncology we are looking for any actionable targets, we are specifically looking for the metabolic ones, either metabolic genes or nutritional vulnerabilities. And here in this project we have looked for metabolic vulnerability in T-cell acute lymphoblastic leukemia, and in the beginning we performed an in vivo screen testing 20 different diets in an orthotopic DLL model, where every diet lacked one of the proteinogenic amino acids. And what really stood out of this screen was histidine restricted diet that extended the survival of these mice by 2 to 3-fold. So we asked ourselves why histidine is so important for T-cell lymphoblastic leukemia. And therefore we have performed a multi-omic profiling consisting of RNA-seq, ribosome profiling, and proteomics. With the ribosome profiling, we could have proven that the ribosomes are colliding at the histidine codons, which triggers GCN2 stress response driven on the translational level. But at the other end of the spectrum, we have seen that the cholesterol biosynthesis is massively downregulated in those cells. And we thought this could be the second hit we can use to our advantage. And to prove that cholesterol is really an essential mediator in the histidine depleted cells, we have performed also an in vivo experiment where we could prove that cholesterol supplementation partially reverts the survival benefit in the mice that are on the histidine depleted diet. And the leukemia simply grows faster when cholesterol is supplemented. So if you'd like to know a bit more how to decrease your cholesterol levels, please come to poster number 3.
Okay. Questions for Simona? What would constitute a histamine a low diet? How can we reduce histamine?
It's actually one major factor would be replacing animal proteins by plant protein.
Okay, always a good idea. Other questions? If not, we move on to our next speaker, Lupindra Chatterjee from Swiss Institute of Allergy and Asthma Research in Davos.
Yeah, so the affiliation may be a bit confusing, but I promise I'll speak about cancer. Starting with, so as you saw in my title, I work with lymphomas. And why? So as we all know, the lymphomas are really heterogeneous, multiple different entities behaving really differently. And despite being studied for a decade or even more, there's still some unresolved questions. So what we want to do, or what we aim to do, is add the functional layer of proteomics on the available prior information and try to understand the disease mechanisms better. But large-scale proteomics in context of lymphomas have been limited so far due to technical challenges. So the first challenge that I had was to set up a very robust and reproducible pipeline, a proteomic platform that can profile thousands of patients' liquid biopsies and tissue biopsies in a very short time frame. So I do baseline. So with the established method now, I do baseline proteomics. It's a project that's funded by the Loop Zurich. It's called Polar, where I work with PBMCs as well as sorted B cells from the patient and fresh frozen as well as FFPE tissue material. And I look into drug response profiles with ex vivo drug perturbed PBMCs. Then we have a semi-automated and very standardized mass spectrometry platform using which I analyze the samples. So what do we get out of these samples? So I've finished measuring a full cohort of mantle cell lymphoma. And then from the PBMCs, we quantified all the different cell type markers. These cell type markers can be used to deconvolute specific cell types, something that Tobias will introduce later. But we also identify the diagnostic markers for subtype classification. Then what do we do from there? The next question is, of course, how these markers change or the proteins change on drug treatment. I mainly work with BTK targeting drugs in context of CLL. And there we saw that despite the patients behaving or responding very differently, the targeted protein degrader had much superior effect on the target than the inhibitor. But then we found that the downstream effects were very converging between the inhibitor and the degrader. But I think our most interesting finding was the dose-dependent effect of the degrader. I worked on more doses of these drugs and also on different lymphoma, and I would be happy to discuss about it if you come to my poster. That's 4. Thank you.
Okay, thank you, Lopamutra. Very quick question for her. If not, we'll just visit you at the poster and move on directly to the next speaker. So, Truhn from the University Hospital of Zurich.
Hello, everyone. I'm a scientific researcher from Westside, and I'm glad to share one of my PhD projects, the fluorescence PCR microneedle array for spatial tumor biopsy. And Speaking of the tumor diagnosis, biopsy is still the gold standard, but it requires a lot of lab work and costs some time, and it needs experience to distinguish the abnormal tissue. Most importantly, it loses the spatial information because of the 2D tissue slicing, and it misses the tumor heterogeneity because which is the key point in precision oncology. So to address this, we present a well-inspired microneedle platform with a heterogeneous variability for 3D sampling and on-needle PCR reaction. The substrate is made of the hydrophobic polymer with a nanoparticle assembled on top, which can separate each needle peak for independent reaction. And the needle peaks are made of the graphene aerogel and hydrogel hybrid, which has good hydrophilicity, absorption capacity, and mechanical strength, which can penetrate the tissue specimens and collect the tumor DNA biomarkers in a 3D manner. In general, the detection begins by cutting the tissue samples into some fragments and then penetrates them with the microneedles. After removing the fragments, the fluorescence PCR will be directly conducted on the needle tips, and then we could collect the fluorescence information of each single needle tip and use the intensity as well as the fluorescence position to reconstruct this this whole tissue and to locate the tumor sites. And we demonstrated the detection of 4 lung cancer mutations in the standard solutions and the mouse model, as well as the 16 patient samples with the consistent results with the clinical pathology. And we also enable the 3D tumor reconstruction of the mouse tissue. So to sum up, we present a platform that enables the fast spatial and minimally processed biopsy with the potential for the intraoperative precision oncology. If you are interested, I'm glad to discuss more at poster number 5. Thank you.
Very amazing new technology. How far removed are we from implementing this in diagnostics routine?
It's conducted in China, so after I graduate, it's handled over by another PhD students, and I think it's moving on to put it with some sampling techniques like the biopsy needle. So it's going on.
Thank you. Next speaker is from the University of Basel, Anna Salamero.
I hope you can hear me well. Well, indeed, I'm going here to present my work as PhD student, which I did in Frankfurt and in Tübingen. So we were working with brain metastases. They have already been introduced, so they have a very poor overall survival. And most patients that are diagnosed with brain metastases also receive radiotherapy. And indeed, what we wanted to do was to find the strategies that in combination with radiotherapy could improve the prognosis of those patients. So in this context, for us, it was very interesting to look at the adenosine pathway because we know that upon irradiation, there is a release of ATP. And this is kind of linked to the adenosine pathway because adenosine pathway, mainly through the enzymes of C39 and C73, induce the degradation from ATP into adenosine, and adenosine is regarded as an immunosuppressive. So what we did was in a preclinical model of brain metastasis, we were able to characterize expression of the members that can be targetable in this pathway, and we found that there was a specific cell expression according to these different ectoenzymes, being C73 expressed mainly in the tumor cells, while the others were expressed in the stromal compartment. And what we found then very striking is that in a preclinical model of brain metastasis, when there is this specific expression of these ectoenzymes and adenosine receptors in different cells of this pathway, we found that the combination of radiotherapy and the inhibition of this pathway, as shown here, was the one that has more effects in controlling the tumor growth and also in translating into a better overall survival into those mice. And then when we were analyzing the tumor immune compartment by different techniques, we could see that it was also in this condition where there was less detection of exhausted T cells and also more presence of macrophages that are able to have antigen presenting capability. This was reproduced in this model with this specific combination because we We are characterizing other preclinical models where the expression of these ectoenzymes and adenosine receptors is different, and it seems like the treatment needs to be tailored according to the model. Similarly, this is also something that we observe in human brain metastases. So it also really depends there on which expression of the members are there, and also in which immune cell or tumor cells, and this then can be then correlated to patient prognosis. Thank you very much.
Thank you. Questions for Anna? Does not seem to be the case. Then we move to another Anna Popova from the Karlsruhe Institute of Technology. We got abstracts from not just within Switzerland, but also from abroad.
Yeah, hello everybody. So my poster title is Functional Precision Testing in Myelodysplastic Neoplasms, Expansion-Free Drug Profiling from Limited Samples. So my name is Anna Popova, and I'm coming from Karlsruhe Institute of Technology, and this work is conducted in collaboration with University Klinikum Mannheim. So myelodysplastic syndrome is a myelodysplastic disorder with a risk to conversion of acute myeloid leukemia with a limited therapeutic option. The functional profiling is limited by a low number of bone-derived, patient-derived cells, CD34 positive cells. Very often the cells are getting expanded in vitro in order to get more cells for drug screenings. However, this can change the properties of the cells and also the drug response of them. So our solution is miniaturized screening, so we can conduct the screening on the not expanded cell populations. So the droplet microarray platform that you can see on the screen consists of 672 hydrophilic spots on a superhydrophobic background, and due to the high difference in wettability, we can form the stable array of 200 nanoliter droplets, each of which can serve as a nanowell for culturing and testing the cells. So in our workflow, we're conducting the screening of 27 inhibitors, 11 concentrations, and 3 repeats, which all together adds up to about 1,000 experimental points. And by using of only 200 cells per droplet, we're able to conduct all the screening on about 200,000 cells in total. So here we can see some example dose response curves obtained only from 5 drugs from up to 8 patients. On the left, you can see the dose response profiles for cytotoxic drugs. With this low number of cells, we can get a stable, robust dose response with a good zeta primes. And on the right side, you can see examples where we can see the different response from different patients, from some showing the full resistance and some showing their sensitivity. We also conducted the comparison of the cells that were not expanded versus the same cells that were expanded in vitro. Here you see the drug sensitivity scores, that red presenting full resistance and changing from yellow to green represents increasing sensitivity. So on the right panel, you see the response of expanded cells. On the left panel, you see the response of the non-expanded cells. So what you can see that the cells on average becoming more sensitive to the treatment, while the stem-like cells show mostly resistance. So with this, I hope I could spark your interest to our screening platform. And if you have an interest in, I'm happy to discuss it on poster number 7. Thank you.
Thank you. How do you discriminate between normal and dysplastic cells in the mixture?
We usually receive already sorted cells in the vial, so we're actually just seeding them on the platform.
Mm-hmm. Thank you. Thank you. Okay, our next speaker is Tobias Scheithauer from ETH.
Yeah, hi, my name is Tobias Scheithauer. I'm a PhD student in Valentina Boeva's lab, the Computational Cancer Genomics group in the Department of Computer Science at ETH. And I want to quickly talk to you about bulk proteome deconvolution. So as Lopamura, I'm also part of the POLAR project, in which we want to study lymphoma on proteome level. And the data that we have is mostly mixed samples. So we take a blood sample and have all the different cell types. And then we want to profile the individual cell types. And one way, I'm not sure if the pointer, yeah. So one way would be to do this manually and to separate the cell types manually in the lab. But with computational approaches, we hope that we can use just the data from a mixed sample and computationally deconvolve the signal into the individual cell type signals. And this is quite established for transcriptomic data, but not really studied whether we can do this for proteome data, so for the mass spectrometry data that we generate. And my project here that I'm presenting is really understanding, can we do this, and benchmarking existing tools for the deconvolution. And for this, I created a comprehensive benchmark across very different axes in terms of data generation and how we generate signatures for these tools. Also across different settings for cell type compositions. And one of the key results that we see is that we probably need to create new tools for the proteome data, because with the existing tools that are mainly for the transcriptomic data, we see a stark decrease in performance when we compare settings that are typically evaluated, which we frame as idealistic, in which signatures and the mixtures are generated from the same data set, compared to a more realistic setting that we will have, in which we have one data set to generate signatures and then use another batch of samples to really profile the tumor samples that we have. And if you want to know how the signature creation and so on plays a role, and whether we can improve this in the future, visit me at poster number 8, which you can find right outside that door.
Thank you very much, Tobias. Any questions for him? We do have time for 1 or 2 questions. Nope, that's not the case. Then we move on to Margarita Liaroux at the Department of Computer Science at the University of Geneva.
Hi, I'm Margarita, and my poster is about trajectory inference in healthy and disrupted hematopoiesis using flow cytometry data. So to begin with, trajectory inference methods order cells from immature to mature and assign them into different cell lineages. To do this, they compute the pseudotime value of each cell, which represents its degree of maturation. In this way, we can track changes in marker expression patterns as a function of pseudotime during cell differentiation across each lineage. So we recently introduced TimeFlow2 to extend our previous pseudotime computation method by adding unsupervised lineage detection. So the method assumes that cells undergoing similar differentiation follow highly similar paths, and therefore groups the most similar paths together along pseudotime. We applied our method to flow cytometry data, and specifically healthy bone marrow samples that were gated for monocytes, neutrophils, erythrocytes, and B cells in different maturation stages. So the method identified 4 distinct pathways within each donor. You can see in the left figure the cells from one donor visualized in 2 dimensions and colored by their pathway assignments. We use the labels from gating to evaluate our results both qualitatively and quantitatively. And now, comparing to the ground truth data, we can see in the right figure that the method identified the expected number of pathways, and that also those pathways matched the populations in the sample. We also found correlated patterns across the donors that were consistent with prior knowledge of hematopoiesis. And we are currently comparing differences in cell proportions and marker expression patterns between healthy hematopoietic trajectories and those from patients with acute myeloid leukemia to help improve diagnosis. So thank you for your attention, and feel free to ask me questions during the poster session.
Thank you, Margarita. Question for Margarita? No. Then we will move on to the second to last speaker of this pitch session, Thi-Huong Lan Do from the University Hospital of Zurich.
Hi, you can also call me Lan. It's also enough. So as Lopa already showed you, you can kind of, among others, have 2 approaches to do cancer or comparative analysis between cancer diagnosis or subgroups. Or the other part that she presented was involving drug perturbation, and this is what I've been focusing on the last few years. So our lab is, among others, focused on using drug perturbation to resolve cancer pathways. And in my particular project, what I've done is performed a screen and generated over 1,000 transcriptional profiles by Okay, so on the left side, you can see also by selecting a cohort of chronic lymphocytic leukemia patient samples, where we have a lot of baseline annotations already available. So we have genomic data, transcriptomic, proteomics, and more. And then the screen entailed a selection of 10 small molecule inhibitors, and those were chosen to target all the main pathways like the B-cell receptor, MAP kinase, mTOR, PI3K, mTOR pathway, for example. Then we treated those cells for 2 days and then read out the ex vivo drug response either by flow cytometry, that would be cell death, but the main point would, in our case, be the dynamic gene expression by shallow depth RNA sequencing. And then we, for the analysis, what we first started with is, yes, you can look at the individual treatments and determine the mechanism of action, but since we have all the 10 different inhibitors at once for the same patients, what we did is also to compare the treatments against each other. And this enabled us to kind of to construct this pathway connectivity of drug effects. And with factor analysis, we were then able to, among those drug effects, identify co-regulated expression modules by the drugs. For example, this module would entail genes that are all downregulated upon B-cell receptor inhibition. So these are all the B-cell receptor dependent genes. Or at the bottom, you see a set of genes that are very specific to the inhibition of MDM2, XPO1, or BET1. And the next steps, what we then did was to take advantage of our annotated cohort and break them down into molecular subgroups. For CLL, one of those major subgroups would be IGHV. And there, for one, so you can have, again, at baseline level, take unmutated, mutated IGHV samples, look what is different. And with the perturbation on top, what it enabled us is to do, like, for example, you have ZEP70 and CD38 as markers, and to see how do these markers change after perturbation. On the top would be examples for stable markers. So if you inhibit the B-cell receptor, nothing happens, or nothing changes, indicating stable components. While at the bottom, you see 2 examples where after B-cell receptor inhibition, you see that this signature collapses. And another approach that we did, making use of this annotation, is to do drug genotype interaction analysis to see really how does or do we find genes with different behaviors in the presence of the drug, a driver, or both. And in this case, we found, or we could group all those genes where we found those interactions into different groups based on epistasis classification. And most of all, among, I think it was like 560 genes that we found with interactions, trisomy 12 was one of the drivers, where we identified more than half of those. And we could link that the presence of trisomy 12 in CLL led to an aberrant drug response to toll-like receptor, B-cell receptor, and the MAP kinase pathway. And if you want to hear more, I'm at poster 10.
Okay. Did you, before you actually performed your drug perturbation screen, did you try to use the proteomic and other multi-omic data to predict which drugs would work?
So I think we have, based on the baseline data, it has been extensively done to try to link like RNA baseline expression to the ex vivo viability response, but I think the result has been rather disappointing. But with this perturbation, we haven't done those prediction things yet.
Okay, thank you. And our final speaker is Veda Mamuzai from the Intercept program.
Hello, everybody. So I'm happy to present you my project. Anti-CD19 CAR T-cell therapy has increased response rates in patients diagnosed with relapsed refractory B-cell malignancies, but a major obstacle still remains. Relapse rates are as high as 50% with most of those patients, relapsing early after CAR T infusion. This shows that there is a high need for therapy optimization and intensification for patients with a high risk constellation. So how can we improve the overall outcome of non-responders? With novel tools for dynamic risk profiling and the establishment of those tools in clinical trials. The combinatorial in-depth analysis of first, measurable residual disease, second, inflammation status, and third, immune environment fitness within our INTERCEPT assay represent a precision medicine approach for early detection of CAR T-cell treatment failure. Patients, below you can see our study design. So the enrollment of the patients are at the time point of relapse. And here I want to also highlight our primary objective first, which is the complete risk profiling of the whole INTERCEPT assay post-CAR T infusion. So patients are enrolled at relapse and we collect samples up until day 28 post-CAR T infusion. And within our co-primary objective, we want to deliver the results shortly after the last sample is collected, so 10 days after day 28. Importantly, this will demonstrate the feasibility of the INTERCEPT assay in a real-world setting with repetitive, frequent assessments over short time intervals. The study emphasizes that longitudinal monitoring can be performed reliably while also generating clinically relevant results in a timely manner. And for more details, I'm happy to invite you to my poster and I can tell you more about our current update of the trial.
Thank you. Any ideas yet as to what is predictive prior to treatment?
So I think one factor is definitely the tumor volume or the response until we infuse the patient, since they often receive tumor bridging therapy to reduce the tumor. So I think that's for sure one very predictive part, but I think all the components are very interesting for the prediction in combination.
Mm-hmm. Do you also look at the tumor microenvironment?
I think
at that stage, do you biopsy?
So within our trial, no, but if there are biopsies and materials left, then yes, we look into that as well, yeah.
Okay, thank you. And thank you to all 11 speakers of this poster pitch session. We now have 3/4 of an hour of lunch break. That lunch break is also designed to have you take a look at the posters. So we grab lunch down here and we move up to the posters and have the food in front of the posters to give the poster authors an opportunity to actually talk to us. Please.
Dear ladies and gentlemen, dear colleagues, it is my pleasure to welcome you back to this afternoon session of the 7th Zurich Precision Oncology Symposium, organized by the Comprehensive Cancer Center Zurich. On behalf of the medical faculty and in my role as Vice Dean of Research, I would like to thank the organizers for putting together such an excellent and forward-looking program. For the medical faculty of the University of Zurich, the Comprehensive Cancer Center Zurich is a central pillar of interdisciplinary precision oncology and a key driver of the international visibility of the Zurich research ecosystem. Precision medicine, particularly in oncology, is a key strategic focus within the UMZH. And it is inspiring to see how a strong and truly interdisciplinary community is emerging in Zurich, closely connected to leading efforts across Europe. This afternoon session focuses on translational and clinical research in hematologic malignancies, highlighting how innovative therapeutic concepts, functional approaches, and data-driven strategies are advancing into a previously uncharted territories of precision oncology. I wish you an inspiring and stimulating afternoon. Thank you very much.
Thank you very much, dear Jana. We are actually very proud to have the Vice Dean of Research coming from our institution, Children's Hospital Zurich, and Jean-Pierre Bourquin, and together with Thorsten Zenz, who is actually the mastermind of this meeting. And I would like to thank you very much on behalf of the other colleagues. And our next speaker is Olli Kallioniemi, a pioneer in precision oncology that does not need to be introduced. Today is going to talk about a project on hematologic malignancies, and you're really our experts in data integration. Thank you for being here.
Okay, thanks so much, JP, and thanks to Thorsten and the other organizers for invitation to this fantastic meeting. So it's always great to be in Zurich, where science is top-notch and the sun is always shining. So I have had a long period of leadership, first at FIM for 8 years and then 9 years at SciLife Lab. And now that both of them ended, or the SciLife Lab ended a year ago, I thought that I'll just be able to do research and just do my stuff. And yesterday that we were at dinner, I kind of complained that I ended up in a complicated assignment, but Thorsten said that that's interesting, that you should talk about it. So I put one extra part into this slide set, which I would never recommend anybody to do for a 30-minute talk. So I will have to rush through the end of the talk a little bit, but bear with me. So before we go to leukemia, I'll start with health data. But before that, I have a few disclosures here. None of them are really relevant to what I will be discussing today. So what we were asked by the government in Finland was that, can you help us to kind of a plan, how should we deal with health data in the AI era? And do with health data not within one hospital or one region, but the whole thing, the nation. And this was supposed to be a 20% assignment for 6 months, and then we're done with it. So it turned out to be a year of 60-70% assignment, and it's still not done. But it's published now. And the idea was that, can we sort of describe what the bottlenecks are in national utilization of health data, present a comprehensive national solution for this, particularly from the light of the AI utilization, where you need massive data sets to learn and teach the models, ensure the compatibility with the international efforts, such as the European health data space. So we call this the Finnish health data space. And then go more sort of deeply into how could it actually be implemented and not just be left at the sort of a loose idea. So it's now published, and even though it's only published so far in Finnish, just download it and translate it by AI, and it's all readable to anybody now these days. So this may sound somewhat familiar to you, regardless of which country you come from, that although we have in the Nordic countries the registries that cover the whole country, all of the other health data is deeply siloed. So we have 21 healthcare regions, or the Helsinki University Hospital region is kind of yet another one. They all have their distinct EHR systems, and there's long vendor lock-ins in all of them, and they are not really standardized in terms of all the nomenclature and so forth. It's complex legislation-wise, and there's national legislation. Now there's also the EHDS European legislation that has come on top of it, and many data controllers, and therefore many decision makers if you need to get permission to use the data. So just to illustrate how difficult it is to get national level data for research or secondary use, I'll show you an example of the FinGen project, which covers 500,000 Finnish people, their genomes, and also their comprehensive registry-based health data. it requires 200 permissions and contracts or agreements to be in place all the time for these 500,000 people to be linked and those links maintained for healthcare. So this obviously doesn't make any sense. So can we do something about this one? Well, as I said, the fragmented silos on the left are almost impossible to break. They have long-term contracts, and that's just an unfortunate thing. But the plan that we kind of set out to do, and I'm not going to describe to you all the details, was that can we layer a secondary layer on top of the whole country that would be easily accessible and therefore utilized in a more simple manner, and leave all of the underlying sort of complexities in place. Because the primary healthcare, that's a little bit difficult to mess up with too much. So to cut a long story short, we went to a very simplified model. So if you needed 200 permissions before, we thought that let's make a system that requires only one permission. So you have a national permit body here, which actually exists already today. One coordinator of all of the national health data, and then one body where you give that permission to. So if you want to use national data, you affiliate yourself with a national R&D institute. And we thought that this would be great. And it's just that it's going to take a little bit to make this happen. So this is just our idea. Don't think that this is actually accomplished. And the moment that we published our document 3 days later, there was already a letter at the minister's table saying that very prominent people don't like this plan. But nevertheless, we push forward. So obviously, we are not starting from scratch. There's a lot of infrastructure already. And we started to think that we should start from existing, like what we call pillar projects. FinGen is one of the pillar projects here. There's a national cancer research hub, real-world evidence, computational pathology, national population health AI model. I'll show you an example of this one in a sec. Finnish brain data space, and then drug effectiveness monitoring. All of these things are either funded, ongoing, or planned activities that could be bound together and represent sort of a joint activities under this umbrella that we were proposing. This is the health data, the national health data and analysis that Andrea Ganna at Finn is working on. That actually shows quite well what is it that we actually want to achieve in the future. This is a database that Andrea is collecting from the registry. So this is not the deep medical data, not the electronic health records yet. But we have a way that he can access 7 million Finnish people who have over the years had 620 million diagnoses. Obviously, some of these recordings in the records are repeated one after the other. There's 1.1 billion prescriptions. That's only in the healthcare setting, not inside the hospitals. 1.4 billion lab tests, and also some education and socioeconomic data. So the idea here that Andrea is pursuing is to put it all into a foundation model and try to develop a full national scale model of healthcare, plus also all indications and all patient paths and so forth. So you could envision that such a database or a foundation model can then be queried for different types of purposes, and maybe there needs to be different types of models for different types of purposes as well, and so forth. But to motivate this to the government decision makers, we also, and to explain it, what it actually means, also for the healthcare profession, I've been using this slide, which kind of illustrates what's the doctor's traditional way of treating patients. They are good doctors because they have learned their books and they know what the medical information is all about. And then they have the doctor's experience, the practical experience of having many physician contacts with the patients. So over a career of a doctor, if you see 20 patients per day, maybe you get up to 100,000 patients over your lifetime. So in the AI era, if we had this kind of a foundational models, we could have this AI model help the doctors to deal with this thing. First of all, the AI would remember. It's funny how you kind of drag this and it doesn't go where you hope it would go, but nevertheless, it will not just read or has already read, but also remembers all the literature and the guidelines. But it would have this real-world evidence or real-world data of all of the patients and all the patient paths and longitudinal follow-up up to 50 years, not with all of the prescriptions and lab values. They are only for the past 10 years, but that this would be a collective experience of all doctors. So then that would hopefully illustrate how powerful this could be in the future and how it would also help the patient, not just the doctor. Then everybody is asking, well, what's the cost of doing something like this? Well, we don't really know, and that was not part of our plan. But we started to make a calculation entirely theoretically that the amount of money that the government puts to health and social care is 27 billion euros per year. And we just said that what if we put 0.2% of that? That doesn't sound a lot of money. What if we put 0.2% of that money and create this national thing? So that would be 54 million euros dedicated annually to doing this, which I would think can do at least quite a bit of things on the research side. So that's our proposition to the government. What do we then say? That, well, we have no idea how would this be helpful, and we haven't done any cost calculations, but based on published information, what if you just expect a 5% improvement in efficacy of healthcare if you have the data and AI? Well, that would be already 1.3 billion as a benefit. So the cost benefit of this sounds, at least on a theoretical exercise, quite good. Well, that was the thing that I've been sort of running in a swamp, trying to get this done. But let me now switch to the research and the data-driven hallmarks. Many of you have heard me talk about the technology-driven, data-driven, AI-driven revolution, and now how we would also like to have jointly, all of us, a translational revolution in medicine, because the left side goes so fast and this side is quite more difficult. And like many of you here, and great to see so many excellent talks along this type of a research focus, you can analyze genotype, you can get a deep phenotype even in a single cell level, and you can analyze the function of the cancer cells and try to integrate that towards a decision on how a cancer is likely to respond to treatment. But it's not very easy to integrate all of this massive amount of information. We've done this on the FIM side, and more recently on the SciLife Lab KI side, a multi-omics investigation of AML using proteomics, drug profiling, mutation, genomic transcriptomics, and DNA methylation, integrating all of that data, and taking a hallmarks approach to analyzing and interpreting the data. And the hallmarks approach obviously is something that comes from the cancer hallmark concept. But essentially, in our case, it condenses 90 million data points to 11 data-driven hallmarks that we think actually encompasses most of the informative data in the entire data set. Skip 3. And this is obviously inspired by the great cancer hallmarks paper, which obviously is a conceptual paper which talks about predefined sort of a phenotypic characteristics of cancer that are common to most cancers. But it isn't something that can be measured. It's not something that can be directly linked to particular pathways in a particular cancer. It's just a conceptual way, but very important, obviously. So we are not working to define the hallmark, the Weinberg-Hanahan hallmarks. We are working to define data-driven hallmarks that are computationally derived from the raw data set. And this picture never comes through from a Mac to a PC. Well, but don't pay attention to the messy picture, but this is unbiased data-driven, tailored to any cancer type that you can gain that information from multiomics information, going from genome to phenome to drugs. It's quantitative, linked to molecular data, and can be applied to diagnosis and therapeutic approaches, and can be personalized to each patient. So again, we're not defining the Hanahan hallmarks, but a new way of defining cancer hallmarks. This is a little bit of a better picture, but it's not even meant that you see all of these things. So this is the different hallmarks on the axis around this, and we have given them these names here that you can debate as to what the real name should really be. In each of these hallmarks, there's a contribution from the various types of platforms that have been applied here. I'm not going to go through this in detail, and each of the hallmarks has actually two ends. There's a positive and negative end to the hallmark. The current version has been done with the MOFA, multi-omic factor analysis, but there's obviously multiple AI ways that you can try to arrive at this as well. Just to illustrate one hallmark here, hallmark 2, that happens to be something that links or goes from a immature precursor to a differentiated monocytic cell type. So if you look at the transcriptomic response, in the top thing or proteomic response here, DNA methylation and the mutations, you see what are the top sort of a genes or molecules linked to this particular hallmark, and the value goes from negative to positive, and everything is obviously aligned so perfectly because that's how the algorithm works, that it lines the things that are associated with each others. I could describe some of the others, but for the lack of a time, I'll just skip forward. And this is like the frequently asked questions that Berend was showing earlier. This is always the question. So which of the omics factors is then contributing most, and which are the omics factors you actually need? And the question is, it depends. So obviously you get, so each of the hallmarks has a different composition from the different platforms, and each of the platforms contribute differently to each of the hallmarks. So it is a little bit complicated, but we do think that there's a lot of value in having that multi-omics composition there, and you cannot easily reduce it just to like transcriptomic data and so forth. I should also say, which is sort of an interesting thing for many of the people in the functional precision format, that none of the hallmarks is defined by a drug response only. So you usually always find some of the molecular components that dictate drug response in leukemia. So this is not very well correlated with mutations, usually. The first hallmark is actually very much correlated. So the first hallmark is an NBM positive and negative. But beyond that, there's actually a lot of the mutations are orthogonal to these phenotypic characteristics that drive these hallmarks. So most hallmarks are let's go to the next one. This is the link to the drugs. And now you see that there's a less of a variability than in the case of mutations. So actually there's a lot of associations between individual drugs and these hallmarks. And hallmarks actually associate a lot better with, drug response, ex vivo drug response, associate a lot better with the hallmarks than the mutations do. And that's a really important sort of a basic conclusion. If you look at this, so most of the hallmarks have one or more drugs that are associated with them, and most of the drugs are only associated with one or a few hallmarks. So this is potentially something that could be used as a means of identifying biomarkers, meaning that the actual hallmark itself is the biomarker for a particular drug response. What was our most sort of a surprising observation was that the hallmark 8, so as you go in the computational analysis, it first defines hallmark 1 variability, then hallmark 2 variability, and so forth. And lo and behold, in the hallmark 8, all of the prognostic information was associated with the hallmark 8. which was a really interesting observation. It's also associated with the ELN classification, even though it actually doesn't have a lot, only genetic components. So you can derive the ELN classification from the transcriptional and phenotypic feedback. There's an association with prognosis in the DDHM8. So if the DDHM8 is high, the prognosis is favorable, and if it's low, it's really bad prognosis. So you could think about that. You condense this complex multi-omics information to these 11 parameters. So you determine the value for each of these hallmarks, and then that is the fingerprint of the patient in terms of all of the dimensions that are important from the angle of leukemia development. And just to show again that it's applicable to assigning therapy for patients. So this hallmark 2 that I already showed before, those patients have an excellent response to venetoclax and do not respond to MEK inhibitors. But if you have the hallmark 2 is high, and again, this is the monocytic differentiation hallmark, then you have the inverse link to the drug response. And similarly, I'm not going to go through this. You can link many of the hallmarks. So if you get a hallmark picture, you can pretty much suggest as to what the patient or what the cells should be treated with in terms of getting an optimal therapeutic outcome. Sorry, this is a little bit messy picture as well, but this is just to show that we've been able to validate this classification also in external data sets, although the external data sets typically only have transcriptomic information available. So that's a little bit sort of a less critical. So this is a way that we suggest that you could practice precision medicine through a data-driven approach, and each so that you drug the actual hallmark within each patient, and not just that you give a drug to a genomic subgroup as we often do today. Some specific examples, I'm not going to go through this. This is again for the lack of time. I'll just quickly explain the fact that there's a lot of interesting observations that could be clinically applied. Thioguanine response in the hallmark 5, or we've been also using the same approach to predict mitostoring. But since this is published, and again for the lack of time, I'll just skip that, just to acknowledge the Swedish group that has all of the recent AML data that I have been showing today. And now my last point as of today, how do we prepare for the future in terms of this translational revolution, as I was sort of suggesting? And when I say revolution, I don't mean that we do the revolution, we together, all of us have to do something to make this happen. So we recently were lucky enough to get funded, and the next speaker, Sascha, is part of this consortium to develop a trans-AML EU collaboration, which would actually go forward exactly from where we were left now after our own study, which is to take these data-driven hallmarks, re-identify them from larger data sets, and then try to make, in the course of a 3-year EU grant, a translational approach to see where we can take them forward. It's not going to allow us to run a clinical trial, and there's not enough money to run a clinical trial, but this is an exercise to really distill these hallmarks, make them assayable, clinical laboratory-ready tests that could be run in any laboratory. And we have both discovery groups, bioinformatic groups, clinical groups that are working together with patient advocates and also privacy preserving data experts and so forth. So the idea is, again, to start from these multi-omics views, and like was earlier discussed here, that we often don't have all of the data, but if you can link it to the hallmarks, you might actually be able to deal with incomplete data sets as well. Do this AI compression, and then the output would be what we call a translational toolbox, which is like you define the assay, you've defined the drug, You define the mechanism of action. You define the algorithms that are needed to define this from the existing data sets, and you develop some kind of a way that you actually report this out. That this would be the mechanistic way that we could then have multiple of these validated, put them in a clinical perspective, validate at least the ex vivo drug response, and do it in multiple labs to try to see that this can actually be implemented. And that would be one way to make these multi-omics observations go forward in a larger way. And obviously this is a scalable approach. This can be done in any type of a cancer. And therefore this is just an example of these things. So I will sort of stop here that I hope that I've at least illuminated some approaches that we can work towards keeping up with the multi-omic progress and the progress in the AI and so forth, including maybe also this health data space analytics. So thank you.
Thank you very much. Great. I'm sure there are questions. Markus.
So very, very interesting data. I was wondering when you come to these hallmarks, how far different technologies and different hallmarks are driving the diseases to be classified as such. Is there differences, or is it always the same?
You mean across the different technology platforms? Well, there are certain technologies that are really driving certain hallmarks forward and are much, much stronger on those. So it really depends. But there is often a contribution. So there's no hallmark that is just defined by a single platform. So each of the hallmarks have contributions of multiple platforms. But I would think that many of the hallmarks could be defined like with two ways. So maybe have a flow cytometric or immunophenotyping assay, and I have a RT-PCR assay. Those would be the two ways to take this to the clinic, to refine the multi-omics sort of a fingerprint to two types of things. Maybe some methylation assay as well. But that's the task of the EU project now, to distill how are we going to best and most reproducibly measure each of these hallmarks for them to be applicable in the clinic without having to do this expensive multi-omics thing. I obviously forgot to say that each one of these profiles obviously is very expensive if you do it in a clinic routinely.
Is there any role to look at this under therapeutic pressure in analogy to intercept, for example, the effort of...
Well, absolutely. That's another line of research. That how would you apply this to follow therapy? How much do the hallmarks change? And obviously, the treatment pressure most likely does have an impact on them as well. So at the moment, these are the diagnostic hallmarks. But then how will they change is another question.
Good. If there are no other questions, thank you very much again.
Thank you.
Yeah, so it's Olli already introduced the next speaker, so it's my pleasure to introduce Sascha Dietrich from Düsseldorf. Sascha and I have worked together for 5-6 years in Heidelberg, but he's now the chair of hematology oncology in Düsseldorf. And is an internationally renowned expert on multi-omics profiling of lymphoma and leukemia. And if that isn't enough, he's also now heavily involved in managing the university hospital as the vice physician in chief. So thanks for coming, and we're looking forward to your presentation.
Yeah, thanks a lot, Thorsten, for this kind introduction. And I'm very happy to be here and to experience the atmosphere, how it is discussed here about multi-omics and how to use it for predicting something good for our patients. Sometimes if you go into reality and discuss with other people, this mono layer view of things, just looking at the genetic level, is is very often around me. That's why I'm very much enjoying the atmosphere here. And the only challenge I'm a bit worried about is to speak after Olli, who presents projects involving the whole of Finland. So my perspective is a little smaller now. And Thorsten said that I worked together with him. Actually, he induced the excitement about precision medicine. So I was a physician in training, working with Thorsten together on the ward, basically, and in the outpatient clinic. And then we decided to work together, and I was a postdoc in his team, and we started to run the first project. So it's really, for me, very nice to be here and being invited. So doesn't go forward. So I want to try to give you my perspective of how I think about how we can predict drug response and what determines drug response. Obviously, there is the perspectives of the tumor cell itself. And the tumor cell itself can be characterized by different levels of multi-omics characterization. This obviously involves the genotype, the transcriptome, the proteome, and so on. And the tumor cell then itself is also embedded into the microenvironment and receives input from the microenvironment. And obviously, this is also an important contributor to drug response. And I will try to dissect this. And this is one of the first studies I conducted with Thorsten together. For me being the first author, Thorsten was the last author of this. So this was the big first drug screen we've run together. And here, the first exercise we did is we took all the genotypes here from chronic lymphocytic leukemia and systematically regressed this on all the drugs in our panel. And as you can see here, 60% of all the variants of drug response could be explained by one or two mutations or genomic alterations in general. So many of the drug responses can be explained with one or the other genomic feature, but there are also drug responses which cannot be explained by the genotypic layer. So, and inspired, all of this is inspired obviously by observation like this. This is the poster child. So here, this is also a study which is quite old now, but at that time, BRAF inhibitors were used for melanoma, and this mutation was also discovered in hairy cell leukemia, a very rare leukemia, and we were the first ones trying this inhibitor in hairy cell leukemia. It worked well, as you can see here. So this is the prime example of how you can translate an inhibitor from one to the other disease because the other disease has the genomic alteration. That works well, but also has challenges because if you use this type of inhibitor, you can eradicate the majority of the clone, but there is a substantial number of tumor cells persisting. And that's because these tumor cells receive input from the tumor microenvironment and they induce alternative signals, and you cannot eradicate these cells despite these cells harbor the BRAF mutation. It's not possible. So you need another drug which targets an alternative signaling pathway. Another approach, and we heard about this multiple times today already, is that you not only profile one layer, that you profile in parallel, for instance, the genomic layer, the transcriptomic layer, the protein layer. And we did this approach also here in chronic lymphocytic leukemia in different contexts. I'm just here illustrating one approach where we used DNA profiling, RNA profiling, and proteomic profiling in parallel. And we used the same approach Olli already introduced, an approach called MOFA, developed at that time. That was one of the first opportunities to integrate different data layers developed by Wolfgang Huber and Oliver Stegler in Heidelberg. And that's basically something like a PCA for multiple omics data sets. And you can arrive here at specific latent factors which describe the variance of the multi-omics data set. And if we do that here for the multi-omics data set for chronic lymphocytic leukemia, for the transcriptome, genetics, and proteomics layer, you see that you have latent factor 1 and latent factor 2 have loadings from all three different layers. We had as an input for this analysis, and they were characterized one layer by IgG, an important genomic driver of chronic lymphocytic leukemia, leukemia, and the other was mainly characterized by trisomy 12. So the major drivers, which were already known, can be characterized by RNA, so meaning transcriptome, the genetic layer, and the proteomic layer. So you find features of all of these layers which would be able to identify and characterize these latent factors. But then there are also other latent factors, and I just want to highlight here latent factor 9 of this analysis, because it was the only other latent factor which was significantly associated with a poor prognosis in this analysis. And the interesting thing about this latent factor is it had only loadings from the protein layer. So we were only able to identify this risk factor of disease progression based on the proteomic layer. So that means that some of the features of the disease you can explain by all layers, some of them only by one layer. And that already illustrates the limitation. If you just focus on one specific technique, you might miss a substantial proportion of variance of the biology or drug response. Another important challenge, if you talk about drug response from my point of view, and you might agree, is that the intratumor heterogeneity also has a big influence on drug response. So as all of you know, tumors are not homogeneous, they are heterogeneous, they consist of different types of tumor cells, and they have different characteristics. And this is here a single cell sequencing experiment of a diffuse large B-cell lymphoma, and just based on the transcriptomic layer, you can distinguish here nicely two different subsets, and because we used here CyTOF in this case, we were able to sort these two subpopulations apart from each other and perform separately on these subpopulations drug response profiling. And you can see that these different subpopulations have different drug response phenotypes. For instance, the green layer responds well to ibrutinib, a B-cell receptor inhibitor, whereas the purple subpopulation responds better to chemotherapy, such as cladribine, for instance. And we could prove also for following with a follow-up sample of these patients that with chemotherapy treatment we could actually eradicate the sensitive purple clone, and the green resistant clone gave rise to relapse in this patient. But the interesting observation, that was not the case in all of the patients we looked at, but obviously in this particular patient, we observed that no single genomic variant here from whole genome sequencing could explain the differential drug response between these subpopulations, further illustrating that the genotype obviously is not the full story, and that there's more than just the genotype which explains drug response. So we try to understand what's further behind the genotype, and we sorted a substantial proportion of these subpopulations apart, did transcriptomic profiling, and tried to summarize this in gene set enrichment analysis, as you can see here. And we found many different cancer hallmark signaling pathways which were up or down in specific subpopulations, contributing to the different drug response, obviously. However, what we could also find is that a significant proportion of the variance between the subpopulations could be explained by maturation phenotypes. In this case, we are talking about B-cell lymphoma, the maturation of B cells. So the maturation from naive B cells all towards plasma cells. So we found hallmarks which were enriched in subpopulations of the same patient in either naive B cell gene sets or plasma cell gene sets, for instance, illustrating that, and all of these B cells were belonging to the malignant clone. So we just included B cells which were obviously malignant. So that illustrates that other biological process can contribute to the diversity of tumor cells within a single patient. And we tried to prove that. Here we prepared a classification framework for maturation subsets of B cells by sorting them and generating bulk sequencing data. And then we used our single-cell sequencing data sets and used the classification approach to classify each single B cell from a single patient into one of these buckets, from naive B cells all towards plasma cells and memory B cells. And as you can see here, looking across the different B-cell entities such as mantle cell lymphoma, follicular cell lymphoma, and the aggressive lymphomas, diffuse large B-cell lymphomas, and marginal zone lymphomas, you see that in all the subsets, you have contribution to all of the buckets. So meaning that you have cells in follicular lymphoma, for instance, which have the phenotype of a plasma cell. So a mature plasma cell, which is CD20 negative. And just think about it, because follicular lymphoma and indolent lymphoma, we treat with rituximab, a CD20 antibody, and these cells cannot be targeted with, for instance, rituximab. And then we try to prove that also in the spatial context, and you see that these plasma cells, which clearly belong to the malignant clone, hide somewhere else. So they are not hiding in the malignant cluster, which is identified in follicular lymphoma to be the main spot of the malignant growth. So these plasma cells are in the parafollicular zones, but they are clearly belonging to the malignant clone. And obviously, as I said, if you treat these patients with rituximab, you would not target this cell subset. So I think this is an observation which I think is very important, that biological processes which are inherent to the cell or the cell of origin do not stop if the cell transforms into a malignant cell. So they continue despite the malignant transformation process, and you have to address this and think about this if you think about drug response. So I think it's fair to summarize that drug response, if you look at the tumor cell, is obviously a function of not only the genomic layer, but it's a function of genomics, transcriptomics, proteomics, and potentially many more. And this is, of course, further complicated by the fact that each single cell of the tumor might be different and contributes to intra-tumor heterogeneity. So one potential approach to address this is obviously to run a functional test which integrates across all these different layers and just read out what you want to predict in the clinic, which is drug response. So as Berend and others already described, we also have run here a prospective clinical trial, in this case AML, where we profiled the drugs. We can nicely show that that we can implement this in a clinical routine. We can report these results back to the treating physician within 5 working days. That works well. We can also nicely predict in vivo drug response. And if we, let's say, use the classification framework of the ELN network and use drug response on top of that, we can further distinguish between drug resistant and drug sensitive, or chemotherapy resistant and chemotherapy sensitive patients, which is nice. But as you can see here, the split is not perfect. And I think this requires a further optimization of the technique, at least in our hands. And as you can see here, as a proof of concept with the in vivo drug response or ex vivo drug response, we can also predict survival of these patients. So far, so good. So this is what I wanted to share with you all about the tumor cell. But if we consider the tumor microenvironment, so the first question, I think I don't need to argue that the tumor microenvironment contributes to the biology of the tumor. This is obvious. But the question is, do we really need to establish culture models which mimic parts of the tumor microenvironment? So I received this question very frequently when I presented these data initially. So I decided in my junior research group leader grant to run screens trying to mimic some aspects of the tumor microenvironment. And we, for instance, here collected a large number of soluble factors and systematically cultivated them with drugs and leukemia cells that worked well. We could identify a number of soluble factors which induce resistance, but in the end, it did not really improve the in vivo prediction of drug response for individual patients. So we were better in some aspects, but worse in others. So it's really, the whole system was not better, at least in our hands. This technique was not sufficient to improve in vivo drug response. Unfortunately, the same was also true, at least in our hands, if we used a co-culture model of stroma cells and leukemia cells. And we also did that here very systematically. We used here, in this case, microscopy as a readout and systematically compared the stroma drug response, or the drug response in the stroma context versus the monoculture and drug response. You can nicely appreciate here the red dots illustrate all the drugs which don't work so well in the context of the stroma co-culture model, but overall the in vivo prediction of drug response was not better if we compared that directly to the monoculture situation, and that is obviously much more labor intense to be performed. So it might be different if we speak about immunotherapies. And you have heard quite a bit of this. If you want to mimic immunotherapies ex vivo, you obviously have to include the effector cells, because you're not directly acting with the drug on the tumor cells. You need the effector cells, and a minimum of such a culture model would also involve the T cells. And a prime example of these is the bispecific antibodies, which on the one hand target the tumor cells, on the other hand target the T cells. The proximity of tumor cell and T cell then leads to the T cell activation, and the T cell kills the B cells. So we established rather simple culture models here for lymphoma biopsies from lymph nodes. This works quite well. If we add the bispecific antibodies, you see this type of clustering in the culture model. We can nicely quantify this here with our microscopy technique. We see that the B cells are killed, the T cells massively proliferate, and this is actually what we expect is also happening in patients if we treat patients with bispecific antibodies. If we do that ex vivo, you see a broad spectrum, a significant variance of drug response or response to bispecific antibodies across different diseases and individual patients. And we also try to explain that with the type of T cells infiltrating the lymphoma. We run a big FACS panel on these samples. We also profiled on the drug response level with bispecific antibodies, and we were nicely surprised to see that, for instance, the number of Helios positive Tregs significantly associated with non-response to bispecific antibodies. And this can be explained quite well because, as you know, Tregs are immunosuppressive. I think for me as a clinician, it was at that time quite difficult to understand the very difficult situation of the classification framework of T cells. So when you start up to read about T cells, there are multiple names and potential sorting strategies around, and then there's a mix up of human and mouse, and that's why for lymphoma patients we decided to come up with our own classification framework. And with the use here of site sequencing, we could identify very robustly 14 different subsets of T cells in human lymph nodes infiltrating lymphomas and reactive lymph nodes. And a very surprising thing for me was that not the T cell itself is significantly altered by the lymphoma. So the transcriptional profile remains rather stable. But what is significantly altered by lymphomas is the frequency of the different subsets. For instance, you see a massive expansion of cytotoxic T cells, of clonally expanded cytotoxic T cells in diffuse large B-cell lymphoma. In contrast, you see an expansion of Tregs and T follicular helper cells in follicular lymphoma and marginal zona as expected. But you see many more naive T cells in reactive lymph nodes, which are only affected by infections, for instance. So just with the help of the T T-cell fingerprints, we were able to robustly distinguish between the different lymphoma entities, and just based on the frequencies. Then, of course, the next question is, why do these lymphomas have so many different frequencies of different T-cell subsets? And the reason is rather simple. So these T-cells recognize the tumor, and these lymphomas secrete a specific cytokine cocktail, and this either leads to maturation into the direction of cytotoxic T-cells or into the direction of CD4 positive T helper cells. And this expansion is mainly driven by the fact that these T-cells recognize most likely a tumor antigen and thereby clonally expand, as here demonstrated by single-cell T-cell receptor sequencing. Then you can order, for instance, these cytotoxic T-cells along the maturation path from naive T-cells all towards effector memory T-cells, and you can nicely plot here the markers along this trajectory. And you can here recognize all the exhaustion markers, and you can appreciate that these T-cells continuously recognize the tumor antigen and at some point show an exhaustion profile. And this is usually associated with a poor prognosis in these lymphoma patients. So all of these markers are pretty well established and known. The only one which is not so well established, at least in the context of lymphoma, is CD39. In our hands, it was a powerful marker, identify exhausted T cells in our lymphoma samples, nicely associated with survival. And I'm highlighting this because this is a potential therapeutic target. There are blocking antibodies around for CD39 and also its binding partner CD73. And if you inhibit these binding partners with monoclonal antibodies, this is a perfect combination partner for bispecific antibodies, for instance. So this is also an interest in our labs. We're trying to use available drugs and combine them in the right way to achieve here a better efficacy. And I was really surprised that one of these screens delivered a hit here. In this case, the combination, the most efficacious combination was bispecific antibodies plus CAR T cells. I was a bit surprised initially because in the end, the effector mechanism at the tumor cell might be the same. So you have the T cell which secretes factors and then leads to lysis of the tumor cells. But this was by far the most efficacious combination in our hands, and this could be also nicely validated in vivo models. And I think the reason why this is the case is because if you have a big tumor mass, the CAR T cells might not be sufficient to fight all the tumor cells. And if you add the tumor infiltrating T cells into the fight against the lymphoma cells, you have more T cells to kill the malignant B cells, and that together works better. And you can see here, if we use this together in mice models, we are able to cure a significant proportion of mice, which was not possible with conventional CAR T cell treatment, CD19 CAR T cell treatment, or bispecific antibody treatment alone. And based on this finding, we are currently initiating a phase 1 clinical trial where we use the bispecific antibodies as an induction treatment, followed by a CAR T cell treatment and bispecific consolidation. So, as all of you know, CAR T cell treatment is a very powerful treatment approach specifically for lymphoma, but there's a substantial proportion of patients not benefiting from this type of treatment. And so we also here set out to understand what mechanisms contribute to the resistance of CAR T cells. And one interesting finding here from this approach was that the number of myeloid cells in lymphoma biopsies from patients who have had undergone CAR T cell treatment were associated with response to CAR T cell treatment. So the more of these myeloid cells were present in lymph nodes, the poorer the response to CAR T cell treatment. And a number of mechanistic downstream work in Roland Ulrich's lab from Cologne has nicely also pointed towards the mechanism of this. So these myeloid cells secrete prostaglandins. These prostaglandins have an immunosuppressive effect on CAR T cells, and these prostaglandins can also potentially be blocked. And this is very interesting because in parallel we observe that if a lymphoma biopsy or if a lymphoma is under hypoxic conditions, these myeloid cells are recruited into the lymphomas. And hypoxia is another disadvantageous factor for CAR T cells. If there's not enough oxygen around, CAR T cells obviously cannot proliferate. So this is another factor why CAR T cells, at least in big tumors, are potentially not efficacious. And we used here one approach to improve or to give CAR T cells an advantage. We cloned myoglobin, the muscle-binding protein for oxygen, into the CAR T cells, and then we provided an advantage to these CAR T cells because this myoglobin has a high affinity to oxygen, even much higher than hemoglobin, and CAR T cells in this case were more active and could here more efficaciously eradicate lymphoma cells and other solid cancers. And here you have to believe me, this is also blurry because of the transformation from Mac to PC. But if we, on top of the equipment of CAR T cells with the myoglobin, also knock down the prostaglandin receptors, we can further improve the efficacy of CAR T cells. And this is also currently being prepared for a first-in-human phase 1 clinical trial. So I think it's fair to say that not only tumor intrinsic factors, but also factors contributing to the microenvironment are an important factor which have an influence on drug response. I just want to highlight at the end one further potential direction, and glimpses of that have already been mentioned in different other talks beforehand. The idea that the spatial context also gives you some glimpse about the functional context of a tumor. So I want to just give you here one example. This is the example of lymphoma. So on the left hand, you see the healthy context. So in the lymph nodes, so the germinal centers are growing very nicely. They expand because the lymph node is fighting with an infection. If you look at follicular lymphoma, these follicles are massively blown up, but the infrastructure and the spatial layout in these lymphomas is completely different. And if we talk about diffuse large B-cell lymphoma, the spatial context is completely lost. And you might agree that also neighborhoods and cellular neighborhoods are completely changed from the normal context through the indolent follicular lymphoma all towards the aggressively growing diffuse large B-cell lymphoma. And this gives you an indication or this could also serve, in my opinion, as a functional readout. And what Felix and Anna, who is here in the audience, a very talented student, did is they linked the surface proteome and the receptor ligand associations with this spatial context, with the idea to use this receptor ligand interactions to predict the spatial context. And that worked surprisingly pretty well. And that tells you that you can use something like the transcriptional profiling to predict spatial context, but you could also use the spatial context as a readout or as a functional readout for the tumor. And I just want to give you here one example from Gary Nolan's lab, the inventor of the Codex technology, as a final slide. And what they've done here is they have used the distance between T cells and lymphoma cells as a predictor of response to immunotherapy, also highlighting that the spatial context may serve as a functional readout for lymphomas. With that, I want to thank all of the people who've generated all of this data, for your attention, and I'm looking forward to your questions.
Thank you, Sascha. Any questions? Yes, in the back, please clap up the silver thing and push on the button. Well, you need to clap up the silver, pull on the silver thing and push the button.
Just a general question. When considering the beta inhibitors for lymphoma, did you observe any loss of beta mutation during the course of treatment when they develop resistance while you do the in vitro screening?
Yeah, I think hairy cell leukemia is in this respect very specific. It's different from melanoma and other tumors. In melanoma, you see in many cases a subclonal mutations, or at least there are also tumor cells which are negative for the BRAF mutation. In hairy cell leukemia, this is different. This is a stem cell disease, and the BRAF mutation occurs in stem cells. So that's why this mutation then commits the stem cells to mature into B cells, and that's why we see that the hairy cell leukemia is in 100% of the cells positive for this BRAF mutation. So that's why we thought this is the ideal scenario to just give the inhibitor, eradicate the disease, and then these patients are good forever. And we were very much surprised to see that patients respond nicely, even if they don't respond to chemotherapy. But if you stop the inhibitor, the disease comes back quite quickly. And that's because the pool of stem cells cannot be eradicated, and then you have a persistent clone in the bone marrow. At least in almost all patients, we could find this clone. And one reason why this happens is because they receive input from the tumor microenvironment, and then we see other alternative signaling pathways contributing to the survival of these cells, despite the inhibition of the BRAF inhibitor.
So you're now giving aspirin to the CAR T-cell patients, or ibuprofen to inhibit the prostaglandins? That would be something simple to do.
Yeah, I think it's specifically, it's locally secreted. I'm not sure if this helps, but what we can show is if we take the CAR C cells and use the CRISPR-Cas technology to knock down prostaglandin E2 and E4, and we have a manufacturing process already in place to do that, we can render these CAR T cells insensitive of the effect of these suppressing myeloid cells.
Okay, I see no further questions. Thanks a lot, Sascha.
Okay, I think it's time for the last session, which I have the pleasure to chair together with Virginie Ullmann. It's on data, but actually the whole day wasn't data. So from that point of view, I think if we talk about precision oncology, it's all about data at the end. And nevertheless, it's a great pleasure to introduce the first speaker of this last session, Michael Krauthammer from Zurich. We have a long-standing interaction with regard to some of the Loop projects, mainly the biomedical informatics platform, but also the AI tumor board, which is one of the projects he's going to present. Michael he's professor at the University of Medicine. His group is associated both with the hospital and the University of Zurich, working on bioinformatics, machine learning aspects, etc. And he's one of the co-PIs of a Loop project, which is called AI Tumor Board, and he will tell about this project. Please, Michael.
Thanks so much. Thank you, Markus. Good afternoon. I'm very happy to talk to you about the Zurich AI Tumor Board project. It's maybe something a little bit different from what you heard before. It's about a very practical project, how to bring AI into the clinics. We heard about the promises of AI in the morning by Daniel Truhn, about all these promises, but what does this actually mean to help oncologists in daily practice? And that's what this project is about. That is what I tell you about today. So we have a couple of goals in the Zurich AI Tumor project. The most important one is to bring a decision support tool into the tumor board that facilitates treatment selections for the oncologists. And that's one. And then another thing is it's cross-institutional. I think at Zurich we're making really big progress in interinstitutional collaboration. We'll tell you more about this later, but this is a collaboration in the University Hospital of Zurich at the KISP, the Children's Hospital, And where do we want to use this tumor board? We want to use it in both standard of care practice and beyond standard of care. And I will tell you more about standard of care and beyond standard of care later, but one is more about guidelines and one is more about data, about omics and other modalities. What are the key technologies that we are aiming at the beginning? First off, we want to, and that's very obvious, and that's within the standard of care setting, we want to help oncologists figure out which is the right guideline. And it sounds very trivial, but as you will see, it's not, and it's a AI-heavy enterprise. And second, we want to help them more in the beyond standard of care setting, figure out in a counterfactual way what might be an optimal therapy for the patient. Let's start with what looks like a trivial problem. So you have medical guidelines, you have patients, and you kind of want to figure out how these two go together. So first off, these are often living guidelines, and that already makes it obvious why you might need AI. So the guidelines are changing all the time, so they're updated, and so AI could potentially just update its own knowledge base all the time, something which is very hard to do manually. It's often multimodal in the sense of there are lots of different sources. There's ESMO, NCCN, there are PDFs, there are websites, and it already sounds like an agentic problem. And we want to solve two major problems. We want to find the right guideline, but we also want to find within the guideline what should be done with the patient. So the sources are all over. They're from ESMO, for example, and they're across different entities, colon cancer, lung cancer, melanoma, and so forth. And you would think they're all machine readable, but they're not. These are all PDFs, which sounds so like last century, but that's what it is, right? We don't have machine readable, or very rarely we have machine readable guidelines. So these are often just PDFs. So to make them machine readable, it's not easy. I think here you see a gastric cancer guideline, very complex paths here, and you kind of have to make them machine readable. What I mean by that, each box here has to become an entity that has to be understood by the computer. There's linkage between the different entities, and so this whole path, and we want to make this in a graph, in a JSON graph, and it's not simple. So you need OCR, you need LLMs and others to help you. And there are different things you need to do. You need to create different blocks, add labels into the blocks, add edges, remove redundant blocks, compare with the image, and so forth. It's a lot of work. And to do that manually takes a lot of time. So we enabled an LLM to do exactly these actions in an agentic fashion, so they can do all these different steps. And we built a tool where you can upload this image, right? You have the first optical character recognition. That's just at the very beginning, and that's all fed into an LLM on your left here, which parses this and gives you a first example tree, and then you can correct it. You can interact with the LLM and say, hey, you're missing this one block. Please add an edge, and so forth. And so it's an interactive tool where AI has a huge role, but in the end, we want to also make this in a way agentic in the sense that the LLM proposes something, corrects itself in a kind of learning fashion until the right pathway has been established. So we've done this exercise now with 4 main cancer types with 20 sub guidelines in gastrointestinal cancer, breast cancer, and so forth. And you can see there are different guidelines for each of these entities. And so far, I mean, that's kind of where we are. So we have this kind of authoring tool, AI-powered. We have 132 machine readable flowcharts that we derived from 20 guidelines and that are cross-verified and parsed. So that's the first step, which is already kind of a huge accomplishment given a relatively short time where we did that, and LLM here, or AI, and we heard it, had here a big role. But now that's just the first step. Now you have to match individual patients to these guidelines, and that's the patient guideline level. And that seems trivial, and is kind of, so you figure out from all these guidelines which one is the most appropriate. That's already a potential help at an AI tumor board, right? The second level is much harder, right? So then you have a patient with some treatment history, and you want to figure out where in the guideline the patient would fit, right? I mean, there might have been an earlier treatment, right? So you have to follow a certain branch, and then you have to figure out, okay, now you're in this branch, and please, you have to follow that part of the guideline. And so that's harder. And we did, and there are a lot of choices to be made, right? The AI has to figure out kind of that you have to go to the left branch or the right branch, and lots of things can go wrong. And we did some early testing, and so with 100 patients and how many mapped to guideline, that worked pretty well. But on the individual blocks, that's still very hard. Only a minority. In a minority of the cases, could we map all the treatments to a block in the guideline? Often there are many treatments where we couldn't find equivalence in the guideline. That might be that that these patients were treated outside the guideline. We don't know fully. But what's cool is with those where we could map them fully, sometimes we could map up to 8 treatments into the right blocks. So clearly things are working, but need still a lot of refinement. Yeah, so that's with regard the guideline mapping, and I want to now move on to the more also kind of important part where you also choose the treatment. And here we believe that the AI can provide evidence to make the decision. So it shouldn't make the decision, but it should provide the evidence. And I think that's a very important slide for me. I feel like the AI should be a partner, should be a discussion partner to figure out kind of what is actually available for this patient. And it's not necessarily a decision engine that is autonomous. And for example, the AI could tell you based on a lot of evidence, like routine data, multi-omics assessment. We heard a lot of these technology this morning and this afternoon, preclinical drug testing and so forth. This gives you already a ranking, and maybe a preclinical drug screen that tell you, well, this kinase inhibitor might work best. But the AI might tell you, well, but you might have a lot of side effects. Or I can't explain to you why it works, but I have this other therapy which has an okay outcome. I can give you a very good explanation why it might work, and the side effects are low. So I do believe we should build such kind of interaction tool, a discussion partner where we can form these decisions. So, and there's a lot of things we can provide the oncologist: the outcome, side effects, uncertainty. How certain is the AI? How much explanation can it deliver? Costs could be important, and if we can bring in patient preferences, I think we should also look at that. So, the source. I'm just going back. The source we want to look at first is routine data, and here the idea is that it's all about finding a similar patient in your vast archive, and you ask yourself a bunch of similar patients. What treatment worked for them? And so that's what we are approaching. We have right now a database of about 50,000 patients with lots of data. Much of it is structured, much of it is unstructured. And I just want to talk quickly about our efforts to go from unstructured oncology reports to structure, again using AI. So here we obviously have a lot of choices these days. There are a lot of open source language models we could use. For those of you who are not that familiar with, that there are big models, like 90 billion parameters, or the very small models. And the things we looked at first, our first approach was to look at small models, like little brains, that can be easily installed at hospitals that might not have the infrastructure to host or train big brains. And so we looked at smaller models and to deploy them at the University Hospital of Zurich. So that's our first approach. So these Llama 3.2 or 1 billion, 3 billion models, but not the 90 billions. So We worked on this in-house large language model development of small models. We published on that recently. And the problem with these small models is they are not very domain specific when you just download them. So we call them, we instruct tune them. So we build instructions to teach them about, or reteach them potentially about oncology, medicine in general. So we built this instruction data set, which you see here. We told them about medicine, about cases and the answers. And so we retrained them and then evaluated these small models. So these are small brains, again, kind of retrained on medical knowledge on USTED data. And so we looked at diagnosis and TNM status from pathology. And, you know, the results are so far promising. I mean, it's not perfect yet. And we can, maybe that's a discussion point later, how much precision do you need? But, you know, with some tricks, small models fine-tuned with RAG and other technologies have quite good performance in extracting these entities. But that's only the beginning. Once we know that the tumor stage, we know which treatment they were on, the more important thing is progression. That's the key point you want to extract from this data. Did the patient progress on the treatment or didn't he progress? And here we work a lot, and we just published on some algorithms to do that. So we're looking at the treatment history and when there was a change in treatment, and that was a surrogate for progression. And doing that, we have everything together. We have all the data from our historical patients. We know the progression, and we can ask which treatment worked for which types of patients. And I think we're getting very close on providing this type of information for the historical cohort. All right, so that's the key technology we're working on so far, the guideline mapping and the predictive analytics. And I want to now just move a little bit into the limitations here and the future. I think we saw the video this morning by Truhn, that these guys playing soccer, and the IT person kind of getting, having some problems with the soccer ball. Yes. The problem here, I think what this video was all about was you have all these great ideas and we're cooking up these cool AI routines. But when you want to move them into the clinic, we're having all these hurdles. And I'm very interested in that. I think we are approaching that on the UMZ, University of Medicine Zurich level. Like, how can we build some sort of framework to facilitate the going from the development into the clinic? But you have to realize the organizational readiness is often very low. You have to talk to at least 15 people from, IT, ethics, governance, the physicians, the patients. I mean, just to make sure that the tool that you're building has any value and actually can be implemented. So really hard to get the organization be ready for this type of tools. The infrastructure has to work. I told you about the problem with the large language models and small language models. The data has to be ready, and the business value, and I guess it's the most important part. You can build the most fancy tools if the oncologists are not going to use it. So figure out what's the really, really key problems you want to solve. And so we're working on all of that, but it's hard. And I just want to tell you about one infrastructure. I think maybe Markus has mentioned it this morning already, that will be helpful for this. Here in Zurich, we're building a ecosystem. We call them the biomedical informatics platform, to bring together the data from the KISP and the University Hospital of Zurich and others into a common blue secure space that can drive the tumor board in a hopefully also kind of real or semi-real-time fashion. And this platform will soon be available to researchers here in Zurich, but hopefully also to collaborative groups, with maybe some of you that came from Germany or from Holland. I mean, this is data that we hopefully can work on together to analyze. And it's not just oncology. If you're in renal diseases, I mean, there's lots of data to explore, and we'll have a beautiful portal to access this data and to make the data analytics available. Okay, I just want to take the last few minutes on the outlook. The first thing is clearly guideline mapping is an important tool. Looking at historical cohorts is a very important issue, but we can have in a modular fashion other things feeding into the tumor board. And we are very lucky to got funded by another funding instrument to build a preclinical testing suite. Some of it is already existing at the University Hospital Zurich and at the Children's Hospital. So these are preclinical testing facilities, either with organoids or with cell screens. And the idea is that patients that go through this testing will also have the preclinical data available in the AI tumor board. So you will not only have data from genetics or lab and other things, but you will also have preclinical testing that will go into this dashboard for oncologists to decide kind of what could be the optimal treatment. And I think that type of integration is key to make this tumor board really kind of available to different stages also of the decision-making process. And this modularity, I talked to Gunnar Reg here also could be extended to H&E slides from pathology, right? We could have a tool here that looks at H&E slides, maybe does some AI type of staining, also makes this available to the AI tumor board. I think to build this modularity allows us to be really more open and flexible about the future direction. Another direction, and that's really driven also by Fabio Steffen and the KISP crew, is to include PubMed, or at least the literature or the realm of the knowledge that is out there, into the AI tumor board. And so the idea of, for example, if you had this preclinical drug screening program integrated, you get this list of ranked drugs, right? And then you want to figure out which one would now work for my patient. And then you send this information to the cloud, to maybe Gemini. We're working with Google in this particular case, partly, and figure out kind of how should you re-rank this drug? So which would be the most optimal drug for this very patient? So that's something also we're looking at, and that should be integrated too. And this can be done very nicely and modular. And finally, I want to say that we're not working alone. I think on a national-wide scale, we have a lot of things going on. I just listened to a poster before of a group which does prediction for single point mutation, whether it's actionable or not. So we could integrate these types of initiatives. But there's a national initiative called NIPO, or National AI Initiative for Precision Oncology, which has overlapping goals with the Zurich AI Tumor Board. And there will be new dashboards from within this consortium. And I hope that all these developments in a synergistic fashion can really lead to a very robust product in the hopefully not so far future. Right, so I want to wrap up with what is the Zurich AI Tumor Board. It's a dashboard in the middle that helps physicians on the lower right, to make decisions. And we will introduce guidelines and help them find the right guidelines faster, right? We want to find similar patients from real-world data. We want to do predictive modeling of that data. We'll have preclinical drug screening from different platforms, hopefully PubMed integration to sort out some of the uncertainties in all this data and bring this all together for the optimal decision making. Now, there are a lot of people involved, and there's not even all of them are shown here. I just want to mention a couple of them. Andreas Wicki group in USZ Oncology, is Nicolas and Laura. Then from my group, and partly also from Andreas's group, Scholt, Matesa, Thibaut, Irene, and Bowen. And then very important from the KISP, Jean-Pierre. I'm very happy for this collaboration. It's been really fun, and I'm learning a lot. And Fabio, a brilliant scientist at the KISP. And then also what's very important, we have super support from the Swiss Institute of Bioinformatics with Anna Sima and Ruche Wang. They actually built the guideline mapping tool. And I think this type of collaborative efforts will hopefully lead to a successful product. And I'm very hopeful, but there are lots of obstacles. And yeah, that's all I wanted to say. Thank you for your attention. For listening to me. Thanks.
Yeah, thank you, Michael, for this overview about the AI tumor board, which was very interesting, I think. We have ample time for questions, so any questions from the audience? Please.
Thank you. I have a question about the clinical guidelines. I mean, there's not only one guideline, there's multiple guidelines, so there's many different societies. So are you planning on kind of building a tool which can translate multiple guidelines into hospital-specific SOPs and thereby advise us how to proceed? Because if I remember, for example, in lung cancer, in gynecological malignancies, it usually takes about a year to collect the guidelines and transform them into an SOP which applies to your hospital environment. Can we have some support of AI?
Yeah, absolutely. I'm totally aware of that we're skipping this at this point, right? We're going directly to the guideline, which is maybe for some of you who are not that familiar. So that is kind of prototypical guidelines that are published, but then each hospital or each group has their own SOPs, which roughly is that guideline plus or minus. It depends, right? And so, and they are handcrafted, and I think the same tool, right, could be used to do exactly that, right? So you remember that kind of flow chart that we're building, right? We could essentially say, you don't have to do a one-to-one mapping, right? It could, you could build your own flow chart that is corresponding to your SOP. I mean, but there might be other ways to think about the SOP. But I fully agree with you. That's maybe a step we should also think about.
Because you do know what is available in the hospital. It depends what diagnostics do you have, what are the therapeutic options you have. So you have the knowledge what is being available in terms of resources, and you know what are options on the guideline. Mapping these two sources would be super interesting.
Good, yeah. Okay, it's very good input, and we're looking into this. Thank you.
Torsten, first.
Thanks a lot, Michael. Yeah, so following up on Matthias' comment, so to me, the guideline is more like a pick the winner. You pick the best guideline that applies to where you are. So to me, it's not entirely intuitive how you're digesting multiple guidelines and extracting something. So what would be the rules that you want the AI to apply?
Yeah, I mean, I had one picture where it was unclear, right? I mean, where you have choices. So if I understand you correctly, there could be competing guidelines.
Well, they're not so much competing, but usually, like, experts will have their favorite, for good reason, will have their favorite guideline. So and then there's the time part that these are, by the time the old white men, if I can say so, agreed, like, and that is in press, it's already in part out of. Time flies. So to me, like, the strategy is, like, identify the one guideline that you think best fits a situation, and then that's it. There's probably much more likely than integrating multiple little slices from all the guidelines into one kind of local AI tumor board guideline. So to me, it's not entirely clear how that AI tool will address these realities that we have.
Yeah, I agree. I mean, I hear problems here, and I mean, again, there's two levels, right? Finding the right guidelines and then dig in, and I'm with you, right? And so we need to have studies with someone like you early before we fully build this thing, right? To say what's practical, what's not practical, what's working, what's not working, and I'm standing here not knowing all the what for example you would like to see, right? But right now we have somewhat a naive approach, right? That you have a patient, right? You can find the right guideline and you figure out kind of where you are in the treatment. If this is not helpful, right, then we'll find that out quickly, right? Yeah. But thanks for the input. Yeah.
Good, Reg.
First of all, thank you very much. So it was really good to see the overview and the different developments. So that's really exciting. and also I agree with the NIPO. I think there's a lot of potential we can collaborate. So I think I fully agree on this point that we need this modularity, right? So we need to think about how we break the different pieces of information down into maybe modules which can talk to each other. Because in the end, probably we need to deploy this to different infrastructures, IT infrastructures, so they're all different, right? So, and we need to modularize this. We need to make sure that these modules can run in different places and so on. So if we would build one monolithic system, I think this would never work. We could never transfer this to another place. So, so my question now is there a good way to standardize these modules? So have you thought of that? So,
right. I mean, it's a very good question, and so we haven't drawn the full architecture right now. We also have the help of Nexus, so we have good engineering knowledge. I think it's also connected to certification a little bit, right? So which part of this do you actually want to certify? And I think that again goes. I would speak for modularity. For example, that you would certify just some small AI engine, right? But how you display it, you wouldn't. And so I think we have to think it through, right? I mean, where do we want to go? What has to be certified? And that will drive also in the end the architecture to me, right? Because you want to obviously isolate what you have to certify from the rest, right? Yeah. Exactly. Right. 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 So I agree. So maybe if you didn't hear it online, the information flow might not have to be certified, and I think that's what I hear from experts. And talking about certification, I didn't talk about it here, but we want to see whether we can have a first version of this just outside the certifiable domain, and then go on to certify key elements of it. So that's kind of the plan right now.
Probably there is a question from the chat which says, thank you for the interesting talk. Besides PubMed, do you plan to integrate clinical key AI, which is also widely used as an external source?
Yeah, I would love to have. I mean, I guess that's about the chatbots, like not just PubMed. Yeah, I guess so. I mean, that would make sense to me, right? That that somewhere Gemini or some of the other chatbots would digest this information. So yes, we plan to do that. Again, here we have the problems about sending data into kind of non-Swiss clouds, and believe me, it's a big headache. So yeah.
Thank you, Thorsten. As a question.
Yeah. So I think a very useful addition to the program could be the dealing with the label, like indications for the treatments you recommend. And we usually have to, for some of the drugs, we don't need to ask for permission. For some, we need to write to the health insurance, and that seems like a very kind of very well structured program. Right? There's the label, there's the Spezialitätenliste,
yes.
which
got it.
To answer the question whether something's reimbursed. And this to me seems like something that AI could very easily do, and this would have a huge impact on our time. And I would encourage all of us to then read the guideline independent of the output. So I think there's some additions that would be extremely useful and save a lot of time.
Right. So it's interesting, and I also think that again shows that this tool might not be used at just one time in the boards, and it could be used after the decision, and then it could help you write the justification easily so you don't have to look up all the sources and write the justification by yourself. I fully agree about that. Very good input, and I think that would fit very well. Thank you.
Okay, thanks. I hand now to Virginie.
Great. Yeah, so I have the pleasure to introduce our last speaker for today, Tero Aittokallio, who is joining us from the Oslo University Hospital in Norway, where he's a professor. And there he is leading computational biology and systems medicine research to develop predictive models of drug combination responses and to integrate multi-omics data for precision therapy strategies. So He will tell us about predictive modeling of drug combination responses, and I'm really looking forward to your talk. Thank you for joining us.
Thank you very much for the introduction, and thanks for inviting me here. And thank you, all of you, for staying here until the end. It's nice. So yeah, my name is Tero Aittokallio. I'm telling what we are doing in Finland. Mostly this is about Helsinki, predicting drug combination based on the single cell data. Here are my only disclosure at the moment. So this is a setup. I don't think I need to introduce this because you have seen similar setups, but this is what we've been developing in Helsinki for the last 15 years. Started by Kimmo Porkka and Olli Kallioniemi, and then many of us further developing it later on. So just briefly that we are getting bone marrow samples from the typically AML patients. My talk is only about AML, but all of these methods, since they are computational methods, they can basically be also applied to other type of cancers. So we get the bone marrow samples from the patients, we test them. At the moment, our drug library is more than 600 compounds. And then from the same patient samples, we also do the genomic profiling. And of course, increasingly nowadays, single cell profiling. So I'm mostly focusing on single cell transcriptomics here. And of course, we are not the only one who is doing this kind of like functional precision medicine. If you are interested, this is a nice perspective article from 2024 describing many of the successful cases, how this functional precision medicine is being used globally. But there are, of course, challenges also, and these are the 3 challenges that I briefly try to address today. So the first is, like we have heard many times today, that the cancer and tumors, they are not homogeneous. There are subpopulations that may lead to different treatment responses. So how to model that and use that in the treatment optimization. And then since I'm talking about drug combinations, what we try to do is then that we try to find combinations of drugs that are inhibiting each of these clones that are driving the disease or the resistance mechanism. And the third challenge that I think is the most challenging one that has been also addressed today, briefly at least, how to find biomarkers to make predictions why this particular combination is working in this patient, but not in the others. In combination therapies, we see a lot of heterogeneity there, much more than in the monotherapies, even with from between the patients with the same disease, but also the same genetic mutations. There are nice examples. This is a Venex trial that is run by Mika Kontro in Helsinki, where they are using ex vivo drug response as a predictive biomarker for venetoclax azadine combination for AML treatment. And what I liked about this study, they make very, very systematic study and investigation what assay type is most predictive of the clinical response. And what they came up was that you should use blast-specific responses only venetoclax. So they use only the blast-specific venetoclax response to predict the clinical response to this combination. Adding this azadine didn't help at all when making the clinical predictions. And another thing that I like about this study, that they came up this threshold for the DSS, that is drug sensitivity score that we have developed in Helsinki, that is around 10. And that's what we've been using also in the past, based on only statistical reasoning. But they also showed that it's actually good cutoff to make clinical response predictions. So this is a nice way how to implement this in the clinic. Of course, you need to do the same thing, the same investigation for each disease, different drug classes. But this is also a nice example. But of course, what it cannot do, this Venex trial, is to understand or analyze why we see sensitivities or resistance of the patients to this combination. And then, perhaps more importantly, what might be good combination therapies for those patients that don't respond to this venetoclax azadine combination. And that's what we are trying to address using predictive modeling. I like this cartoon slide that my PhD student Kristen has made, just to emphasize what we are trying to do here. So we are trying to tackle, sorry, it's not the pointer, this right-hand side case, that we try to find combinations for individual patients, but also for individual clones or subpopulations of that patient. So it's, of course, highly challenging problem because we typically have in equal one cases, that we don't have too many cases with same kind of clones driving the disease and then the same combination. So this is really individualized combination predictions at the clonal level. We have developed, of course, a lot of tools how to analyze the combination data, drug screening data. So that's not the problem at the moment. We can test compounds together in patient cells and get flows, dose response of this kind of combination effect landscapes, like we call them, and then quantify whether there is synergy undergoing. So that's not the problem. The problem is typically that we don't have too many patient cells available to test like tens or hundreds of combinations. Typically, we can test in the patient cells perhaps top 5 combinations. If we are lucky, we have enough cells for 10 combinations. So the synergy finder we are using routinely in this kind of combination analysis. You can analyze 2 drug combinations, 3 drug combinations, higher order combinations that you can implement and test different synergy scores like please zip, lower. I don't want to go into details because there is ongoing debate what to use, but you can try them all with this tool. But of course, you should understand how they work. Synergy is of course one thing and that's nice thing because typically if you have synergy for the drug combination, you can at least in theory decrease the doses of the individual compounds, which might lead to less adverse error. But of course, that's not only one factor in successful drug combinations. Efficacy in clinical applications is much more important. And then, of course, toxicity, that we are not like having overlapping toxicities or dose-limiting toxicities for these combinations that we try to identify for individual patients. Like Berend said this morning, it's very easy to kill cancer cells. And with combinations, it's even easier. You just combine 2 very toxic drugs and then you kill everything. But how to make it selective, how to make it safe for the patient. So that's the more important thing. So how do we do it? We have developed this tool SC type that enables one based on only one single cell data matrix from patient sample to identify different cell types quite accurately and fast. How we are using is such that we try to identify what are the healthy cells, malignant cells, because then we try to find combinations for the malignant cells that are not inhibiting at least too much the healthy cells. But this is a widely used tool that you can use for other type of applications too. There is a nice database for different cell types that you can use. You can also update the database with your own markers there. If you have for your own cell type that you know that this should be included, you can also put that there. And we are actually now developing a new version of this C type, so it should be even faster and more accurate. But that's what we are using. So we get the samples, we do the single cell transcriptomics, and then we identify what are the cell types in that patient sample. And then we make predictions what might be good combinations for that individual patient. So just to summarize, these are the things how we at least define what are successful drug combinations, that are effective, they are killing the cancer cells, but they are not toxic, that they are not inhibiting too much the healthy cells. If you want, they can be synergistic. There are some benefits of that. But then the problem is how do we identify these combinations for individual patients? And that's the machine learning problem. All of these other things are just statistical scoring or experimental profiling. This last part here, how we identify few combinations for each patient that we can actually test then in the patient cell. So this was the first pilot study where we started making these predictions using both the single cell RNA-seq data and ex vivo drug responses. At the moment, or at that point, we used XGBoost, that is quite standard machine learning model based on the boosting machines, but you can replace with any of your favorite fancy AI model. But then what we do in the end that we get the based on specific drug combinations. Bit of more details. We get bone marrow samples. We do the drug screening for this 600 compounds. This is a CTG assay, so quite simple viability assay. Then we get from the same samples the single-cell transcriptomics. Then we have collected a lot of target information for those compounds that we are profiling, that we know what are the pathways, what are the protein targets. So we do enrichment analysis, and that we use as an input to the machine learning model and train it first to predict the monotherapy responses using the DSS scoring. But then later we use the exactly same model to make prediction of the drug combinations just by combining target sets of the drugs together. And that's how we get the input then for the combination predictions that is shown here. And then we do two different experimental validations because I don't trust any of the predictions unless we can also validate those in the patient cells. So we use the same patient cells that we make the predictions. We do the CTG validation, the flow cytometry validation, where we can go to the subpopulations and see how much we are killing actually blast cells and how much the healthy cells. And especially the flow cytometry takes quite too many cells. As you cannot do it for tens of combinations. So that's the limiting step, because we also do always this dose response matrices, that not only a single dose, but typically 5 or in this case also 8 doses that we test the combinations in these patient cells, just to see also where are these dose ratings where we see synergy, if there is a synergy. So this is basically what we did in this pilot study that was already published 21, so quite many years ago. But this is basically still showing the pipeline that we are using at the moment. So at that point, we had only 4 patient samples with single-cell data where we validated this model. And like you can see, they are very, very heterogeneous patients based on different genetic mutations, previous treatments. So they are not at all like homogeneous patient cohort. And this is something that we always see in the real world applications, that we really need to do it for individual patients. We don't learn too much from the other patients in the same cohort when making the machine learning predictions. So these are really like for individual patient predictions. And this is the clinical or treatment history, just to show you how this, that they have been treated with multiple different, these are typically very late state patients. And the same thing is that we have also now extended to these bigger cohorts. How do these predictions work? This is now for the CTG validation, just in the bulk-based drug response combinations. Upper part is showing this kind of common combinations that we predicted that came from multiple patient samples, the same combinations. And there we see that there is a pretty nice difference between those combinations that we predicted that should be effective compared to those that we predict or the model predict that should not be effective. The lower part here is that there are these patient-specific or unique combinations that we only predicted for individual patient samples. We still see a significant difference between the two groups, those that we predicted effective or not, but it's not so huge. And that's, of course, that is much more difficult to make these predictions for individual patients. But like I said, that's what we need to do because these are quite different patients. And why they are so different? This is just showing the UMAP plots for 3 of these patient samples. And like you can hopefully appreciate that they are different from based on their molecular profiles. We see even different type of blast cells in these AML patient samples. In some patients, we can identify NK cells, in others not. So that also makes the more contributes to that the predictions on the top are that the combinations that we predict are quite different for the individual patients. This is now based on the flow cytometry, and there we use the lymphoid cells as the healthy cells to quantify how toxic are these combinations. So there are two bars. I'm not sure if you can see. The upper part is how much we are killing the blast cells, and then the two lower bars are that how much we are killing T cells or NK cells, and that's quantifying the toxicity of the combination. So this is quite similar to what Berend was showing in the morning, the first talk, that you quantify both of these things, the efficacy of killing cancer cells and then toxicity to healthy cells. In each of these cases, we can always find a combination that is both effective in inhibiting blast and also not too toxic if we put the threshold, for example, 50% of the lymphoid cell killing. All right, this was already published some time ago, but I just wanted to show it how we are doing these predictions. But after this publication, we have done some nice further developments. The first one was that we don't use at all the drug response data. We just use the single-cell transcriptomics data to make the combination predictions. And the other thing is that we also wanted to test it in the solid tumors, in ovarian cancer, and using organoid models. And then the last thing that we wanted, of course, see also how much this correlate to the clinical responses, and that's where we are using Venex trial samples. But the first thing is that my two students, Alexandra and Tristan, they teamed up and came up with this idea that let's just forget the ex vivo drug responses. Let's just use the single-cell transcriptomics data to make the predictions. And when I heard that, I thought, no, that's crazy idea. I thought that you need to have the functional responses. But then they convinced me that let's at least try it. And actually, it is quite predictive, only the single-cell transcriptomics data. So this is how that model works. It's basically the same done earlier, but now we also identified the genetic subpopulations or clones in each individual sample. And then we start targeting the combinations to the major subclones there. Here, since we don't have any more the drug response data, we need to pre-train our model. In this case, it's a light GPM model that we were using to make the predictions. So we collected, or the students collected a lot of data from links, that is the transcriptomics response profiling in the hundreds of cancer cell lines. Then we had the structural similarities to compounds, and then we also collected a lot of viability data from the pharmacodp. So pre-training the model based on that, but then we always apply to the patient samples based on the single-cell transcriptomics data, and then make the predictions of what might be good combination therapies for that individual patient. In this case, we had 12 AML patient samples where we had single-cell data. Again, the same thing, very, very heterogeneous. The UMAPs profiles, the gray points here, I think, are the blasts. So even those are quite different between the patients, not to speak about the other cell types that we see in these patients. We first validated with the CTG data because we are routinely doing this CTG profiling for all the patient samples in Helsinki, and this is just showing that there is again very nice difference between those compounds that we predict are effective and those that we predict that shouldn't be effective. And each of these points, it's individual compound and they are different between the patient samples. I think I highlighted here, for example, navitoclax has a red point, and it's showing different responses in different patients. But this is already based on the data that we have, but then we also wanted to do prospective validation of these predictions. So here we selected 4 AML patient samples that we had enough cells available, and then we did the infer-CNV clone identification just to find 2 major subclones in each of these patients, and then the normal cells, and then predicted what might be good combinations to start inhibiting those major subclones in these 4 samples. And this is the result when we did the combination testing again in matrix format. All of these points are different combinations for different patient samples, and like you can see, they show all positive synergy, meaning that they are at least additive They are not at all antagonistic. They are additive, and some of those combinations are even quite highly synergistic, meaning that the synergy score is higher than 10. And I find this quite impressive, that based only on the single-cell transcriptomics data, you can make predictions of clonal level synergy in individual patients. So I was quite positively surprised about this result. But then, like in the pilot study, we did the flow cytometry where you can then see the combination responses in different cell types. And again, we just compare here the blast cells and the lymphoid cells to show what are the toxicities here and our selective responses to the combination. Here's an example from 2 patient samples. And these are the different combinations that we predicted. On the right-hand side is the efficacy, how much we are inhibiting the blast cells. And there, in both of the cases, you can see that there is a nice, if not synergistic, at least like increase in the efficacy when you go for single drugs that are the first 2 bars there to the combination that is the red bar in each of these combinations. Whereas then in the toxicity side, which is again the lymphoid cells inhibition, we don't see this kind of like additive effect when we are adding more drug to the combination. And one more nice thing about this model is that in the parentheses there are also these numbers, so we can also predict what might be the effective dose for those combinations. Of course, this is a ex vivo dose that shouldn't be confused with the in vivo treatment doses, but at least you can get some kind of idea what might be the good effective doses for each of these compounds and in the combinations. Yes, and then we also wanted to show that we can go to solid tumors. It was much more difficult than I expected. It took more than a year. This was in collaboration with Christian Wennerberg's group in Copenhagen, but they used then the ovarian cancer organoids to also test predicted combinations in those patient samples. The problem with the organoids, like many of you know, that there is typically the outgrowth of the other cell types, so it's quite difficult, or you need to be very careful which time point you start adding the drugs there. But at least we could show that, again, we have nice difference between how much we are inhibiting the PAX8 positive cells that are the cancer cells in ovarian cancer compared to the PAX negative cells that are the healthy cells. We also had to use some fibroblasts from the other individuals just to make it selective enough, but at least we could show that you can use this same method also for the solid tumors. And that was the whole idea, because drug testing in the solid tumors is much more challenging than in the blood cancers. All right, but that was again only ex vivo combination response validations, but how about then the clinical response? So to do that, then we teamed up with the Venex trial team, with Sari, Tanya, Heikki and Mika, because they have already done all of this thing, that they have tested these compounds, and then they have correlated the venetoclax response to the clinical responses of the treatment. So what we wanted to do with the Venex samples and with this setup was, of course, that we make predictions how these patients would be responding to azacitidine venetoclax combination, and then we compare to the clinical response to that combination. This is what we published last year in Cancer Research. We had only 9 patient samples with single-cell data from the Venex trial, but already there we could see that there is a nice difference on the left-hand side in the predicted synergy score between those patients that respond to the combination compared to those that don't respond. At the moment, we are profiling, single-cell profiling. We have we are planning to do more than 50 patient samples with both diagnostic and relapsed and refractory samples in this, and to make this prediction again, and also to try to find new combinations for those patients that are not responding to this combination. On the right-hand side, just as a comparison, if we just use the CTG responses to these 2 compounds, we don't see any difference between these responders and non-responder groups. Just showing again that you need to have the single-cell transcriptomics data and the machine learning model to make these accurate predictions. So I think I start to conclude here. I think I don't need to convince this audience about the benefits of the functional precision oncology, but I try to do it anyway. So it's a good way and very fast way how to prioritize target and drug combinations for individual patients. And it also this patient-specific and selective combination. Instead of suggesting very toxic combinations, we can also go to the selectivity analysis, which I think is very, very important when we go to the clinical translation. And it's also fast. In Helsinki, we do 3-day drug sensitivity assay. Then we do modeling in my group that, depending on the student, takes 2 or 3 days. We get the predictions, and then we go again to the patient samples and do the flow cytometry validation. But less than 10 days, we get the combination predictions that are validated in the same patient samples to the clinicians, and they can or not use them in the treatment. But the problem, of course, is that then what to do with these patients that don't respond, that we both predict that they don't respond to this combination, and then we have also data to show that they don't predict. So this is something that we are currently trying to address, and we have a collaboration with Mobius Biotechnology and with the Phoenix team, where we are doing this single-cell profiling for more than 50 of these patients in the Phoenix trial, with the aim to make predictions and also compare to the clinical responses for new combinations, not those, because those are not responding to the combination treatment. So then we are making predictions what might be a better treatment alternatives for these. And then we are using both the ex vivo validation and also then some kind of clinical decision support system. So this is a project that is started this year. It's a 2-year process, so hopefully in a couple of years I have some results to show from this pilot project. But I think I'll end here. This is a big project in Helsinki. I think this is quite outdated slides, so I don't show it longer. But I just want to thank my group who has done all the work, and also other patients for providing the samples. And thank you for your attention. Happy to take any questions.
Great. Thanks a lot, Tero, for the beautiful talk. Are there any questions in the audience? Yes.
A great talk. Thank you so much.
Thank you.
We heard a lot about foundation models, how we can integrate data across the world to train models. Maybe, and just I'm a little bit familiar with the domain. I know that drug screens in different places are very heterogeneous. The synergy scores are not exactly compatible always, and so all these problems with replicability and so on. Can you comment a little bit? I mean, you seem to be using all the data that you produce and you train with your data, but would it make sense to look a little bit outside and include screens from other places? Or
Yeah, very good question, and that's what we are trying to do. So at the moment, these are the results that we have in the Helsinki setup, which is of course standardized, and it's the same. But currently we are combining data from 4 different AML cohorts from Helsinki, Oslo, Karolinska, and also PTML by Jeff Tyner. And of course we see a lot of like patch effects and differences, which are mostly because of the differences in the assay setup. So I have one student who is now trying to understand what factors in the experimental assays are contributing most to the variability. And if we can then standardize those factors in the next experiments, then I think we can get much more consistent data. But already I think with, I'm not sure if the foundation models or some other machine learning models, if we have good patch effect correction, we can start combining data and have bigger cohorts. But like I tried to convince earlier, these patients are very, very different. So it's quite difficult to learn like predictive signal from the other patients, especially if they come from the US compared to the Nordic cohorts that we typically use in our machine learning. But I think we should at least try to do it. I'm not sure if it's successful or not.
Perhaps we have time for one last question, Ms. Dustin.
Yeah, so thanks a lot, Tero. So in the clinic, sometimes we have no trouble combining 3 drugs or 4 drugs. So you know, is this technology expandable to multi-drug combination?
Yeah, I think so. We have tested like 3-drug, 4-drug combinations, but then we cannot have too many doses because otherwise it will explode logistically in the experiments. For prediction, no problem. For scoring the accuracy, no problem. But the testing in the patient cells, that becomes quite difficult. But like the Venex trial was showing, and this is something that we discussed yesterday also with Fabio, you might not need to have all those combinations or all the drugs in the combination tested in your ex vivo assay, even if you are predicting like 3-drug, 4-drug combination. It might be like Venex trial, they just used the venetoclax to predict combination response. It might be that you only need 1 or 2 drugs of the combination and test those in the ex vivo, and still they might be predictive of the combination response. But like with the Venex trial, you always need to design that study for each combination individually. So difficult to say.
Great, thanks a lot. So I think that we are on time. Let's thank Tero and the speakers we had in this session again. And Thorsten has the last word for today.
I will be brief. It's nice to do a short wrap up. I had a great time. I had an exciting day. I hope all of you feel the same way. I want to thank the speakers. Thanks for coming and presenting and discussing with us. Warm thanks to the poster pitchers. I think this was a great part of the meeting. Silke is always our pro in organizing these meetings. Thanks a lot for helping us. This is a cooperation effort from the Loop Zurich, from the Comprehensive Cancer Center. You saw the Vice President for Research, so it's a I hope you can, for the people from the outside, can feel some of the energy of the people who are working here. And I think with that, I just want to mention some of the sponsors. So we have pharma sponsoring for this particular meeting, but we also had this final session, which is a cooperation with Grand Round, which is a educational program we run in our department that's also funded, and this was a good opportunity to co-host this. So thanks again to all of you. Have a safe trip home. The weather is not great. The weather forecasts are very good and very crisp, but from what I learned today, we're moving in that direction with discovery data and the ability to predict patient outcomes. So thanks a lot for joining us.